sides, rocky, crusty area.
You want to look in this area for a while?
I actually think it might be a good idea, and I'm hoping by the time we identify a rock, I can just get you a number here.
Pretty round, basically.
Yeah, round and like 15 centimeters each. Ish.
Right next to the GoPro, I think. Just to the right of the GoPro.
Yeah. Same there?
Yeah, that's right.
Yeah, maybe something on top that's not too buried. Let me see what that pointer here. And I guess these look okay. Okay, awesome. Thanks. Or even this one looks beautiful right there.
Okay.
Lower right.
The one right in front of the rail?
Yeah, right there. Looks nice. So this kind of bumpy texture here, that's like ideal, but can I do some zooming around and see which one of these might be good?
Sound effects?
Can I do the sound effect too?
Totally worth it.
All right. Yeah, I like this one. It's about 15 centimeters. And it's bumpy and lumpy, kind of what we're interested in. So let's do. Actually, I'm just gonna wait a couple seconds and just poke around here while we get our sample number. I'll take an image of this sponge. But that's the one we're going to go for, I think. Got some squat lobster. This looks like a farreid glass sponge. So we've got some gastropods, crabs, ophiuroids, and then I saw over here a little, might be a polychaete worm or an aplicophoran. It's kind of nice too. Looks like an aplicophoran. Lots of squat lobsters right here. Once you start zooming in, you start seeing a lot more life. These are very nice too, like this lumpy, bumpy texture. Really nice stuff. But I like the other one. It's the right size. It's pretty compact.
Still your favorite?
Yeah, still my favorite. So can we do an in situ shot there?
Sure. No?
No? Yep.
Got it.
Alright. I'm gonna zoom out a tick. Put some lasers on it. Alright, can you get the glow out of the shot? Just for a second. Alright, hit it one more time.
Hit it one more time.
Yep.
Got it.
Alright. Now, you can open up the sample storage,
Okay.
and it's going to be a 15 centimeter roundish rock with sediment on top and polychaete worm, or just worm. And that's it for now. And then we will we'll do a we'll take a look around it after we pick it up before we put it in the basket to see if there's any other identifying features on it. All right, I think we're ready to go start up collection. 372 is our next number. Thanks. Yeah, sorry.
372. Rocks. Is it a rock box?
It's a rock box, rocks.
Okay.
Rock box, rock. Say that 3 times fast.
Okay, 15 roundish rock with sediment on top with worm.
Yeah,
That's
yeah. Yeah, it's nice. The underside, so just write this in the notes. The underside is uniformly dark with red splotch.
formally dark with
Yeah. Red spot. Yeah.
red spot.
Yeah, it's a nice rock.
Actually, hold that at the top.
Oh, yeah.
A little bit of that.
Now we just roll it in and
It's like following the
follow it in.
almost like it's one on top of the column, top of the rows.
Yeah, if you just balance on there.
Just so like every you go, you don't iron all the way, but it's
Drop the pen.
Yeah, yeah. I'm gonna roll it back up. Go back so you can roll it in my arm.
Sorry, I always pick the biggest rocks. It's not that big, but it's like it's almost perfect.
Actually, watch it out. Like this, but it's sliding on. Yes, that might make you.
It's fall off. It is the gray and water is so warm.
You're getting loaded. You can roll it in and down.
Pick it up around.
It's a fine rock.
Hey, I am going to be in the wet lab setting up another experiment. But if you need something, I'll be on WhatsApp. It'll be easier than
Yep.
Slack. That's what I'm telling you.
Okay. Yeah.
Um,
Yeah. Well, we're just it's a bit shallower than we anticipated, and so
Yep.
that's fine. And we'll be on watch here for another hour, and then next team will come on.
Perfect. And can I just say something? Because
Yep.
if you are listening, hello, my name is Randy Rugin, and I'm specifically saying hi because we just finished a ship to shore with the University of Guam. And you know, a lot of you are probably tuning in right now because we just chatted about it. So if you're here, I just want to say welcome to your first deep sea dive. We're so incredibly happy to have you. And if you have any questions, Steve and Anamede are here, and you are in very capable hands. I'm going to the wet lab to start another experiment. And now you know what it looks like because you were just there on a ship to shore with us. But for everyone else, I'm excited to be doing this first dive on Titov Island here, an island, goodness gracious, Titov seamount here in the Pacific Remote Islands Marine National Monument, Helen and Baker Unit, and we're very grateful to the U.S. Fish and Wildlife Service for allowing us to work here. In addition, grateful to NOAA's Deep Sea Coral Research and Technology Program for their support, of course the Schmidt Ocean Institute, Conservation International, our colleagues at the U.S. Geological Survey, and so many others who have helped to make this expedition happen. So we are so excited. We're going to see what we see. And welcome University of Guam.
Thanks for everyone who just joined. We're in the midst of setting down on bottom for this dive, and then we just initiated a rock collection for some of our colleagues at the U.S. Geological Survey, who are very much interested in the very rare rock samples that these make up. Not many scientific cruises get a chance to come out here, and any way we can collaborate and share the opportunity for data collection is something that excites us. It tells us a little bit of something about these seamounts, as well as about the substrate that the biology likes to live on. So as Randy mentioned, we're diving today on Titov Seamount, which is a guyot in the southern part, one of the southernmost guyots of, or the southernmost seamounts of the Pacific Remote Islands Marine National Monument, Helen and Baker Unit. A little bit of history about this site is, so we several of us on this expedition were on a cruise out here in 2017, which was the first time that this site had been mapped and dove on the NOAA ship Okeanos Explorer. And we were diving primarily on the far western ridge of this feature at much deeper depths. And then we did 2 dives on that cruise on the far western ridge. We are this time diving on the far eastern side of the seamount on a separate ridge that extends off to the east and south a bit. Our plan for the next couple days is to do a dive here and then do another short dive of similar length on the northern side of the seamount on a ridge that extends off to the north. And then we have some other plans for the next few days, but we'll get through these next few dives here and then let you all know. So the rocks we're collecting today, and we have been collecting all expedition long, there are a couple goals associated with them, namely that we're interested in the crust formations that occur and precipitate out of the water onto this rock. A lot of what you're seeing on the seafloor is not actually the rock itself, but rather a crusty composition called iron manganese crust, or polymetallic crusts that precipitate onto this rocky surface from the seawater. And this is a process that happens over millions and millions of years. And there is basalt. The basalt rock forms the underlying substrate here, and that's originally cooled lava, basically, that erupts from the seafloor, and then the crust kind of evens out all the angular bumps and grooves in the seamount. If we were to cut this rock open, for example, we might see that it does have a core of basalt or other types of rock, but for the most part, it's just the crust we're viewing on the outside. With many of these seamounts, what we're seeing, perfect, awesome. Can tag that one. 372 in the box. Good. Little fist bump. Teamwork.
Swing and a miss.
Yeah.
Deepest fist bump.
Oh, I forgot it.
Great job. So if you were joining us for our dives on Howland and Baker, you might have noticed that as we go shallower, there's less crust. And on a lot of these seamounts, well, many of the islands, almost all of the islands, but also many of these seamounts, sometimes there is a carbonate cap, which is rock that's typically produced, so the type of limestone typically produced by reefs, reef-dwelling creatures during periods of lower sea state and sea stand levels throughout geological history, dating back as far as the Cretaceous in some places. And over time, the seamount subsides due to the motion of the tectonic plates moving the seamount away from the original hotspot volcanism place, or just the increase in sea level, rise in sea level. This seamount tops out at about 1,100 meters, somewhere in that range. It's a very, very large feature, so it does warrant multiple dives because there are multiple different faces that life could live on.
Pretty much we hardly find anything like corals and other things in this area, just rocks.
But I mean, if you find a good place, it moves really good. I just don't think that's
You said rotated golf
That's a black coral. Yeah.
What do we have there? Do a quick zoom in. It's,
Mm-hmm. With a shrimp.
I think that was a small plexaurid, actually.
I thought it was
Okay.
Take a look at the next one.
Yep.
Yep. Can I just do a quick flyby zoom of this yellow stalk coming in? Go in. So we have a yellow plexaurid here with an ophiuroid or brittle star right next to a glass sponge. They're both really tiny, maybe less than 10 centimeters both. Good to note our first large corals. We saw a mushroom coral as well just a few minutes ago.
You got full zoom capability if you want.
All right, push in.
I'm not afraid of the numbers.
No, I got it here. Nice. Very nice. Okay. Good.
What color is that?
It's a plexaurid coral, so it should be one of the bubbles there.
This one.
Yeah, that one. All right, Ray. Thanks.
Oh, there's another one on there.
That's a mushroom coral.
Yeah.
Yep. Saw a couple of those when you were running around, saving us from certain doom, having to go back and fix our sample numbers.
Seems like mushroom corals are, not gonna say gold, but just not very popular. Sampling
Yeah.
things that-
I think they're underlooked and undersampled as well. They have they are quite diverse, but it seems like they have
Mushroom,
there's
Blixarid, coral.
very few species that we could visibly discern, and so it requires a collection in most cases. But that whole group was recently revised, and the name Anthomastus, which is what we've been calling them for years and years, has changed now. But it's not quite clear. It's not like something we can characterize without sampling it. So oftentimes it is undersampled, but they're definitely not overlooked. They're some of the most abundant corals on these seamounts. We did sample one in this depth range a while back, so we were just often drawing inferences. If we can look at it morphologically and genetically and draw some conclusions based on ones we've sampled at other sites. But from my approximation, there's about 2 different Anthomastus that occur in the bathyal depth range here between about 500 and 1500 meters. One is a longer stalked one, Anthomastus tahinotus, which we've seen extensively on the previous couple dive sites, especially shallower. But yeah, potentially a couple different species in this area.
Oh, there's a
Definitely
big hole.
well manicured cave. Careful, you might never leave that cave. There's a bunch of crinoids here as well. Stalked crinoids or sea lilies. So a lot of the times with the Anthomastus too, we know a little bit about their reproduction, which helps us understand why they're so clumpy and patchy. Is that most of the time they're brooders. So rather than broadcasting brooders, yeah. So they, instead of broadcasting their gametes into the water column, what they do is they'll fertilize their eggs and then hold on to them until the larvae are competent, and the larvae will then disperse. Usually not very far. They don't really need to go very far. In fact, it may not be evolutionarily beneficial for them to go very far because they never know if they're going to find a mate again.
Is that a sea cucumber right there? Mm-hmm.
I think it's the backside of an anemone.
Oh, yeah.
Just coming up the
Anemone.
other side of it. It does look like a sea cucumber from certain directions.
Strange.
I tilt up just a tick. Like that.
So we're making our way up.
Yeah, we're going up to somewhere in the 11 to 1200 meter range today. It actually, that's surprising because I don't think we, well, no, that's not true. Interesting story about our dive track today is we planned it starting out at a slightly steeper slope to try and capture what looked like it could be a rim of a volcanic crater that collapsed. And so what we're climbing up now is what we think is the outside of that crater, and it will drop down a little bit as we go through the center of the crater, and so we'll see what happens here. I don't know. There was some debate and some bets going on about what was actually down here.
Who won the bet?
I don't know. No geologists on board to break the tie, I guess. I was betting on volcano because that just sounds cooler. But as soon as it starts to flatten out, it's probably going to drop down a bit, and then we'll. It's not a huge drop down. It's maybe like 10, 20 meters or so.
That's heavy rock. No, I know it's up. I have to see what triple.
Those are stony corals.
That is right. Yeah, good find.
I know there's a current.
We'll take a zoom on them and see what we got here. Any one of these white coral colonies. Can we take a zoom?
Yeah.
Have a chance.
Which one would you like, that one or that one?
And
The one on the left, I like. It's a little bit clearer on our same plane, or any of them really. Yeah, that one looks nice. It's a little bit bigger, possibly older.
There's some Ecological up right there.
Oh yeah, yep, on the far left. Before we go there, I'm gonna zoom in over here on.
Cup coral.
Yeah. Yeah, no, I'll manual it. That's fine.
Oh, no,
Oh.
it's in auto right now. So
Okay.
I'm
Yeah,
not
that's
moving.
that's fine. Just leave it in auto for a
Okay.
second. Yeah. Nothing here has too big of a depth of field. So it looks like might be a cup coral maybe, or an anemone. And I'm gonna zoom over here. And what we're looking at here looks to be go for a tight zoom. Abandoned ship. Clearers are abandoned ship. So what we have here is a colony of Enelopsemia. Enelopsemia rostrata, most likely. I'm looking at some of the morphology of the polyps to make sure that I'm making the right call, since it does have some characters of another coral that's similar to this depth range called Madrepora oculata, but in this case I was able to find the characters I needed towards the bottom of the colony where some polyp loss is occurring that indicates it's an Enelopsemia. Alright, we can go up. I just wanted to image that one.
Good?
Yep.
No, I had a little bit of flush. I didn't have a lot.
Some Iridicordia colonies. So we're kind of like starting off in a depth range where we ended our dive yesterday. We ended at about 12, 20 something meters, and we're a bit deeper, but we're seeing similar fauna. We've done a lot of work across the Pacific, especially the equatorial Pacific, and it seems like 1,500 to 1,600 meters really is the depth range where you start seeing your stony corals and shallower. Of course, we have solitary cup corals all the way down to several thousand meters in some cases, a couple thousand meters or more, but they have very different energetic requirements and are able to live at deeper depths. Yeah, what's the current doing? Northeast to south, west?
Oh yeah, it's going. Yeah.
Northeast, southwest, yeah. mean, it look, you can see it.
Try to fix them up. Save away.
That's why I had to get a little bit further. I didn't think that would be.
Every 970, so that's.
Okay.
So it looks like this is kind of what we were calling the rim of this crater, and it does look really different. I don't have the, I was gonna ask him to put up the bathy when he gets back, so we can see where we are relative to this cratery thing. We'll get that up for everyone in a few seconds.
That's not really good. It's
Oh, is that a sea pan?
Exactly.
So
exactly.
beautiful.
No.
And a glass spongia right there with the
That is,
crinoid.
so we got a sea pen, possibly Halipterus in the sediment, right?
Yep.
Sea pens like the sediment, but this one right in front is a Chrysogorgia, a golden coral.
In front? Okay.
Yeah, right there. Yeah. It looks spongy, but when you look closer, all the polyps are very, very tiny. There's a sponge there in the distance, though.
So big. A massive one. Sponge.
So the coral we just passed, we were looking at some ophiuroid brittle stars as well. They were probably in the family Ophiocanthidae, known for this long spines on their arms. We're also looking at associates of corals, the relationships between a coral host and its associated organism. One of the samplings we are also doing, which is new for this Falkor cruise that we did not do in the past, was a new we're developing
Wait.
a new. Yep.
Is this
It's like a ctenophore. Ctenophores have this.
I thought it was like a
iridescent-
jellyfish.
It's a type. Yeah, it's a type of jelly. I mean, some people call them jellyfish, but
Comb jellies?
comb jellies is the
Oh,
common
comb.
name.
Comb jelly.
Comb. So huge blocks.
Some really cool lights at their little.
Yeah. You get that, and they're bioluminescent too. Well, some regularly bioluminescent.
Just have to slow down a little bit for the rest of the
Sure.
Do you think this is a stony coral that's dead?
It looks like a bamboo coral. You can see the dark bands. So
Okay.
There, but it's not live. It's assuredly dead. It's covered in hydroids. But it's amazing how much crust, like all the smoothness of the rock is all that crust precipitate,
Mm-hmm.
and it's pretty impressive. I mean,
Is there anything hydroids won't grow?
I don't think so. I mean, if they have a preference, I'm sure they'll settle on any structure rather than rock. But what I'm surprised is that when you have a rocky high, you don't end up with like carpets of hydroids either. I guess they like the biology. Yeah, I mean, another one of those groups, hydroids in the deep sea. We always think of them as like contamination, growing on my coral, but it's their biology as well. They have a diversity, and so whenever we get a sample, we try and sample hydroids as well, so we can confer with our colleagues ashore. just gonna do that.
There's a sea star right there, the very first one.
Oh yeah, can we look at the sea star?
You wanna look at it?
Yeah. Only because we were noting from our explorations on the Okeanos Explorer that no evidence of sea star on coral or sponge predation was observed at this site. So we just wanted to get an idea of what kinds of sea stars we have living here.
This is more kind of like yellow. Like we have seen an orange and a white. So yeah, cars. Or maybe it's orange, but by looking at it quite far, it looks like yellow.
A tiny little two polyps of a vector gorgia up there. See that?
Mm-hmm.
Maybe a newly settled. By that newly settled could mean like years.
Well, I thought that too.
Very nice. Very spiny sea star. Not all sea stars are coral predators, of course. Many of them are deposit feeders. They may be feeding on animals in the sediment or on the sediment itself, extracting the organic material. All right. Yeah, I think that's a good idea. So I think what's happening here, so you see where waypoint 1 is?
Yeah. I'm following it back around.
Yeah, but I mean, we don't have to stick on where the other. I was just curious, towards the center line there between 1 and 2. That's kind of where we think the center crater is. So we're like,
Go down into it?
yeah, a little bit,
Okay.
just to see
I thought
if it.
normally I always want to come up down. Okay.
Yeah, I just wanted to poke down there to see if it is sediment covered, and then we can come back up to the ridge. I think we're going to be on the ridge for most of the dive today.
Oh yeah, that waypoint is in Austin.
Which one?
The second one kind of, but it's just so you know, it's on the like, your little caldera.
Yeah.
It's like halfway down.
Yeah,
It wasn't at the top of the bump.
Is it on the north side of it, or?
Here, so I didn't move it because I thought you want to see the face of the wall, but after I did the higher resolution color,
Yeah.
it looks like this. See? So instead of being up on top of that,
Yeah.
it's right here.
Okay. That's
So
fine.
just so you know,
Yeah.
it's gonna be, it should be like that face along your.
When you boost up the exaggeration, it looks like it's
I know,
totally
it's like-
gone.
This
Yeah.
is only 2.
Really?
Yeah.
Yeah. Huh. All right.
I was
Coming
curious
out,
what the.
Come down a little bit more, and then if we don't see anything, we'll zag back up. It might actually be just part of. I mean, we're seeing less sediment here than. Okay. We're seeing less sediment here than in other places.
I was wondering what the geology looked like.
Yeah, me too. I was
Volcanic
curious.
versus
I mean,
real
it's
world.
definitely really rocky, not so much sediment.
Because there's so much current down here.
Yeah, yeah.
Sediment can't sense.
So these are the rock from the volcano, like
Yeah, at
Like
some
what you
point
were
in
stated before.
history, yeah, there was some sort of volcanism here that formed the underlying seamount. Yeah. It doesn't get soft down there, it's pretty solid. All right. Well, that's fine. Yeah, we can go kind of migrate back up towards the ridge.
Just want to go straight or like follow the kind of parallel to the
Yeah, parallel but upwards. Yeah. That seems to be where we're seeing more of the stuff was on the crest there. And so I'm fine as long as you guys are okay not following the waypoint track, just kind of going up to the crest of the ridge.
Okay, so
I think
like starting this way, we just climb uphill.
climb up. Yeah. So what happens is where the Falkor waypoint is, this ridge goes around and it kind of bows outwards.
So you're saying we're gonna be going up this way, like up to
Yeah.
No, he's
up
saying
to here. Is this uphill?
no, he's saying.
Wait, so
This
this
is
rock
called
is
the-
like,
So we're gonna come up to the hill and then we're gonna follow around the hill.
Yeah. I think that makes the most sense. Yeah, we're definitely seeing much more coral fauna up there, so I want to make sure we target.
okay, we can see some of the crinoid right there.
Just
Many crinoids and
Go back up to
sea
the-
star.
Yeah, shoot up there. That's fine.
But we hardly seen the corals.
Yeah, there's not much coral. I mean, there are some small ones, right? But the coral like to live up on top of the ridge where the flows are stronger. And you'll see, watch the marine snow here. It's pretty slow moving, but it is moving. But all the Enelopsemia and all the stony corals and all the big bamboo corals are up on top of the ridge. And as soon as we clear this ridge crest and we see blue water ahead of us, we should get stronger currents, which are driving that biology. Another farreid sponge. So Victor Gorgia over there, a purple coral.
Pisciculture.
Victor
okay.
Gorgia. Yeah, we'll peek in just because it's the first one. Specifically, trying to look at the sea star on it, the brittle star on it.
That's a spongia.
Yeah, that's
right next
a
to
nice farrea.
Mm.
Yeah. Kind of looks like a head of lettuce. I don't know if this one's going to give us the disc side, but that's fine.
You wanna go to the other side?
Yeah, let's do that. I guess that's easier for IT. Ready? All right, going in.
I move a little bit.
I think that's okay. Yeah. So we're looking at an octocoral here, Victor Gorgia, with an asteroschematid brittle star. You can tell there's current here because it's vibrating the mount on the camera, which is this, a little bumpiness you're seeing, a little turbulence.
It looks like a bamboo coral.
Bamboo corals typically don't have a lot of pigment. Sometimes they're brownish.
Just only one
yeah.
color.
Victor Gorgia So if it was a bamboo, you will see those clear nodes,
Yeah.
black and white bands in the coral. And then this one, Victor Gorgia, has very few color variations. They're usually pale pink or white. So that's one of the giveaways. And then I'm going to take a look at this sponge over here too. So this sponge is a glass sponge called Phorea. Hold that for a couple seconds. Sometimes these things have associates. For example, there's some squat lobsters in here, hidden. There's ophiurids as well. All right. Oh, there's chitin in the back too.
Randy was asking for some
Yeah, that's
sponges.
that's what we're looking at right now. These Phoreids, very hard to get species IDs from without collecting them, because a lot of the characters that we're interested in differentiating species for sponges are often in the shape and morphology of their spicules. So we need to basically grind them up a bit and then look at the siliceous spicules that make them up under a microscope. Okay, great, all set. But it does seem to be one of the two sponges that dominate this area that we've seen so far. The other one being a euplectellid that we saw just a few seconds ago. But definitely on this ridge crest seems to be where all the activity is happening.
Did they ever say what the new technology that was being implemented for the first time on this expedition?
Oh yeah. I started a thought there and I got sidetracked. Speaking of being sidetracked, Synephobranchid eel, cutthroat eel. Saw a couple of them. All right. One of the most common fish down here, along with the halosaurs and cusk eel. So that we are looking at a new technology. Yeah, wow, look at these Phorea.
There's a lot of sponges.
All over the place. Let me know, folks who are watching, if you want to sample this Phoreid. I don't know of many Phorea samples from this area, but we have some pretty good sized colonies here. So the sponge, the technology we're testing is called eDNA, or environmental DNA. We're collecting some data in support of some preliminary observations of deepwater coral and sponge communities using eDNA, and the mechanism by which that happens is we collect some water sample, and in the water sample at a certain depth you're supposed to get a representative idea of what kind of biology is around you, because the water is not just water, it's combinations of mucus and cells that are shed off.
This is a big sponge.
Yeah, some of the biggest we've seen. Yeah, this Phorea heads can get pretty good sized. Yeah, so and they shed off all these cells and mucus into the water column, and those cells and mucus have DNA in them, and so by taking a sample of the water, we should be able to filter that water, capture the DNA and cells on a filter paper, preserve it in a solution, and then take it back to the lab and sequence that material. Can we look at that coral head over there? There was a star or something on this side of it.
Crinoids.
Just wondering if it was a crinoid or a hydroid or a persingid. Or ophiuroid. Just gonna take a quick zoom in. We don't have to sit down, I'm just gonna take a stable.
It's a
there we go.
crinoids. Yeah?
We have the yellow one is a crinoid, but this one right here in the middle is a persingid sea star, which is a type of asteroid sea star. It's actually closely related to those, the ones that eat corals, but this one is largely planktivorous.
And it's pink, like light pink.
Pink, yeah, pinkish white.
That coral should be right now.
Yeah, I mean, the skeleton, yeah. It looks to be fresh, where the polyps are growing. There might be a slight tinge of pink because there's a thin layer of tissue.
Did you see the crab over there?
Yeah, I was gonna just shoot over there. So the left part of the colony should be white. The right part might not be because the tissue covers part of the colony on the outside, but in here it's dead, so it's just calcium.
White
carbonate.
balance on that, so it looks better.
Yeah, let's go over there first. Do a white balance, and then we'll do crab and.
Here, look at it. Let's go to the others.
Exactly true. Like it's kind of like different in color.
There might be a little bit of live tissue on this side, but it definitely doesn't look as nice, nicely pigmented as the other side.
This is a stony coral, yeah?
Right. Yeah, this is Analepsami. It's the same one we've kind of been seeing. A little bit better.
Alright, camera.
Great, thanks. Alright, so first this persingid sea star. We see these all the time on the rock surface, but they actually do occur on corals quite a bit. But these are not predators, to the best of our knowledge. It has very long spines that are actually quite sharp, and definitely not a party to handle them. And then I'm going to zoom here on this chirostylid squat lobster.
There's like a big ridge here. This over here.
With a crinoid as well. So these squat lobsters, you'll see that it kind of has these long arms, and it's using these long arms that have sensory structures to capture anything that it can fit in its claws in the water column. So basically like fishing with really long arms.
Can I ask you a favor? Do you have your sensor? Can I use it as a flashlight for a second?
All right, very nice. That one's a very small one. So we've seen much larger, but it's good to get good imagery of that as well. While we do a pilot change out, I'm going to do
There was a
poking around
shrimp
for
on that
this
side, yeah.
nematocarcinid
big.
shrimp.
Oh, there's a little coral on
Yep,
that side.
little baby Buxorid.
Where's the courage? And a shrimp swimming aside from the coral.
All right. So we're just kind of cruising. The plan is we're going to kind of follow this ridge crest here where the currents are nice and just kind of go off. Yeah, there you go. So kind of head, in a semicircle around, back to our track, meeting it somewhere between 2 and 3. Thanks.
I think I haven't introduced myself, yeah?
Hmm?
Oh, there's a sponge. Another sponge.
That one looks very similar to the ones we were seeing yesterday, strangely.
Is this an anemone or a coral?
Anemone right there,
Anemone,
yeah.
Okay. Coral, like cup corals, they're pretty small, yeah?
Yes, usually they're small. Sometimes they can get quite big, fist-sized. The ones that are from shallower water. But typically, the deeper you go, the smaller they are. sponge that was tall and columnar that we just passed looked like Regadrilla.
So there's another question from there. Did the disappearing sea pain from yesterday
Oh,
make it
yeah.
up in the push core?
Great question. I've yet to see him or remember to talk to him when I see him, but Russ expertly extracted a very problematic sea pen that loved to try and escape from the ROV a few times, and he did get some of it in the push core too. So we did get about half of the sea pen. So consider it a victory after all that. But even getting half, it was only about 4 to 5 centimeters of sea pen, which really is only about maybe 10 polyps or so. So very little material, but you know, it's valuable nonetheless. It's a pretty decent density of Analopsamia here. Small colonies, since we don't typically find the large colonies until we get much shallower. But the fact that we're having really steady current across this, colonies are all oriented in roughly the same direction, suggests that this is a fairly regular flow that these corals are living in, and that this is sufficient for them to grow, reproduce, calcify. If the current was variable, for example, we might see colonies facing in multiple different directions, which would indicate, you know, they don't really have a distinct physical cue that they're using to settle. Oh, there's a rat tail.
Oh, that's a fish.
Yep, rat tail fish on the right.
Cocktail
And then we've
fish.
got a Synaphobranchid eel, this cutthroat eel on the left. Really nice big blocks here, and I'm wondering if this will tell us something about the geology of the area. I'm not a geologist, but we partner with geologists who can help us make sense of what's going on here. Anthomastus mushroom corals. Take a look at this one right here.
Damascus.
Kind of look on it on a flat plane, if possible.
That's a black coral.
Oh, actually, no, changed my mind. Right here. This interaction here might be interesting. We have a sea spider next to a yellow coral that looks a little ragged. That might be a possible predation event. Free to zoom?
Oh, wait.
Alright. So I'm looking here at a pycnogonid sea this is great. A pycnogonid sea spider on this rocky surface. It's really low to the rocky surface. It's really trying to hang on here because the current is so fast that I'm sure if it was perched up high like other times we've seen this spider, that it would probably be blown away. But I'm really interested in what's going on here because we have a plexaurid octocoral that is it an auto or manual? Okay. Yeah. So we got a plexaurid octocoral here, one we've sampled so far. In fact, I can tell massive cenonchymal sclerites that are kind of left over after all the tissue's been scoured away or reduced in size. But what's interesting here is that the skeleton is showing because most of the polyps have been denuded, and the ophiuroids are largely on the live portion, meaning one of them is on the live portion. The cysteroschema is on the live portion of the polyps, whereas this ophiocanthid is clearly on the dead portion. And just the branch tips really have live polyps, but we really have no idea what could be causing this right now. It's suggestive that the pycnogonid maybe have done this, but it's not certain since it's not been observed, but it might be a scene of the crime.
Which is why I have a collection request
Okay.
for both the pycnogonid, sorry guys, and some of the tissue. And let's I'll ask Tim if he wants part of the oath, and or the oath, and I'll come in. Okay, so if you're just tuning in, we attempted to try for a leg of a pycnogonid. We thought we were trying to get it for stable isotopes to try to solve the mystery of whether or not they actually can eat that type of coral. They molt, so they can grow back a leg, but we didn't want to take the whole organism. But as you saw, turns out they've got very hard legs. So at his next molt, it will be fine. But we decided to have all his legs still on the seafloor. You know, it's always a gamble. We are really careful with our collections. We try not to push it, and we try to take what we need for science. We just needed this one for stable isotope and metabolite analyses, but we really didn't want to take the whole organism. So we tried, and we'll maybe try another one some other time. Is that going on in Anthomastus?
This is a bit of a coral
What's going on
smorgasbord
here?
You've got so the base of this is a bamboo coral. This right, and then you've got growing on one of the branches where it's died is a scleractinian and lepsemia, and then on the underside you've got attached to it an Anthomastus. So everything's focused on the bamboo coral base, which is really neat actually. You see there's quite a bit of tissue here covering the entirety of it, and they're all living in harmony. Maybe, we'll see.
It's very cool.
But yeah, I'm not sure. You know, this bamboo coral looks pretty tortured, so I can't even put it into clade because it's got some weird branching pattern. But yeah, neat. All right, we're gonna do a quick change here, and we'll be back in a minute. Caldera hypothesis is, I don't know, I'm not, I mean, it's certainly like really
keep
rocky
going.
and really current speeds here.
Yeah.
So it could be really promising guide for biology because it's really fast currents and really setting it free. So what we're doing, I forgot to mention to the watch lead, what we're doing is we're kind of off track at the moment since the waypoint 2 is kind of down the side of a slope. We're kind of going semicircle around on the ridge crest
Okay.
and rejoining it somewhere around waypoint 3.
Stay on the ridge. In other words, really.
Yeah.
Stay on the ridge. Aye, aye.
Get on that Sammy.
Let's take a look at this one.
Going straight to.
Cool. Steve, you want your sunglasses?
Yeah, I'm putting all my stuff everywhere.
Seriously. Slob.
Seriously.
Might go and catch the last couple hours of sunlight today.
Couple hours. Lucky you.
So she's been on top deck all day.
Hey, I was not on all day.
Sunrise, sunset, tan.
Wow.
Big hemicorallium. Lots of crinoids.
It's got worms inside. Yeah, probably. Yeah. It's always tough to tell. I mean, the older ones, right, are more porous. You know, like the one we collected the other day was pretty solid
Thank
still.
you.
And I wonder if there's like an age where they have a maximum amount of polychaetes growing. We don't really have a good polychaete sample from them, so I think it's kind of mostly the same kind of coralline movement seeing too. I was hoping to see some other species.
A lot of crinoids. Orphicanthus. Yeah.
Can I have control of the iris?
Sure can. Go ahead.
Thanks.
I'm gonna go pack some stuff into the lab.
All the polyps are closed around that brittle star.
Yeah.
Not all, but
I mean, polyp behavior is really just influenced by anything disturbing them. So if they're being touched by something, they're not necessarily damaged or upset about it. They just
yeah, but if the brittle star stays there long enough and there's not nutrient sharing between polyps, it can do damage to that section.
that's true.
Shrimp down there?
Shrimp.
It's Caribbean. It's a big colony.
Yeah, it's nice. All the crinoids are on the other side. It's like 10, 20, 30, 40, 50.
There's a lot of current, right?
Oh, yeah. Which way? It's going down straight towards us.
There you go.
Okay, we can keep going.
You going?
Yeah.
If you need anything, just slap.
Do you want us to?
Pretty good current.
There's a lot going on down here.
It's a lot more. It's a crinoid.
I'll be in the lab.
Is the current coming right at us?
Yeah.
Okay. So we just had a Science Watch change. My name is Alexis Weinig. I am a research scientist with the U.S. Geological Survey,
And I'm Brian Kennedy, a PhD candidate at Boston University.
and we are on a. Did we map this? No, we're at Titov
We're at
seamount.
Titov,
Titov seamount on the southern end of the Pacific Remote Island Marine National Monument,
Howland and Baker Unit.
Howland and Baker Unit. Ooh, let's look at the crabs in this Hortigorgia. It is really blowing. Do you like? Oh no, you don't like flying in crab? We've got a number of scallop lobsters in here. I'm just gonna. Are you gonna set down, or you want me just to zoom in a little bit?
Try to sit down.
Okay.
Just trying to find it.
Yeah.
up there, that's where you live. Real low. Oh, girl. I really plan to.
It's got 3 associates.
Yeah, 3 different squat lobsters.
Okay. Try that out.
Okay. And they are hanging on for dear life too. Lots. Looks like the Gastroptychus, Chirostylid. You can see on their legs they have little hooks, which they just kind of latch onto the coral branch, which is how they're able to stay attached even with this high current. And when we try and collect them, we also have to battle those little hooks, which are pretty well adapted for this environment. Europtychus from Tim Shank. Alright, that's good. We can keep going.
Keep moving.
Lots of Anelopsamia colonies here. Oh, there's a bubblegum coral. Can we take a look at that one? Sorry, that was abrupt request.
It's fine. Go forward.
Thanks.
It's very stable.
The coral?
Yeah.
Sebastian's.
As in,
Not moving, you mean?
not moving.
But I do, I
Strong
think,
current.
so
Oh, there it
see,
goes.
yeah,
Yep,
I was
nope,
like, I do
nope,
think
there
it's
it goes.
bubblegum. Lots of brittle stars. Wait. Oh yeah. This has all of its polyps out. Okay, I think that's good. Victor Gorgia, that purple coral. So yesterday we left seamount 3 inside the EEZ.
Sure.
That sounds right.
It was the third seamount we have visited.
Okay,
Outside the monument, inside.
there you go.
US waters.
Yes. And we have sailed south to Titov seamount. We crossed the equator yesterday, which was also our halfway point during the cruise, which was also summer solstice, so halfway through the year. So, and it was karaoke night, and it was pizza night. It was a big day on the ship.
It was all the things on the ship.
Cody and Jared had some really nice duets on the karaoke machine yesterday evening.
No way.
What kind of whip is that?
Bamboo. Take a quick zoom. I don't need to sit down for it. It's blown out. Yeah, we can keep going. It's a little.
Coming out of bamboo or?
It's a bamboo.
Okay.
It's just blown out.
Didn't have a ceremony yet.
Hmm?
So congrats to the new shellbacks.
Oh yeah, we haven't had our ceremony yet, so we'll be doing that on our crossing back up the equator just because of the amount of work that we have going on right now. Makes that a little bit difficult, but on our way out of the equatorial Pacific, we will have our ceremony for all of our new shellbacks.
It does technically mean that the logs are trespassing in Neptune's realm for the next few days,
Yeah,
though.
but Neptune has granted us access out of his good graces, so we'll take it. Can we take a look at these two corals together? We might collect one of them. Okie doke. Ooh, what's coming up? Oh, okay.
I hope it does.
So this is a weird anomia where half the polyps are facing one way and half the polyps are facing the other way. So normally all of the polyps on an Anomia, this white coral, face in the same direction. This one has a little of both. Don't see any associates on there. We have a Victor Gorgia with an Astroschema sea star So, I'd like to collect the white coral.
Okay.
We can take most of it. So from right about here and up,
Okay.
it is brittle, but that thick base is pretty thick.
Okay.
You know, so it might try and some of the smaller branches might break on you as you break off the rest of it, but we'll see what we can do.
Not there.
Yep, that's great.
Okay, cool thing.
Yeah, just you can probably grab it with the rubber or the red
Yeah.
bits now.
That secured on there.
Yeah, it just popped right
Yeah.
out. You'd think there's not too much that would be coming around doing that to them, so
True that. True that. But some of them are just like stuck on there, stuck on there.
yeah.
Like, oh, it's gonna come up easy. No. It's gonna be a good one 3D print.
Mm-hmm.
Really good.
And that can go in a bio box.
Yep, yep, yep. Alright,
Sample number?
373. Great job. That was awesome, Jared. Thank you.
All right, sample 373 in BioBox B.
Porcelain. You want to use that purple one too?
No,
No?
no.
Why way?
There's just to the right after he puts away the arm. I'll look at there's a sea star. There you go. I'll just take a quick look at that. Woo! Not quite sure, but it doesn't look like one that we've seen predating on corals before. That's good.
Yep.
Yep, thank you. We're on dive 11 for this cruise, so we have done, I think, 3 dives in international waters, and we have done about 7 dives in U.S. EEZ waters, both inside and outside of the protected area. We've been able to map quite a large amount of the area within the protected U.S. waters, on top of the data that already existed for Howland and Baker and Titov seamount, adding a lot of data both for mapping and as well as this video data to what is really inside these waters. Even though we're very far from the continental U.S., we are still on U.S. territory, and there'd only been maybe 6 dives prior to us. 7?
10, I believe.
Oh, 10.
Between your cruise and my cruise.
Between Nautilus and
Yeah.
EX. Yeah, so the EV Nautilus, run by the Ocean Exploration Trust, and the NOAA ship Okeanos Explorer have both been in this region before. So we're adding to data that was collected by both of those vessels. Cruises that I was on one of those and Brian was on the other, so we have resident scientists that have done work in the region before, and hopefully everything that we're gathering over our time here will add to a much bigger picture than what we had coming into the cruise.
Updated waypoint bridge.
And it looks like we now don't have
Thank you.
near as many Anelopsami colonies, if any. Or other corals. Looks like a couple plexarids. Can we take a look at one of these, Cody, the yellow corals, whichever one?
I'll go straight for one of them.
Cool. Cool, thank you. Yeah, I would say plexard. Is that what Steve was saying? Did Steve say anything about yellow corals before?
Roll them quickly.
Okay.
2 minutes before you get out.
Oh, gotcha.
I know that he was looking at one.
No
That one with
worries.
The, 6 spotter.
Oh, right. Yeah. We'll say plexard. We can keep going.
Keep it low.
Oh, that's an interesting little
Yeah.
cut out of that rock.
Probably a little collapsed lava tube or something.
Lava tube. dive plan today? Oh, here it is. Wow, so many waypoints. Oh, we're going up and down.
How long is this time?
It is estimated dive length of 12 hours plus up and down time, so 14 hours. We've already come up quite a lot. So nice over to Gorgia. We started at about 1,650 meters below surface, and we'll be working our way up to a peak of about 1,100 meters. But then it looks like it's kind of a bumpy ridge at the top, so it's a little bit of up and down. It's a very stark change. A lot less corals than we were seeing just right before.
It's a big sea star.
Yeah, Hymenaster, Slime Star.
Go look at that while it should move.
Okay. It has sediment like on it, which is kind of unusual. This is a type of sea star known as Hymenaster, but also commonly known as the slime star. If you collect these, which we will not be doing, and bring them to the surface, it will fill a whole bucket, 5-gallon bucket with slime. So they are tricky to try and preserve, but pretty ubiquitous. I've seen them in any ocean that I've ever studied. Sounds good.
Yeah.
There's some ripples in the sand. Camera has a hard time with that stuff in the water, huh?
Just trying to focus on it.
Yeah.
That sponge.
You want to look at that sponge?
Yep.
Right over here. Sorry.
We saw a big one of these a little earlier that we didn't stop and look at. assuming it's dead, but I just wanted to confirm that it was dead.
Yeah, I'm gonna get a look at the structure.
There you go.
Thank you.
That's more even degraded than I thought it was.
Yeah, it's really covered in sediment.
Yeah. Okay, that's good.
Okay.
Ready?
Yes.
Yeah, perfect was around. That's house. The rock house.
Yeah.
Patrick was
Patrick, yeah, I was gonna say.
Was there?
There's
Here's-
a little field of in El Absamio.
Yeah, looks like they're growing kind of on the
Oh, are they on
underside
an
of
overhang?
this. Yeah,
Oh, protection.
that one maybe fell. Oh
Take a
no,
look
it's
at
growing.
that stuff. Well, that'd be pretty cool.
It is pretty
We'll
cool.
take a look at that whole lot.
Yeah, we'll look around. This little couple. Oh, maybe it's an anemone down there. See any sponges? Lots of old where bases used to be.
Yeah.
So it looks like many things have fallen off over time. Many have tried, yeah. Not all have succeeded. There's a little chrysogourd.
Chryso in the corner
yeah.
too.
It is a big rock. Curious too what would have been what created those holdfasts because I don't see many dead skeletons around of anything, whether it's Nelopsamia or other types of octocorals. Is the current still as strong this way? Okay. Ooh, there's a big coral.
It's a big one. Look at all the squirrel lobsters on it. Or not squirrel lobsters.
Are they brittle stars?
Brittle stars.
It's a nice big colony.
Primnoid, I assume?
Yeah, yeah. I think it's Calyptrophora. Some crinoids. Lots of ophiocanthid brittle stars. Is this something we could try and take a collection of?
Sure.
Okay.
Where at?
Probably whichever branch is easier for you to reach. We just need a branch of it.
With the crown one?
A sea star or a ophoroid if possible, but if not, it's okay. I think the collection is more about the core. Yeah, like this one on the end. Can I come in?
Yeah.
Okay. Go look where the crinoid's hanging on has no polyps. Gives you a real good image of the current.
Yeah. You go full zoom. Is that max?
Not yet.
You're good to get a shot of the wounded edge.
Okay. That's maxing. Let's just do that real quick for 10 seconds.
Turn down the iris just a touch if you're gonna do that.
Huh?
Turn down the iris just a touch if you're gonna do
Okay.
that.
That's all I got.
Okay.
Come out a little bit. I don't think we have any spots we can get the ophiroid with the coral.
Doesn't look like it.
Yeah, okay. So we're putting this in a quiver, yeah?
Yes. Or wherever you want to. We'll go
Yeah, I would like to put it in a quiver. So is this too high to reach right here?
You got it.
Okay, cool. Yeah, first quiver. Quiver 16.
Quiver 16 is correct.
Thank you.
Correct. Very nice.
Can we take one more zoom before we take off?
One more zoom. All yours, Alexis.
Thank you.
You're welcome.
Whoop, okay.
You know, let's see the retracted polyps.
That's full zoom.
All right, we're good.
All right. We can carry on.
Paracliptiform.
Okay. Get it? I'm not. Oh, there's a rock pen down there. Anthanastis, more Nelopsamia, Victor Gorgia. Oh, that's a fish.
The fish. The Casquil.
Oh, there's. Can we look at this sponge?
Yes, ma'am.
Looks like it has corallivory on it.
And we will likely want to collect it.
Spongivory, yeah. Spongivory. Don't give me that. Don't roll your eyes at me. This will probably be a collection too. There's a couple of sea stars on there, I think. Let's see once we. It's like maybe partially dead, partially alive.
So it looks
or
like
it
to
just
me,
has
yeah.
like.
No, it looks like it's got dead and alive tissue still on it.
Like as soon as I pop over that rock. What do they make me? Oh, it's thread the needle there.
It is. It's right. You can't see the.
Yeah, it's,
You're good to go.
Okay.
Pictures of me real science cameras and thread.
Oh, okay.
You're in focus now?
Thank you.
yeah, it looks like there's 2 stars on the back side of it, which actually might be a pretty
I think
good
they're
idea.
on the inside.
Or on the inside. I'm seeing
There's 3 of them.
3?
Yeah.
Oh, yep, I got the 3rd now.
But the top bits of this,
Wait, where's the 3rd?
there's 1, 2, and there's 1 up here in there. See?
Oh yeah, now I do. I was mixed in with all the brittle stars.
Yeah.
See if we can actually see their stomachs.
I don't think we're in the right. But it's the same.
On that
white-
one, yeah. So that you're right. It's on the inside, eating out.
Eating, yeah. But it's on the. It looks like the last time we saw spongivory or something that looked like it was eating a sponge. It was a similar type of sea star. I remember it being white because
Yeah.
most of them that we see are not.
It was whiter than these, even.
Lots of little amphipods crawling around. Yeah, look at them.
Sea lights.
Doot, 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 doot. That is a thing. Google sea lights on your off time. Don't do it. It'll terrify you. I have a friend that does whale necropsy. So if a dead whale comes up on the seashore, they go and they take samples and study it. These look like little snails too.
They get sea lights.
And whales get sea lice. It's a type of isopod that like embeds itself into their skin. It's terrifying. And he always sends me pictures of them when he goes and does it because he knows I hate it.
Have you seen the hagfish now?
Yeah.
The hag?
Yeah. Your face.
I was on that Peter Gertis cruise.
Uh-huh.
And we did that whale pole.
Yes.
Rosebud. And we got a hagfish they wanted to sample. We brought it up and holding it. Slime everywhere on it. And it grabs me right here.
Really?
Yeah. I say, ow. I thought, oh, little teeth. And then the girls were like, oh, look at this. I said, oh, God. Terrified now.
All right. What's the plan for this?
I think we're actually gonna probably leave it.
Oh, okay. Do we get enough images?
I think we got enough images in the video, certainly.
Okay. Then we will carry on. It's definitely first time I've seen multiple sea stars feeding on. I've seen sea stars feeding on dead sponges, but demo sponges, not the glass
I mean,
sponges.
I have to admit, like when you look at these things in the lab, like I don't see any tissue on them.
I know,
Like what in the
yeah.
world are they eating?
I don't know. The one we saw before really had its stomach out. Those
Yeah.
I don't see that, so I'm not sure. Or maybe
I mean,
they use
there's
it.
no other reason there. I have a hard time believing there's another reason that the sea stars are up there.
Well,
other
there's a
than-
lot of like amphipods and stuff that live on
It's
there.
True. Don't get me wrong, but
Yep.
Yeah, a little cutout.
Fish bags. Aren't they?
so another flexor coming up.
Yep.
We didn't need to land. I think we probably got what we need from that
Oh
one.
yeah, we don't need to set down.
Spider next to it.
No spider next to it.
Black coral? No. Oh, it looks like a
A
decapitated
crinoid
crinoid.
Yeah. I always find myself wondering, like, what's just out of our light range.
That eats
Like,
crinoids.
I always think
I
about.
know, and especially that eats crinoids.
What?
I mean, we see a reasonable number of these decapitated stalked crinoids, and
Oh.
I'm really not sure what eats them.
Oh no, me neither. That's another thing where it doesn't seem like there's any tissue. Like when you bring a crinoid up to the
Yeah.
surface, it like.
You ever seen a baby recruit?
Can we take a look at this one? A baby crinoid?
Yeah, they're super cute under the microscope.
Oh, I don't know if I've seen one small enough to be under a
Or
microscope.
they're like, just like 2 mils.
Oh.
They're super cute. All these hollow pillow basalts.
Yeah.
We had lime in there.
Thank you. Here's sea lice.
Oh, wonderful. Thank you. It's like this. I'm gonna have mine.
I'll show you another one.
Oh, but thank you. So helpful of you. You're so kind.
I know. Just trying to share the world with you, Cody.
Thanks. Is that the bottom?
It looks like a little anemone, yeah, or
Oaf
That-
jumping?
jumping oafs.
Thrill seekers right there.
Bailing out?
They do often, certain types of them bail out when they feel the. We think it's because they can feel the vehicle approach and the pressure. Give a nice close up of polyps. Looks like this is an arella type of primnoid octocoral. Okie doke, we can keep going.
Well, I figured you were up to something else. Oh, stuff.
Oh, there's a
It's a
crab.
Hamalid.
A
It's like
Hamalid crab under there. Wow, look at that. It's all cut out.
I think cutout's the right word.
That's when we need our mini ROV to go spelunking.
It's
Can we
that,
take a
yeah,
look here?
that might be,
Yes, ma'am. Crab?
or that might just be a brittle star.
Oh, it might be a
crinoid.
crinoid. It's a crinoid.
that's
We're
probably
looking
right.
for a specific type of ophiroid,
We're looking for a deep purple, almost black ophiuroid.
but this is a deep purple,
Close-
almost
close
black
to the
crinoid.
That color, but a brittle star.
Do you wanna look at it?
I'll take a quick look, yeah,
Okay.
since
Go
we're
ahead.
already here.
Here, man, focus.
Thank you. like the polyps are not retracted around this. Look, they're like even being squished by the Siri.
It's pretty cool though.
It is a cool. It's an LSU crinoid.
Full text.
Oh, there's a little cup coral I'm gonna look at over here before we take off. Oh, looks like it might be dead. Oh no, it's alive. There we go. We can keep exploring.
Exploring. On the beach. There.
Do I have any starbursts? No, I'm out. Oh, I do.
What's on the other side of this big one?
Look, they're under overhangs again.
Yep.
I'm really beginning to be confused by this overhang thing.
Why?
Why are filter feeders like overhangs?
Because I think the way that the water moves around the overhang, that actually there's a lot more flow underneath it than above it.
Skeptical.
Okay. You can test it.
Do you have an ADCP with you?
I don't. That's why I said you can test it. Oh. It is a change in terrain. No more big boulders. Now we just have kind of a flat crust.
And that's me slowly coming back away from the creek.
Okay.
So we followed that ridge.
Yeah.
Go back.
Wow.
pavement is a super different terrain than what we've been seeing up on top of this little plateau now.
Yeah, it's tough when our bathy grids aren't that refined and you can't.
Good sized cusk eel.
Oh yeah,
Fish.
fish button.
Keep going. So this is a slow, steady slope.
Let's see, waypoint 3 says we should be at. Where's the circle? Oops. $14.99. We're a little above that.
Seem about right?
Yeah.
Okay. I'll just keep going in this direction.
Yep. We got a slime star. A swimming sea cucumber, or not? I mean,
Yep.
a benthic sea cucumber that's swimming. Yeah, we can take a look.
Current is you can
Just
swim in on
floating along.
it. Here, man, a lot of photos.
Okay, yeah.
You say that the current has slowed down up here?
Yes, it has. Maybe we'll hold a position there.
Oh wow. Okay.
That's a big slowdown.
He's just cruising.
Cruising, riding the wave.
What a life. Just floating around the ocean.
Eating mud.
Eating mud. Just cruising. Yeah. You see, I think I'll just take a nap here. Eat up, float around some more.
So some of these are land, take on sediment, and then swim while they digest it, and then land and take on some more. And some spend all of their time in the water column and just pluck out food from the water, and then others live almost exclusively on the bottom but can jump up and swim away when startled.
Or maybe when they want to.
Or maybe when they want to. That's true. We only really see them do it when we think we've startled them.
Yeah.
Little fish. Can't quite tell what it is. Probably a rat tail.
Coming down to it.
We don't need. We
You don't want to look at it?
nah. We can keep on moving.
Do a waypoint for you, Bridge. Yeah, Thank you. Now I'm holding back in this little bridge here.
Another pretty stark change in terrain as we've moved off the pavement back into the pillow basalts. Sea state's picking up a little bit. We're beginning to rock and roll a little bit more in the
Is that the?
in the
Didn't they
control
say
room.
that trade winds were supposed to kick
yeah,
up
we
today?
had a nice couple days of really easy weather, and the trade winds are coming back. Nothing we shouldn't be able to dive in, but unless we get a strong blow and strong currents at the same time. But the seas are definitely picking up a little bit more than where we've had the nice easy ride the last couple days.
It's a halosaur.
There was a halosaur.
Another halosaur. A couple anemones.
Is that a plex?
I think it's a
Yeah.
primnoid.
Primnoid. That's what I
Yeah.
meant. That's what I
yeah,
meant to say.
I knew that's what you meant.
Snap in.
Okay. Coming on in. And the fertile star down there. That's good.
Right, go.
Thanks, Cody.
No problem.
Bunch of little sponges. You
Yeah,
see?
just have that.
Maybe we could try and get a rock
Like
that
a
has one of
sponge,
those on
maybe.
it. Little plex. Just gonna snap in.
Okay.
Snap in. Oh, a tiny bracingid. It's so tiny.
Oh, that's cute.
That is definitely what that sea star was eating the other day.
Yeah. Yep.
That's amazing. It's like a little tiny display. That's good for us. Makes me wonder if there's got to be some chemical signal that the corals give off, because like that bracingid is right underneath there.
Yeah.
Back up. All right. Some things and stuffs. We got another primnoid octocoral. Yeah, we're moving up slope again. We're kind of on that pavement. Oh.
Nice spot.
Oh. We have a Narella, I believe. And then an Anelopsamia has a little hermit crab on it. It looks like an anemone right below the polyps too.
Take a look at one.
Yeah, we can take a look at this one from the other.
Rotate so we can
Sounds good.
straight onto it. The one to the right there?
Yeah, sure, yeah.
That one.
We can also take a look at some of those sponges when we.
There you go.
Thank you. That's what I was wondering back there. Let's look at these sponges right up here. looks like a crusting sponge. So it just grows in a thin layer over the rock. Been seeing a lot of these spread out on both the pavement and the boulders. Okay, you wanna go take a look what that thing in the distance is?
Let's do it.
You can't tell if it's a sea pen or like a tall whip.
That's a big sea pen.
Those are a couple big sea pens. We'll just do some video of them. I don't think we're gonna collect these because they're giant.
Won't fit in ROV.
Halopteris.
Alright, try that.
Okay. It's a nice little sediment meadow. Hey.
Beautiful, beautiful landscape. Can you click for a second?
Two other sea pens right over there.
There's 3 sea pens. 'Cause there's an Umbelula too.
That's what I meant.
Oh,
Yeah. Sorry.
2 others. Got
Two
it.
other kinds. It's a sea pen party. You don't normally get such high sea pen diversity in one
It's true.
sand patch.
This is where it's happening.
Oh, there's another
Yeah.
sponge.
We might, that might be Walteria. We probably want to go over there.
Okay. Oops, sorry, Cody. I started.
You're fine.
Okay. Oh, 3! We got
Oh
4
my
kinds of
goodness.
sea pens.
This is the
It's
most
crazy.
diversity of sea pens ever in the same sand patch.
Yeah, all the species of sea pens are represented here. I think.
Nothing
It's
makes Steve
juveniles.
come running like 4 sea pens.
I just wanted to point it out because I'm too lazy to type it.
You're fine. Do you want any of them?
No.
Okay. I think this is the one we collected with Russ the other day with
That's
the
the
push
sneaky
cord.
one. Yeah, that's the escape artist.
That's the sneaky one. Then we have this one. Which we did collect a couple dives ago. We collected this one as well. I love how the little mysid shrimp are always around the coral polyps. They must feed it. Can we hop over to look at this sponge a little bit closer?
We sure can. Hoppity hop up.
Hopping around. Okay. I think we're sliding. Is that okay? Looks like a sea pig too.
That one will probably take off here in a second.
And that's it.
Okay. I want to look at the sea cucumber first, in case he swims away.
Yeah, because you usually get a shot of its oral disc like that.
It's a very strange. It has a ring around its.
Well, those are. You see, those are its like it generally grabs sediment and puts it in its
Oh.
mouth with those things. So it's just looking. It's kind of reared up in a way
Yeah.
I'm not used to seeing.
Very translucent. Looks like it has a collar on behind it.
Yeah.
Fancy pig.
It's a fancy pig. Oh. Hanging on tight. And then what we really came for. So many brittle stars. And a shrimp living inside.
If we can cut the top off that,
Is
we
that
probably
the?
should.
Okay. Oh, there's another shrimp down at the bottom. Okay, so we're gonna sample this, but we just need to take a piece off the top.
Okay.
So not the whole thing.
I'll be right back in. That much?
A little bit more than that. That's probably good.
Yeah?
Yeah.
Yeah.
Oh, the sea cucumber hasn't left. I'm surprised.
It's really stuck on
I
there. I
would've
don't know if those are ones that will flap,
I usually think of the super translucents as ones that do.
but I think they don't normally have those little appendages. The
Maybe.
ones that swim.
375.
Yeah.
Quiver 15. The avalanche is starting.
Great. Cool. We're just gonna keep, carrying on. We kind of, our track was a little more back to the left when we came over to look at some stuff. So. Yeah, I wonder what'll happen to those shrimp, if they'll stay in there or if they'll.
I'm guessing they will, assuming the sponge stays healthy.
Ooh, a nice crinoid. Actually, can we take a look at that one? This is one, I think it's isocrinus, where they have the, like, the cirri are, like, attached to the rock, and they can actually, like, move themselves around, but they're on a stalk. I can snap zoom if that works.
So I guess it's official.
Snap zoom. I mean, you had your window to change it. Yeah, so behind it you can see the stalk Adam, can we fly to the side a little bit to see the stalk behind it?
Yeah.
Thanks. The stalk is much thicker. I think this is like they've been dated to a longer lineage than even just the stalked crinoids. These are like the oldest living crinoids. Yeah, here we go. Has quite a different morphology than the other kinds of crinoids we've been seeing. You can see how it's clinging onto the rock, so it can actually move itself around versus being stationed in one place like the stalked crinoids. Okay, that's good.
See that one on the back there?
Yeah, we can just keep going. We've seen that one. So we have a sea urchin. Something right here.
Yeah.
Very large sea star. Can we turn lasers on?
Hold on.
Yes. Up there. Yeah, it's like 20 centimeters. Doesn't seem to be doing much, but it's probably the biggest one we've seen.
Yeah, I think so.
That's good. There's a little stalked crinoid. Let me know if you see anywhere that you think would be good to take a rock. We took one at the
Yeah.
beginning, but
And we have come 400 meters, haven't we? What was bottom three?
no, 16 something, so we've
Oh,
come
okay,
like
200.
200. Big sea cucumber.
A lot more encrusting sponges today than we have seen
Yeah,
previously that I remember noticing.
those are really hard to collect though.
Yes, I'm not. Was
No,
not.
I'm not. I'm just saying, like, We could try sectioning one, but or getting a rock with one. I think those are our options if we
Getting a
want to
rock with one would be the.
have a representative.
We've sampled them before.
How'd you do it?
We tried to do it in the such sampler and we had to get stuck before.
Oh.
So the best way is just kind of gently scoop them into the box.
Oh.
You can probably put it in a quiver easily too.
Yeah.
How large it is.
Do we have, but we don't have a scoop, do we?
No, you just put it very gently with your arms. You don't
Oh,
crush it.
okay.
You just kind of pick it up and just like very gently with the coral quivers.
Yeah,
Coral quivers
yeah.
work well with it.
We'll take a look at this little sea star on the rock. I can just snap zoom.
You have to give it a better name also.
There we go. That's the same kind of ones we've been seeing on the rocks, but not on corals. Same with over there. Ooh, actually, can we take a look? There's an ophoroid on that coral that I want to look at.
I might flip around a bit.
Can I just snap on this side
Yeah.
then? Because the oral disc is... Oh no, it's just Ophiocanthid. It looks darker. Disappointment. Wah, wah. That's okay. We can keep going. I'll be right back.
Hi. The urchin there looks like on the side of that rock. So we're just passing through 1430 meters underwater as we come up this kind of the south ridge on Titov Seamount, which is the southernmost seamount in the Howland-Baker unit of the Pacific Rhode Islands Marine National Monument. This is dive 10 of the expedition, and we're about halfway through. These white corals you're seeing that we've been seeing a lot of here are in Elapsamia, which is one of the few still sclerotinian corals that you can find in the deep sea that actually make a calcareous skeleton. And I don't remember seeing them in this kind of abundance pretty much anywhere else.
Go, Brian?
Why don't you I'll keep you company until, did Alexis go to dinner or?
She did. Yeah.
How you doing?
Good.
Ah, it was you I was looking for.
Yeah.
Hey. I just wanted to know if you wanted me to stand with you tonight. Watch.
It's okay. Anyway, you want to do
I
it?
will.
You want to?
Part
I'm
of
sure
it.
what'll be
Part
happening.
of it at least. What?
I'm not sure what will be happening then. We'll be on top of this. Riding the plane.
It's all good.
I know it's all good. Love it.
I just wanted to check if that was okay with you.
Yeah, yeah.
That's all I'm planning for.
Of course.
Okay.
You guys looked at that already?
But yes, we think we've seen several of those.
That was it's like our 5th sea star like that.
Yep.
How much of that sponge did you get? Okay.
Top few inches.
Yeah. We definitely want more than that. But we'll try to get more. They're only the second one, so they couldn't do the whole thing.
It's a branched bamboo.
Branched or unbranched?
Unbranched.
Is it unbranched? Is there a whorl back there, or is it branch right
Oh, I
there?
don't know. I thought it was curling into
Maybe
the
it does.
page.
Get an angle here with Adam.
You could be right. It could be a branch.
Maybe you're right. No, I don't think it's unbranched.
I don't know.
He's riding right up the bridge. And
Oh, no.
Can you
Nope.
slide this way, Adam, a little bit, just to go around? No.
Unbranched.
Not bad. Not bad.
Should have taken wages before I sped around.
Get the whiteboard out.
I still have too many prizes that we aren't using. We gotta have more guessing contests.
3.5 meters. I don't know if you're writing down.
Huh. Yeah, 1425 meters. How's the current now?
It's a lot. As soon as we came out of, got him on top, the current dropped off.
Oh, good.
Yeah.
Yeah. See you later.
Okay. See you. The amount of Sammy also dropped off pretty quickly.
It did. We got out of that initial kind of dense patch, and then dropped off for a little bit, and it's picked back up to common, but not nearly as dense as it was.
Mhm.
And there's all these little encrusted sponges on like the side of pretty much every rock. It's not every, but
You got one.
pretty common.
There's a whole bunch of this one in the upper left there.
All right. You want to slide over that way? I see a crinoid.
Do you want me to look at this?
This is the yellow flexor you've been seeing, right?
We've seen a lot. Yeah, as far as I can tell,
Yeah,
those
with that,
ones we've seen a lot
with
of.
the astro schema of, so we're good on that one.
Yeah, one corner left and right there.
Do you want to go eat?
Yeah.
You
sure.
go eat, and then I'll go.
Sure.
Yep.
I thought you were gone.
Well, I can.
I raced to get here to make
You
sure
raced?
to make sure the seat was
Occupied.
Have you been seeing that one?
A little bit. You can zoom on it.
Well, yeah. Let's take a look at this one then, Adam, please. I've been watching, but I may have missed one. Is that? Yeah. Thanks. Good. It looks like it's one of those. It's a PRIMNM with
Yeah.
a master schema in there, but it's sort of a the central axis is pretty short to the first branch, isn't it?
And it's on there, it's just coming.
No?
No.
And then
You don't think so?
No.
All right, maybe that's just. I'm guessing that's astro schema intactum. We've seen this quite a bit. Anything else you want to see here? Anything? I don't
No.
see any damaged polyps. I don't see anything.
HC2 in there?
Yeah, I don't know about that axis. Okay.
One of you guys want to
I'll go. Or I'll go. Did
You're
you
up.
already eat?
No, you're up.
Okay.
Whatever it was before when you switch it on, it works. It's just negative is trying to, that's HD2.
Hello.
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 We got some little corals coming up, I think.
You know me. I love little corals.
Doesn't matter. We'll spot something.
I know we will.
Even if nothing's
Hello,
there,
Tim
we'll spot
Shank.
something. Hey, Randy Roach.
We are hanging out in the control room with Adam and Jared and Jimbo. You guys want to say hi?
Titov.
That's it.
Hello.
What?
Hello.
Hello? Say it louder.
Yellow.
How do they say
There you
hello in
go.
Ireland back home?
That's Adam.
How you doing?
What's happening?
That's how it
What's
is. All
happening?
right. All
There
right.
you go.
Excellent.
Alright, moving on.
Moving on.
Moving on.
Pretty great. I've been wanting to see Titov for a long time.
Really?
Yeah.
Oh,
it's
I'm
really
excited.
great. In 2017.
Yeah, we didn't make it this far
Yeah.
north in 2017. That's what we're doing. We're on the
No.
we're mirroring our
Yeah.
2017 expedition here in 2021. We're on Titov seamount
Yeah.
in the U.S. Pacific remote.
Titov.
Yeah, Titov.
You know, it was named after a Russian cosmonaut.
I was wondering if you knew
It
the
was
story.
discovered.
Yes, tell
It
me.
was discovered by the ship Vitiaz, which was a famous exploration research ship the Russians had in 1961. They found the
Wow.
seamount, and they named it after a cosmonaut. His name was, I think, G.
Titov?
Titov.
So wait, why did they name it after him though? Like, why? What was the rationale for this particular
I think-
seamount? Do you know?
I don't know.
I don't
I
know.
don't know. And it, I can't remember the spaceship he flew in, but it wasn't like it was a well-known one. But it was a large anemone over there. Yeah, it's a Titov,
Wow,
Russian,
amazing.
named after a person. A lot of seamounts are named after people, especially in the Atlantic.
I know, but I always wonder, sort of like, I mean, I guess there's a lot of paperwork filed. I could go look this up. I just haven't had a moment to do so.
Yeah.
But
A lot of the classic taxonomists that describe some of these species we're seeing back in the 1800s have their name as seamounts now, like Farrell. Good, a lot of them in the Atlantic, North Atlantic. A lot of the great coral taxonomists got seamounts named after them.
there you go.
There you go.
Well, I wonder when Auschwitz seamount will be named. He's destined to be a great coral taxonomist.
Absolutely.
Don't you think?
Yeah.
I don't know. Some
I
kind of
did-
plexaurid?
Yeah, I think he already is. It's
Yeah.
that yellow plexaurid we've been seeing quite a bit of.
Yeah.
It's really weird. It was down. We have lots of sort of zones of things. We have the Inalopsamia, Scleractinian hard coral zone.
Yeah.
We have this crinoids.
I'm surprised that the
Critic origins.
Inalopsamia fell off, actually. Right? Where'd they go? Is it still their
I don't
place?
know. You know,
Why
we've
not?
also seen a current change during the
sure.
during the dive. There's very little now, so maybe that says something. Not seeing the sponges anymore. All right, Randy, here we go. We're gonna get something here.
I know. I'm looking at the chat, and I have to admit, I had this panicked moment because I'm a professor and I teach 200 students, but I'm not teaching a class right now. And Chad, I don't know who you are, but I do not teach Bio 902. But you made my heart drop because I was like, did I forget to grade something last spring? I just was like in this panic moment. But
What in the world?
I have no idea. But
Yeah.
Um,
Okay.
that's funny. But no, not teaching classes
Yeah.
right now.
So we
Oof,
gotta-
teaching in COVID, that was a thing. But Chad, I wish you luck.
No.
Ooh, nice.
What do we got coming up? Let's angle up here a little bit.
There you go.
How about that? There's something. There's something.
That is something.
That's something. That's a nice fish with a parasite on it. See that
I do see it.
white
Oop,
thing?
and here he
Probably a
goes.
copepod
Woo!
right there. Yep. A little bit of a relief change here for dinner.
For sure. Yep.
And
You know,
that's
but in all seriousness, just going back to the classes thing for a moment, I
What?
actually do use footage like this in my classes all the time.
Oh yeah.
Absolutely. We do this taxonomy slam in my marine bio class, right? Where we just spend a couple of weeks going through all the basic characters of invertebrate biology, right? And sort of like figuring out all the characteristics. And then I show them a deep sea dive. And I'm like, before this taxonomic slam, you couldn't have identified anything. And now you can look through and you know what you're talking about.
Right.
It's pretty, you know,
Yeah.
it's good.
Well, that's
It's
wonderful.
really great.
That's wonderful.
So, that is definitely what I should do is show it to them beforehand, make them feel totally
And see,
like,
yeah,
oh my
that-
God, I
that's
need this taxonomic slam. That would probably be better.
I often show them things that we don't know, right? Have no idea of what it is. But it's a great learning tool, telepresence even.
For
us,
sure.
Us, broadcasting this out right now to people around the world is so educational for people. And you learn right along with us.
Absolutely. I've had my classes call in before and get to like say hi and ask to whoever's talking.
Yeah.
It's
Oh,
fun.
yeah.
Yeah. But you're right. It is
Great.
such a great thing to be able to have telepresence and speak to everyone.
Yep.
And I love those moments, right, where you get to replay them and you're like, this was unknown, right? Like the first time we found the monoplacophorans,
Yeah.
which was only a couple years ago.
So this is the seamount where in 2017 they a heterocarpus shrimp, a large red shrimp.
Oh, this is
actually
the one.
attacked-
Yes, I watched that. It was
attacked
amazing.
I think it was a dragonfish that was bigger than the shrimp, and took the fish down to the rock.
Oh, yeah.
And picked out the stomach. Did you hear
Fish
that,
versus
Sarah?
shrimp. Shrimp won.
So
It
this-
was outrageous.
So the shrimp tackled this fish, and brought it down to the seafloor and started ripping out right at the stomach, and kept ripping and ripping and ripping.
Shredding it. We'll find the
pulled
footage for
out,
you.
Out of the stomach, got into it, pulled out a little fish, and ate it.
I'm
Never
pretty
seen
sure.
before since, I don't think. And
No.
it's unbelievable, right?
What kind of shrimp was it?
Heterocarpus. We'll
Yeah,
see
one of
it.
these big red shrimp.
Maybe we'll see one today.
And it just, I
Point it
think
out.
the fish may have, I'm not really sure what had happened, but anyway, while the whole thing happened while whoever was narrating the dive, I think it was
Amanda DeMopoulos
Amanda and Steve.
and Steve, I think.
And they were watching a coral, and you could see the fish and you could see the shrimp, and they were separate from each other. And while they're looking at some coral, the whole thing happens in the background, and all of a sudden they realize what's happening, and they're like, "Oh my god!"
Freaking out.
It was amazing.
And we just let the battle play out. Watched it.
And yes, it's just these behaviors, right? We don't ever get to see behavior like that. It was amazing.
So
All right.
We're in a gorge here with no one living there. We saw one earlier today.
Wait.
You see crabs?
Wait. I'm gonna do a snap zoom.
Oh, Randy.
Who's this?
Oh, yeah. That's
Yes,
one.
of
That's
course
a
I see
shrimp,
something.
Is it? That's a shrimp, I think.
Timmy Shank.
So
It's my
either-
job to find
it's like
things.
It's either a shrimp or it's 2 or 3.
It's a shrimp.
Yep. It's probably a Bathypalmonella,
I
the
want-
large claw.
I want to know where my corallivorous jelly is. I look at every single one of these and I got nothing.
You know, you're right.
I know. Ay. We gotta find it. Oh, here comes the J-Rod beauty shot.
I'm just looking for your little guy.
No, he's not there. When
There's
he's there,
something
it,
right there. What's that to the left?
Where?
where?
On the end there? That's a mysid shrimp over there. Here.
Yep.
Let's point to that, maybe?
Yep, exactly. When's there, it's really obvious.
So your cosmonaut flew in Vostok 2, apparently.
Yes.
There we go. So I just found
That's
the
it.
naming stuff here.
That's
There
right.
you go.
That's right. Do you remember the Vostok too? Oh,
Technically,
no.
I might be. No. Actually,
You've
probably
heard of
not.
it, though.
No, yeah, I have heard of it. Yeah.
Not
Right.
quite that.
So there's a
This is
question in the
our-
chat that I feel
yeah.
really compelled to answer, which is it really that bad to throw my plastic straws into the ocean? And I don't know
Oh, gosh.
if this is just a question to get us started, but yes, it's really that bad to throw any plastic in the ocean because it doesn't degrade quickly. It breaks down into microplastics, which then are ingested by plankton. And even while it's macroplastic, other things will try to eat it. It just, you're trashing these beautiful ancient habitats, and there's a better place for that straw, which is the trash, where it will get dealt with at least differently than being put into the ocean. So we've done some work on microplastics in the oceans, and we found them on seagrasses. We have found them that corals actually eat microplastics, and there's all and microplastics can vector diseases into corals and all kinds of other things. And we've also done some work on macroplastic debris. Tim and I published a paper on that, actually from the Phoenix Archipelago and throughout the rest of the Pacific.
Oh,
Remember,
yeah,
that
right.
was just
That's
this
the
last
same with
year. Yeah, me too. And we found big pieces of debris that had been down for however long. And there's a paper led by Diva Amen, and it doesn't break down. So for sure, please handle your rubbish responsibly.
So on this cruise so far, I think we've seen one bag and one. What else we've seen?
We've seen a
can,
bag, we've seen a
Fishing
can,
line,
some
somewhere,
fishing
yeah.
line.
That's it.
So there's definitely anthropogenic impacts, even down here in the middle of nowhere, in the equatorial Pacific,
Sure.
thousands of meters down.
Yeah.
You know, there's a joke, there's a cartoon where it's like a person at the grocery store, and they're trying to buy a fish, and they're asking the checkout person for a plastic bag, and the checkout person responds, "It's in the fish, sir."
Oh God.
So just think about that.
Yeah. Well.
Second person to orbit the Earth.
Oh really?
Yeah.
So Titov was just informed by Jimbo, our lead technician extraordinaire, said that it was the second person to orbit the Earth.
Wow.
Titov.
They're the second person to orbit the Earth.
There you go.
There he is.
Who was the first?
I don't know.
But it was
Jimbo's on it.
Gagarin.
Gagarin. That's right.
Wow.
That's right. So there's another trying on the other story I tell, which is this recently published data that even in our deepest trenches, 7 miles down, which is 11,000 meters, we actually have microplastics in the guts of animals that live in the deepest parts of our ocean already.
I'm sure they're here too. And you know, one of the things that's terrible about it being in the guts of these organisms is they don't really leave. And so they're taking up volume that could, I mean, food is so sparse down here anyway. If you're lucky enough to be able to get something, if you can't digest everything because your gut is full of microplastics, that's just, I mean, you're basically starving to death with a full stomach because your stomach's full of plastic. It's horrible. And the impact that we have, eventually they'll die, right? And then the plastic will be re-released into the ecosystem for somebody else to take in. It's when that happens and it's big stuff, it's called the ghost fishing, where you have fishing gear, like derelict fishing gear that will catch fish, and then they'll get all clogged, and so for a while it won't be able to catch any more fish until the fish die and decompose, and it'll be able to resuspend and catch more fish. Ghost plastics is the same kind of idea. They can just be re-released and re-released and re-released to the system. So yes, please,
Wow.
please.
How much plastic do we have inside us?
That is
We
an
do.
excellent question.
certainly
We definitely do.
No, that there was a study that they looked at 4 people and 3 of the 4 had microplastics in their guts. We just zoomed in on that one, so we've seen that one. But
I think that's that Norella that we've been chatting about.
Yeah.
So pass that one up.
Yeah.
Yes, please.
Thank you.
The other thing is, as you know, that even in toothpaste, have you ever seen this done before? They take all the toothpaste out, but they sieve it, and
The what?
you'll get a vial of beads in your toothpaste.
Yeah, well, not
That's
anymore.
part-
Now it's been banned.
They got rid of it?
Yeah, it was banned a couple
Yeah.
years ago. They were used as like a dental abrasion.
Yeah.
Right, but now they use things like apricot pits and things like that instead,
Oh,
which
really?
which is much better.
Oh, yeah.
Yeah,
It's a good idea.
it is a good idea.
Yeah. Should do more of that kind of stuff.
It's a lot of apricots. They're delicious.
That's a lot of apricots.
Tim
Think about that
shake.
next time I brush my teeth. I've been using less toothpaste in case there's beads in it.
You know what this is?
All right, here we go.
I can't.
We've
I
seen
can't
it.
traverse the seafloor
That's
without laughing
Paracalyptrophora.
hysterically
You
with
saw
you.
that one,
Yeah,
too.
that is the Paracalyptrophora. Same thing, or what we think is Paracalyptrophora.
Yeah.
All right, can we do a very quick?
What do you want to do?
Okay, there's a
Looking for your-
plexurid at the base, and then I'm going to look for the jelly really fast.
We're going for a look.
And I will. You got a shrimp. Just one. And some barnacles on the dead section of coral. All right, great. That was really helpful. Thank you. Then I'm going to take a quick peek over here.
Look for your jelly.
Yes, I'm just going to do a fast scan. If it's there, it's a little red dot, and it's very obvious. Reddish, pinkish, orangish spot.
Kind of blows in the wind, right?
Nothing. Yep.
Yeah. I don't see any associates on there.
No.
Mm-hmm.
Oh, what's going on here?
Where?
It looks like an
Some
opus.
corallivory?
I don't know. It's kind of an odd structure from afar.
It's like an astro screen that one up. Paracalyptrophora. But we'll see. Or that may be a chronoid too.
Yep. Thank you. I'm having trouble
Yeah, we're talking about
zooming.
So we're at 1392 meters. Where does that mean we are in the process here?
It means that we're about halfway there.
We're past waypoint 4.
Halfway home.
We've still got
Except,
some uphill to go.
remember the top of this dive, right? We're just going to drop down into that rim. And if
Yeah.
it's interesting, we're going to stay there for a while.
Yeah.
So we don't have a waypoint for it, but...
Roger that.
You
but
want to look at this one?
no. Now I know what's going on. All those crinoids and that ophir all over it. Thank you. Okay. Maybe
So I
yawning
think,
again.
I think we dropped right into it today too. We got on the bottom.
We did. We were right there.
And,
So we are diving a side of Titov seamount that's never been seen before. So actually, you know what's so appropriate?
What?
He was the second cosmonaut or second person. There we go. What? Can we take a look at this,
It's
please?
a rock.
It's.
That thing there?
Yeah.
It's a rock pen.
I know, but it's on.
I know. It's great.
It's great. We
Question
saw
mark.
one in the sediment earlier.
I know. We've seen all the groups of rock pen. I mean, sea pens.
Yep. Okay, so then great. Beautiful. Thank you. So if Titov was the second person to go around
Yeah.
the Earth,
Yeah. Orbit the Earth.
orbit the Earth,
Yeah.
this might be the first seamount where actually by the time the EX tracks plus our tracks, we'll have gone
Have
all the
orbited
way
it.
around. It's true.
Yeah.
I mean, not
Not
that way,
orbiting
but
it,
like
but
I've seen all four.
yeah,
Yeah.
we've gone
So
around it.
it
Yeah.
like seems fitting.
We could.
We could.
We could just map the circle around
No.
it. Just-
We have too much diving to do.
I know.
We got diving to do,
Got
Tim.
an anemone coming up and stalk crinoid. There's a little more sediment here. Mm-hmm. Another yellow plexarid with a orpheroid living in it. Looks like those ophiuroids, the ones I just saw there, it's the same species throughout these yellow, living in these yellow colonies. Yeah, I think they also live on other corals as well, but they are certainly consistently on these other colonies.
Yeah.
And when you see those, you're not seeing any kind of the crabs that we see on other corals. I haven't seen any other worms or things like that. It's just that one brittle star species.
Yeah. And to answer the question in the chat, they're asking what jelly we're looking for. It's probably related to Agena, but I think it's a new species. It's undescribed. We're working on describing it, so we're looking for more examples of it so we can do a better job of describing it.
Yeah. So the narcomedusa, yeah, is what
Exactly.
we're looking for.
And we've seen it sort of directly. It's got this little internal structure that's sort of perfectly shaped for a chrysogorgid polyp.
What do you got?
Anemone.
Yeah.
And a shrimp that's not eating a fish this time.
That's not the same shrimp.
You sure?
That's a little nomadic or sided shrimp.
Oh, you're
Yeah,
right. You're right. You're right.
It's, genus is globally distributed in the deep sea, so
Yep.
when you see heterocarpus, you know it. It looks more,
Yeah, it's pretty.
getting
It's
more the size
beefy.
of a lobster.
Exactly.
It's a brute.
All right.
But it often lives on primnoid corals. You might see it on this dive.
Maybe.
What kind of coral?
Chrysogorgids, so including Iridogorgia and things like that.
Yeah.
Hello.
Mm-hmm.
Mm-hmm.
You want to go here?
No, you
wanna
go.
eat? Have you eaten?
You go. No, but you go.
Get outta here.
No, you.
He
What?
came back first. Let's just see.
Okay. I've done a
See you
terrible
later.
job of vlogging. I haven't learned a thing.
There's Victor Gorgia
All
with
that.
an Elder scuba.
I don't know what we were doing. We were being Tim and Randy.
I was writing them down.
Okay.
Totally old school. All right. So we had 3 more colonies in the now. Samia came up. Quite a few Iridogorgia came up. 1 with a shrimp. All without crabs. Still seeing this yellow plexarid. Primnoid or 2. Stalk crinoid. You've seen that guy, right? That's the. You did a rock sample already?
We got a rock sample with them. Steve grabbed one when they first touched down.
That's right.
So we'll probably be due for one 100 meters vertical or something.
We're looking to do a Niskin, and we found some diversity. Still?
Yeah, so
Okay.
Steve's, we've gotten a lot of the eDNA stuff, so I think the instructions we passed down from Steve was to really just target like super high density
Yeah.
communities now.
Okay.
And we've got a good range of things, so really just be more judicious about only firing bottles in areas of super high density.
Yeah. It's a serious manganese coating on these rocks covering the basalt.
Yeah.
You can just. So I'm assuming the seamount's 80 to 100
That's
20 million years.
that's the best guess for this part of the Tokelau Ridge. Yeah.
Yeah.
So the geologic time scale always kind of makes my head hurt when you think about how thick this crust is, even at the rate of like whatever it was, 1 millimeter per million years. Still turns into a lot of crust after 80 to 100 million years of time.
Another Iridogorgia down there.
But these same
Yeah.
rocks have been, on the surface the entire time, or exposed to the seawater. There's a nice little grouping of.
Is that a rat tail? Or is that a chimerid?
Rat tail.
It's a rat tail. Anthomastus. Do we want a rock or not? Even just loose?
Yeah, I mean, if those are, that looks like they should be in pretty good. Yeah, let's grab a rock.
We'll get a rock. We'll get a rock. In the Alexandria field. This is not. There's quite a few up here.
Yeah,
Came on
it
top
looks
of
like
this.
it continues on up out of sight too.
Might not be Niskin worthy though.
No, I don't think so.
So we're setting down now to see if we can take a rock here that's coated with this iron manganese crust for colleagues at USGS to look at the chemical composition of the rock, look at the age of the rock, try to provide some information on the formation of these seamounts we've been visiting. Okay.
Maybe. Well, you gonna sit here with this leg like this?
Yeah, it's a little annoying.
Do you want this rock right here with the coral on it?
No, not with the coral in it. No, we'll leave that one.
Oh, that's a nice rock with coral on it, though. Okay, but we gotta get good imagery, right? Of whatever rock we take.
Yeah, but we'll let them find the rock that's loose for.
Is that big enough?
Yep. All right. You want to leave it there for a second? Let me and kind of drive the camera.
Yep.
All right. I remember it. All right. Can you rotate the wrist? All right, perfect. That should be good. All right, I'm full wide.
Okay, what number are we on?
That was 376. Rock sample.
What do I say about that rock?
Trapezoidal, botryoidal crust on one side.
Oh, I do not know how to spell trapezoidal.
I'm going to zoom in on, I think it's a crinoid, but I just want to make sure before we lift off. Whoops, wrong button. Oh yeah, crinoid on a stalk. Okay.
Yep.
Anything you want to see here in the hard corals?
No, I don't think so.
All right.
They're all a little broken
Let's
up.
keep
huh?
on moving.
They're all very. Look like somebody came around with a
Yeah.
baseball bat.
Well, they are up here in this little rock field, so we don't know.
Oh, maybe they rolled around.
It's possible. But as soon as we kind of hit that crest, we're picking up a decent number of additional corals here in the current. When you guys came over with that rise, did the current change appreciably on you?
In the last time that I looked to now, yes.
Yeah.
But same direction, it seems like. It just calmed down.
Calm down a little. We've got a little forest of. I really can't stand saying the name of this coral.
Analopsamia.
Yeah, I mess that up every time I try
Do
and
it.
say it out loud. Analopsamia?
Samia.
Samia. Analopsamia. Okay. I was trying to put an extra syllable in there. I've seen a lot of crinoids on a lot of the corals today. And I feel like we're seeing more of these kind of larger, like close to Venus flytrap style anemones today than we have the rest of the dives so far.
Plexarid.
Yep. Continuing to see these little small patches of encrusting sponges, but they're pretty ubiquitous on the dive today. What is
What is
that?
that? Yeah.
Can we stop and take a look at the
Plexarid.
no, the orange bit between the plexaurid and the sea star.
Anemone.
Oh, yep, just the side of an anemone. All right, I'm full wide. Let's keep going.
So there's a question about what is marine snow when we say marine snow is just what's falling through the water column. It's just normally dead small plankton or some sediment that can get caught up, and it's just what is up in the, like as we're flying you can see the specks. That's all marine snow, where it brings nutrients down to deeper waters from the surface. And also sediment, so we can see there's kind of like a slight blanket of the white sediment over these rocks, and we see some places where there's much more sedimentation and other places where there's virtually no sedimentation. It just depends on the rate of the current and how much sediment is in the water to begin with in that region.
And it's totally a catch-all term for pretty
Yes.
much anything that rains down. It can be fecal matter, it can be old mucus nets, it can be whole bodies of plankton. In surface primary productivity, and you think about transport of fixed carbon from the surface to the seafloor, it can get really complicated. There's things, the microbial loop, the viral shunt, all these things that the biological oceanographers have kind of figured out about net export of carbon from the shallows into the deep. That all affect basically the quality of the marine snow that these deep sea benthic communities are reliant on for food. Because with the exception of some chemical processes in mud and hydrothermal vents, those being chemosynthetic communities, and some whale falls. Yeah, I knew somebody was going to catch me on that one. Basically all of the food that gets down to this depth comes from the sun still, just like pretty much all food on the terrestrial environments. Have we seen that one before?
Yeah,
We
yeah.
have? Okay, that's just the Norella?
Yeah.
Okay. Little brittle stars on the side coming up on fish with potentially with some I'm trying to see if it had a parasite on its other side, but it doesn't really matter. This looks like an probably a type of cuskeel or ophidiid.
Little caves.
Yeah, all these little hollow pillow basalts. These form where you've got flowing mafic lava that hardens on the outside, and it creates basically a small lava tube, and leaves the hard rock and on the outside, but then the lava can move on, and so a lot of times they are hollow. A couple more primnoids and another plexaurid here. Matulate crinoid here on the left, a couple of them actually.
I guess our rock didn't have any sponge on it, did it?
It did not. And this appears to be a rat tail. The main thing I use to tell the difference between a rat tail and a cusk eel is that predominant dorsal fin, first dorsal fin that sticks straight up and is detached from kind of the elongated caudal fin that runs along its back is kind of the quickest way to tell the two families apart. Another anemone, another crinoid here in the background. Long arm shrimp. Just coming up, approaching 30 meters. On the little kind of not so hot looking plexaurid here with another one. So this is a Titov seamount, which is a geode, which means it's flat-topped. And so we're running up one of these rift arms up the side ridges, and we'll get up to the top, and it should be a flat, very flat, kind of like a mesa would type feature. And when we were here in 2017, the flanks down here where we are right now were pretty much like this. We were more on the north and the west side of the feature in those dives than where we are now. But once we got up to the kind of edge of the flat top is where we really saw a pretty spectacular coral community. Yeah. Let me see another hole in the pillow basalts. These are old pillow basalts. I mean, we don't believe this feature has been active in 80 to 100 million years. But I did some work in 2016 in and around the Marianas, and we were seeing pillow basalts that were 1 and 2 years old, and they have this really beautiful glassy sheen to them sometimes, which is quite amazing. Looks like we've got a bamboo stalk here on the right. Corallivory. Can we take a look at that, please, sir? The bamboo whip. Yeah, the bamboo whip. The bottom of the bamboo whip had a sea star.
Okay, so not that one up to the right.
Nope.
Actual bamboo.
Yep, the actual bamboo. So one of the primary focuses of this cruise is looking at coral predation and corallivory. The primary kind of coral predator in the deep sea appears to be these sea stars. There are other sea spiders, iplecopherans, a few others that appear to eat corals, but this goniastrid sea star is a pretty voracious coral eater. So we're trying to get a better sense of the rates, what are all the predators, and exactly kind of the feeding habit. So like this looks kind of unusual. This is surprising to me a little bit to see the live tissue up top and see the sea star down here on the bottom. with the assumption that it would have already eaten its way up, but has returned bound to the base. And so what I'm curious to see here is if we can see if there's any tissue below the sea star, which
You may come around.
does that look like tissue to Alexis, or is that sheath on the proteination
No,
scale?
that's just the skeleton.
Okay.
I can easily come around.
Yeah, if you can, why don't you see if you can spin around and give us a look on its ventral side. So sea stars eat by actually extruding their stomach outside their body cavity and then wrapping around whatever they're trying to digest, and then externally digesting it and then sucking up the nutrients off of its prey outside of its body cavity. So what we're do here is once J-Rod gets set up, we'll take a really tight look at the right where the mouth should be to see if its stomach is extroverted or not onto the skeleton, and see if there's any live tissue, which I'm just not seeing anything here. So I'm guessing we won't actually see its stomach at all here. It's just holding on.
What's that yellow bit?
And I have no idea what that yellow bit is. You stable?
Mm-hmm.
All right. And those two green lasers you see will give you a sense of scale. So they're 10 centimeters apart. So the little sea star friend here is just about 4 inches across. And I
Maybe a
have
parasite?
no idea what that green thing is.
Might be a parasite of some kind.
Yeah. Alright, that's all the zoom I got.
Well, he's just hanging out.
Yep, I don't see any evidence of active eating or anything.
I know Randy wants to look at the coral that's behind
Yep.
this too.
Oh, really? Okay.
Yeah. But I mean, we should look up
Yep,
at the.
absolutely.
Oh yeah, there is a sheath of tissue.
So you think that?
See
Yeah,
the
I mean, I see
difference?
the interface, but I wasn't sure if that was. I thought you were saying the other day that there's some kind of secondary sheath beneath the actual tissue between that and the skeleton.
No.
Okay.
There was one time where we saw one where it was like just a very overcalcified part of the skeleton.
Okay. All right. Well, let's go look at the interface with the damage and the bare skeleton, and then we'll move on.
So many sclerites in that coral. I cannot imagine eating that. All the little rigidness you see are calcareous sclerites, so little bits of calcium carbonate that are embedded within the tissue. So it's just always amazing to me to think that the sea star would be able to process those.
That's a pretty clean looking edge. I don't see any patchiness or whatever. It just is eaten up to that point. All right, which coral want to go?
It's on it's facing, if we face the other way, it's behind
the
this
way?
coral.
Okay. So we need to pick up and
Yeah, pick
run
up
around.
and turn around.
All right, I'm full wide. Can we go back to the other side of the rock we started on?
Yes, you may.
Thank you.
Yeah, like there's that one that's got a lot of dead she wants to look at.
Oh, so it is that one.
Yeah.
Oh, sorry. I thought you said it was on the other side of the rock.
Oh no, it's this one.
Okay.
I heard that too.
Shush. Both of you.
There wasn't this one?
Yeah, this one.
Okay.
Or is it going on the other side of the rock?
What do we have? Look at the crab here, though.
Yeah, we will look at the crab.
Going up. Look It's cold. Stuff it there.
Might be stabilized.
Okay. Alright, we'll take a look at the tissue interface here first, and then we'll check out that crab. You want to get Tim's opinion on, see if Tim's paying attention?
Is he sprinting
That is
down
a
the hallway?
very
That's a weird
spiny.
crab.
That is very weird crab.
Can I either have iris control or... Okay.
Try it up in the shutter speed.
What would you get there? Try it now. Okay, hold that. Grab the one out of further in real quick. Try that.
Am I all the way down? I don't feel like I'm getting any there we go.
I don't think that knob's working.
Yeah, that
Okay.
knob's not working. It's only doing one increment.
Okay, well then,
Just tell one to stop.
we'll do. That's actually back one stop. It's brighter.
1, 2.
Steve
How's
said,
We'll figure that out too. Alright, so this doesn't look like nearly as clean
said-
of an interface between the...
You're in autofocus right now.
No, I'm in auto... No. Okay.
There you go. You're manual right now.
Okay.
You own an island.
Very small bit of it.
Geological palace.
Okay. So this is a much more ragged
yeah, let's look at those black
interface.
dots.
Yeah.
Number 1 sun is going to look right
But down here it looks pretty healthy compared to the rest of it. So we're particularly interested because we're doing a lot of coral immunity and corallivory work on these interfaces where you have live tissue and dead skeleton nearby, near each other. Alright, let's see what these, if we can make any heads or tails of these black spots. Nope, too far. That's all the zoom I got.
Can we focus this one?
Yep. Focus the closer one.
Don't know. I've never. Trying to see if it's like. Are they like on top of the skeleton, or are they in the skeleton? Ooh, look at that guy. Are they on all the? Are they on everything that's bare?
But while we're here, let's get a good look at the crab, and then we'll go hunting for the limits of the black stuff.
I don't know what this crab is.
Porcupine crab.
Yeah.
Named it.
Oh, I guess we.
species.
Wow, I love it though. It's so cool.
Well done, you two. Well done.
With the Ophiacanthidae brittle star here right next to it. Alright, let's see see how extensive the black dots are. So the black dots don't appear to be everywhere.
They are all over that.
Yeah, but I don't see them further out distally all the way out.
Well, let's zoom in.
Yep. So you got hydroids over here, still got black dots.
And like a discolored skeleton. It's not just
Yeah,
black dots.
that's true. What are these little bright things over
Those
here
are
and here?
snails, I
Those are
think.
snails? Yeah,
Yeah.
that was my kind of initial reaction as well. So yep, there are dots out here this far. Does Steve want a section of dead skeleton for spot ID?
I think we should take a sample of it. There's a lot of it, so.
The one on the bottom right has like a lot of dots.
Yeah, it doesn't have to be a big
Yeah.
sample, just something.
Yep.
One branch.
And it's weird to see the crab not, like in an area where there's not polyps. You don't see that very often.
Just taking a break.
Yeah.
Oh, what's that white?
It's like a shell.
Looks like it's got
Oh,
lots
it's
of
like a little
different
snail over there.
lots of different snails on
Looks
it.
like a cicada.
Is that a? I don't know what that little wormy thing is.
That's a really small brittle star, huh?
That's. I mean, that's what I'm kind of wondering if that's a. But I don't know.
There's barnacles, there's.
Different
This
types.
actually look like little zoanthids.
Yeah, those look like zoanthids. I agree. Oops, too far.
And all that other stuff's hydroids, little things sticking out.
This looks like there's hydras and zoanthids on here.
So hydroids don't have the like polyp, the tentacles on the
okay.
polyps.
So that would be,
So that would be, I would say, a zoanthid.
okay.
Yep,
Little snail.
Little snail.
There's another one of those.
And yeah, there's another whatever those are.
Oh yeah, they are like weird little wormy things.
Yeah.
Okay, let's get the weird wormy things.
Yep. So you want to call it
You want that?
just
yeah.
right there?
Yeah.
Yeah. You can just grab that. Dash in a quiver.
Okay.
Dash it.
We gotta put her over. I'm not sure. They look like that.
I think we saw
Steve said the crabs they saw, that genus in the Caribbean, and it turned out it was Neolithodes. Which I knew is what you were thinking, Brian.
Brightness, or you give them what you got?
I think I wouldn't have gotten Neolithodes, but I was thinking something Lithodes.
Lithodes?
Lithodes or something
That's
like
what
that.
Tim said was Lithodes.
So we're just gonna take a snip here of this dead skeleton so we can get a better sense of what these black dots are on it, and then there was a couple. Yeah.
Right there.
Okay.
What's
It's right there. The probably a little snail, but
super brittle.
And that fell right where, the same thing as that cutting.
Super brittle.
oh, that was a hermit crab.
Oh, okay. We don't need to.
That one pole stopped. Going down the road. It's hollow, hold up. Is that the hollow? gets it in a little bit closer?
I think as perilous as that it's in there, let's just put it
Yeah.
away. I think we've got enough imagery of it in situ.
I'll bring the pump down a little bit.
Okay, this is FK 21 dash.
377?
Do you want anything else from here?
I don't think so.
No,
You know,
we
it's
can't collect crap. What quiver? 14.
14.
Alright, good to go.
Yep, I think we're good here. Got a new handle on the slurp?
Yeah.
Nice.
A little bit of a modification.
Little bit of crates.
Couple primnoids back here.
And some plexaurids.
And some plexaurids.
They look a little degraded, so it might be worth looking if there's evidence of tissue damage. There's definitely hydroids.
Yeah,
But you don't really get hydroids unless there's bare skeletons.
right.
You want to look at it?
Yeah, you're good?
I think so.
Okay.
Let me get the camera up there.
So we're just going to take a look at the damaged areas on these two corals and see if we see any signs of what's caused the damage or
Or if we
the
see any
extent
more
of the
black
damage.
spots.
Or we see black spots, which I do see black spots.
Oh yeah, me too.
Let's go.
That's like it's a little crazy.
Can you give me manual focus, please?
All yours?
Thank you.
No, I didn't look at that.
Nice little brittle star
Mhm.
and barnacle here as well. Lots of hydroids here, or actually zoanthids probably.
Cool shot.
It's funny, I've been amazed by all the primnoid skeletons we've hit today and how golden they've been, considering that they're not gold corals.
Yeah, it is actually surprising. I feel like they don't look like that normally on the surface.
Lots of different things living here. Whatever the black spots are, you got amphipods in there, you've got hydroids and
Zoom on
zoanthids.
the black spots that are on
Oh
the
yeah,
hydroid.
look at that.
Are those little white spots that hit the post that you're saying?
No, that's all I got.
Or full zoom.
Yep, I'm full.
Okay. J-Rod, the little guy with the red face
Yeah,
is an
he's an
amphipod.
amphipod. Alright, while we're here, let's look at this plexaurid back here.
Deep sea gumbo right there.
Try to do one at
Yeah.
a time. If you do both focus and zoom at the same time.
That's
you
what
get
this
to a
is,
different-
stutter? Okay.
Yeah. Somebody was supposed to pass that information on.
Don't give me that look. This is impromptu Brian taking over the camera today. I didn't give him a lesson. I'm sorry.
So it looks like the tissue break here from dead to having still tissue is like an inch up that middle branch.
No black
Yeah.
dots.
Right there.
Yeah.
There's some black spots up there as well. All right. Anything else with these two we're gonna look at, Alexis?
No.
All right. Full wide. All
She
yours.
said collect black stuff
Wait.
on that species, please.
Okay, never mind. Still.
I would just collect that coral.
The flexorid?
Yeah.
So we want to take that whole guy.
That whole thing?
Yep.
Or
Yeah,
like
I
the
got
part that's
At least, yeah, if
The
not, if
branches
either
that
the
are
whole thing
dead.
or the two branches on that side,
Yeah.
either one is fine.
Okay.
I'm pretty sure you can.
Good. Go for grabs? Did you get some good pictures or you want to try again?
I think we got all the imagery we need.
I'm just looking for my water bottle.
Alright, that's gonna have to go in a bio box.
yeah.
Which bio box would you like?
I don't think we care.
Well, there's
Your
something
full
in-
left and full right are all. The center one's got something.
There you go.
A and B are fair game.
So good for A, right?
Yep, you're good for A.
$370.
378.
Bio box A.
Yep.
Good takeoff or you want one more right here?
I think that's good.
Slacks, anything else? Yep, we're good.
That's nice.
Yeah.
Should be working.
Oh, let's take a look at this that has the multicolor
Oh,
on
yeah.
it.
So, I'm gonna look at this top one. Oh, a nice big basket star on top of
Yeah.
it as well.
And something else that's not all
Yeah, I can't.
Lexarid.
There's a whole lot of things going on there. That might be Kulamana,
Oh,
that section.
Just leave the dark one from the right back in. Depth sensor on my own.
Probably a barrel.
Before that.
Oh, so I didn't know you had loaded that one.
Yeah, I was just trying to get a stable with the pair was all over.
Your line, that's not good, right? To everybody, I didn't know you had loaded that one.
I'll go with that.
You can do it now.
Wow, there's a lot happening.
Yeah, it's a lot of happening on that one coral.
So many ophoroids.
I don't even want to speculate what all is going on that one yet.
Are you gonna wanna like sit for a while?
Yeah, probably.
I mean,
I can't get a handle on what's on the left until we look closer at that. This definitely seems to be a popular spot, though. This whole rock has corals all around it. And you see that brittle star with the arms missing?
Yeah. That brittle star narrowly escaped something. Cost it most of its arms. There's so many colors here.
Yeah.
This is really
so I
different.
think this down below is Acanthogorgia, based on how
Yeah.
bushy it is.
So we're really lucky that this ROV has these extendable skids that you can push out and then get a toe hold on the rock to balance on. It's a really, really nice addition to an ROV. It allows us to be able to kind of hover-ish on these ledges and vertical walls and get a much stabler view than trying to free fly it. So J-Rod, our expert pilot here, is just trying to get a toe hold so we can balance here while we get some really detailed imagery of all the things going on here on this coral.
that is some slippery rock. Have to get up on that piece, I think.
Probably catching some turbulence from around the rock too.
Yeah.
Comes my left, it dips in a lot. I was trying to just put it
Yeah,
right
it's definitely
on it.
zoanthids on the left. I just don't know what kind they are. I mean, I never know what kind they are, but now that's what I think that is.
Yeah.
Trying to get some zoom right there.
All right, coming in.
Reposition.
All right, what do you want to look at first? Too many options. So many options.
Let's start with the
Purple.
yeah, purple and yellow.
Purple first eye. Can I have manual focus, please?
You're in manual.
Okay.
I can pop it auto real quick.
That's fine, I got it.
We'll just turn the knob forever. It's okay. Yeah, it looks like some type of plexaurid.
Multicolored
That
party
has-
party
yeah, it has
plexaurid.
blue and yellow with lots of ophoroids, and then growing up it is some type of zoanthid, I think.
That's really different. So you think that's all one coral?
Yeah, it is. So sometimes they have two colors. Like some they can be multicolored like that. Swiftia is actually too. It just
It's true, but it's
looks like
similar
one.
colors as opposed to
Yeah.
very contrasting colors.
Right.
It's 3 oaths on this section. And we've got a pretty zoanthid, hydroid covered dead section here with a hermit crab.
What's interesting is I don't even know if it's actually dead because the
Or-
skeleton.
Yeah, you're right.
You know.
Still, the coloration continues on.
So I don't know if it is actually growing over the tissue.
And that looks like live tissue.
I know I've seen this before, and now I can't think about what the actual, like this type of overgrowth. I can't pinpoint what it is. My brain is
And
not.
we've got, here's all the zoanthids on this
Yeah.
other side that are overgrown and has killed the tissue.
Yeah. It's like a disco coral.
Yeah.
Or a rave coral, I guess.
We got this big, beautiful basket star all unfurled here, right on the kind of the line between. It looks like it's got some arms around the
It's nice and slow.
the plexaurid and some right around the zoanthid.
Yeah.
Oh, let's look at the interface between over here. So that looks like that is dead.
Look to the
Okay.
left again. a little
Left.
bit.
Okay.
I'm just gonna show you a little bit better image.
There we go. And the light. Oh, look at that chirostyl or that.
Oh yeah,
Spot lobster.
right in the middle
Tucked
of
in the middle.
Is that better?
Yeah, thanks. Let's go just get an ID on him.
Some shrimp. I'm not locking because I'm too engrossed.
He's got three little spines coming off at the rostrum.
Yeah. It's tucked back in there pretty
That's,
safely.
That's one of the good way to identify these is the shape of their rostrum right between their eyes. So it's something we always try and get a good view of.
Did not know that.
Looks like an immunida of some kind.
Basket stars are just cans.
They are so cool. They are potentially one of my favorite deep sea organisms.
Things of nightmares. Just imagine falling into a basket star and just swallowing the whole giant basket star.
Or a basket of basket stars.
Basket of baskets.
Oh wow.
With sea lice.
Well, once you throw the sea lice in there, it does get pretty terrible. I think one of my favorite things I've ever seen in the deep sea was a whole field of basket stars,
Oh,
hundreds and
yeah.
hundreds and hundreds of them, lined up on the side of this seamount.
I think we are going to want to sample the blue and yellow coral.
I want to get a shot of this interface between the
The top one or the right one?
zoanthids.
Wow, so many over.
That's not me. Coming forward.
Sample it?
Yeah.
Good. Mess it up different.
Well, yeah, I do want to look at one of those
Yeah.
too because they look very different too.
Yearly sampling, I'll just kick down a lot more.
I like this one. This on top. Johnson?
I hear you. If I rest that thing on top of my front porch, it's gonna be hard. I think.
This might be a good, if we can, like even sitting lower and looking up at it and trying to do a beauty shot of the whole thing.
You wanna get a whole shot of it before we start stepping it?
No, if you're set up here, I think we're
Yeah,
okay.
if you're set, then we'll sample first.
I have one more thing I want to image before
ahead.
you go for the sample.
Tim would like a zoom on the percentage at the base of the plexarid.
So I just wanted to get a shot of where the tissue stops and the zoanthids start. Okay, nice shot of the central disk of these brittle stars as well. Alright, so Tim wanted a percentage at the base.
Do you want it blacked out?
Yeah, sure, if you wanna do that.
I'm gonna turn it more like the uppers, like that, turn it on.
Yeah.
Wow, that's crazy.
That looks cool.
It does look cool.
That's the shot. I was,
Can we take lasers off?
yep. So demanding.
Please?
That's a cool shot there.
Done in that.
Sports Center, highlight reel.
Stop messing with the focus, Brian.
Sounds like good for right here. People having PTSD at work.
There's a comment about why you might hear Slack coming through the audio. We use Slack on board the ship to communicate with each other. So sorry
No, I
if
just
that
got
sends
away
you
from
back
it.
to work vibes, but we use it for work out here too. So we can have people sending requests or IDs from
Sure.
different parts
You
of
wanna
the
pop it
ship
in, pop
without
it
all
in.
having to be in the control room.
Alright, that's it, right there, closest. Back in.
Yep. Alright, so Tim wanted,
There's a percentage,
Percentage
or
at the
I
bottom?
think it's actually an override, but he said percentage down at the bottom of the plexarid. I don't know if we can see it anymore.
nope. We just
Oh, it's
lost
right
it.
there. It's right there. On the, just to the left of the acanthogorgia.
Oh, yeah, the one that's missing all the arms?
Yeah. Oh, I think that is a percentage. Maybe it's right. Yeah. So it has 2 long arms. Many short, squatty arms.
Bad autofocus. I want far field.
It never focuses on what you want.
No.
It's regrowing 3 arms.
Route 3.
I'm sorry, 6. I can count.
There's a little, oh, look at the nice little polychaete on one of the fully grown arms still.
Let's just stay here. You're not allowed to go back to.
No
I'm
breaks
not
for
allowed
you.
to sleep.
No breaks for you.
No
You get to go
sleep.
right there.
Alright. Anything else
You have to stay
imaging?
on the ship.
Anything else from the imaging from
No,
the peanut gallery?
sir.
Get your own room here.
Alright guys, can we take a section of the blue gold with an oath?
With an Opie.
With an oath. Pilot's choice. Where you think that's the easiest to do?
Yeah. Just take one oath.
Yeah.
How's this right here?
Yep.
Perfectly
Perfect.
fine.
Alright, slow.
Thank you.
So slow. I think I won't. I think I might go.
Oh, that's what we do.
go for it. I'm not sure we got the whole thing.
Can we either turn the left light off or dim it? Is that possible?
Yeah, we'll try that. Let's turn. Is that one on? And this one on?
Oh, it's pretty dark. Never mind.
We have to do this one.
It's got its own cool effect to
It
it.
does. It looks really. Ooh,
That's too bright.
that's too. Yeah.
That's way more better.
Yeah, that's better.
That's cool.
Go give it here too. What is that? Fish.
Oh, a whalefish.
What's a whalefish?
Velvet whalefish.
In the rear camera?
Yeah, it's in one of the rear cams. It's a kind of has a body form similar to a cusk eel and has a, but generally oriented vertical, and it's bright red. Sorry for those of you at home who can't see it.
It's on the camera. That's on the back of the vehicle.
Yeah.
This looks just fine. Drops too far.
This is a dramatic shot.
It is.
I'm not gonna lie. I do not feel like we're doing this right now. Freaking out. There.
Is that enough of it?
Yep.
Enough? So flexible. This is a very,
Very flexible.
Like a blue bubblegum pop?
Nice.
Got it.
Still got the oak.
oh. That looks like a rock. Very rubbery.
Yeah.
Oh, we didn't switch the numbering on this yet.
Oh. Okay, 21-379.
Yeah.
What quiver is that?
That is quiver number 13.
Town.
Nice, Cody. So all the samples that we bring to the surface, both corals and the animals that are associated, like the brittle stars, all get preserved for identification back on shore. So everything gets used in multiple ways for different types of science, but they all do end up in tubes.
All right. Any requests for anything else before we move off?
There's another coral to the left. I don't think we can see it from here, but it's a very yellow, bright yellow.
Oh, that's right. Just don't want to look at that one.
Yeah.
The one right at where my foot is?
Is it right where you put it? Not that
No,
one. No,
it's
it's a
around
bigger
that. It's
colony.
around the left side of the rock from our perspective.
This is pretty though. I don't really want to leave. Goodbye.
I don't want to go to sleep. Does that stuff smell so good?
Smells so good.
What smells good?
James's tea. Some kind of
Oh, I know. It smells. I sat down the other day and I was like, this smells amazing.
vanilla something.
Smells like Christmas.
All right.
There's that super
Yes.
golden one right there. Lower one or upper one is fine.
Yeah.
Sea cucumber's gonna go hungry hanging out on the rock.
And it's right where I wanted to live.
You think?
This looks like a Canthogorgia, but we'll take a close look at the polyps. Or the little baby Bursingid.
Oh no, little baby, yeah.
That one we saw earlier
Hiding.
is the cutest Bursingid I've ever seen.
Yeah.
The babyest Bursingid.
I find it interesting that they're hiding. Seem to be
Yeah, they
definitely
just seem to be
hiding.
using the coral as protection.
You're gonna want to sample this or just look?
I think we're just gonna look. Yeah, it's definitely a Canthogorgia. Which I don't know if we've been seeing a lot of today. It's mostly been the plexarids, which are like what's behind it. Oh, there's some aplock. Lots of aplocks.
Lots.
Look to the left too.
Oh.
There's one, there's one, there's one.
Yeah.
There's one,
Yep.
there's one.
Right. That's a lot. But no like obvious damage here.
No, you're right.
This is one of those creatures
But
that we wonder whether how
they
much of an
actually-
impact of corallivory it is. They're definitely species that we know are corallivorous, and we see them here, and like here, there's no damage on this coral around where the worms are hanging out.
We call them worms. They're actually
Yeah, they're
mollusks,
mollusks.
but that's
But
easier.
they have a worm-like body plan.
Yes.
Some hydroids with amphipods here on the edges.
Lots of little brittle stars all
And
over too.
Bazillions of brittle stars.
Bazillions.
Bazillions.
It's a quantitative amount, if you were wondering. Little amphipods.
Anything else here?
There was something at the base.
There's
but
the
not
percingid,
the percingid,
not the
but there
percingid.
was that.
Looks like a little baby sea cucumber or a nudie.
I think it's a nudie.
Nudibranch, another type of mollusk.
Are we max soon?
We are all the way in.
That is definitely a nudibranch.
Yep. Fairly common in shallow reefs. We do see them in the deep sea as well, but not as commonly. Right? Anything else? Lots of. Look at all the.
I mean, I would say we're supposed to sample for Mayplac offering.
I don't
No?
see any damage.
Oh, okay. What's that up in there on the left?
Look at the little baby crinoid too. I'm going. Potentially a benthic ctenophore?
Oh, maybe.
Very potentially though.
I don't know. It's hard to gelatinous something.
Yeah. Look at all the little. This is like the nursery coral. I feel like with all the little baby oaths, the little baby Bersingid.
Okay, we can keep going if you don't want to sample for the.
No,
What was that?
I don't know.
So you get that one?
It was small though. All right, I think we're good here. You get a good sense of the direction of the current here as you
Yeah.
watch the marine snow get moved from left to right across the screen.
Autobots? Roll out.
It's a nice rock.
I got
Yeah,
the-
get
I got
a
the
nice,
little turnouts.
Good rock.
good rock.
Jungle lady, yeah.
Morning.
We still have not found that little corallivorous jelly yet, though.
Mm-mm. I mean, but I think we only saw like 2 instances of it in 2017.
Yeah.
It's not very
I think it was more than
prolific.
2, but
Fantastic.
Do you know what that coral down below is?
no. If you have to ask me what a coral is, you know we're in trouble.
What are we at? 1313. What waypoint are we close to?
We're still in between waypoint 3 and 4.
Okay. That was a fun rock.
Yeah, good rock.
Yeah, you see what I'm. We're looking at the same thing.
I think that was primary. So we got an urchin, looks like a rock pen.
Yeah.
More of these plexaurids as we move on. Another urchin. feel like this is kind of in the dive of plexorids.
Yeah, we are in their range. We are in their most, where you find most of them, kind of right above 1500.
Another sea cucumber that looks like it's going to starve to death soon.
Another rock pen. Some more Nelsamia.
Large coral coming into view here.
Oh, nice.
Bunch of different crinoids.
Let's take a look at the polyps.
You darken it up just a touch.
Own it.
You want to zoom?
Yeah, you can just, we don't need a land.
Okay.
This is that.
Just do a. We can just do a snap zoom here.
It's a primnoid. I can't remember what the. Is this that one we saw like a couple dives ago? Where we saw it and there had only been like 1 or 2 sightings before with one collection on the Okeanos.
Right. Is that all you need, Alexis, or do you need more?
I think that's good. Let's
Okay.
just do a wide out. Yeah.
Several ophiuroids.
Yeah, there's a bunch
2
of
different
little
colorations,
ophiuroids.
yeah. Alright, anything else here?
No, I think we're
Alright, I think we're good.
Oh, there's a little anemone. Oh, look at that. A little snail or hermit crab. I don't see the crab, so
No, it's up face down. Do a little white balance here.
there we go.
Good to move on?
Good to move on. Yep.
Ooh, skeleton. You can snap zoom on it, Brian.
Okay.
I just want to see
Micro to zoom.
what the morphology. Let's come out a little bit. Yeah.
Sure.
Yeah, it looks like a big Nelsamia.
Yeah.
Okay, that's good.
Alright, forward.
It is dead.
Good observation.
Correct,
there.
J-Rod.
It's your moving on close and typical. Spots big cool, buddy. Ciao.
Big anemone.
Yeah, pretty anemone. Why are these sea cucumbers on rocks? It just seems strange to me.
Maybe they. I don't know.
Maybe they got lost.
Maybe they like it. It's a Canthagorgia.
Yep.
We're definitely getting some bigger colonies as we move
Yeah.
up.
Not that one.
There's a rock right there.
Oh, it looks like
Oh, here's a nice.
it's like good little outcropping of different things.
Yeah.
Looks like it's all down the right.
Something violent must have happened at this size of a rock just sitting right
Huh?
here. Something violent must happen at this big of a rock just sitting right here.
Volcanic eruption violent enough?
Yeah.
Geology.
Rocks and geology.
What-
Anything interesting on this one, or just a quick scan and go to the next one?
I don't
Big
see
fly
anything
trap
that
anemone.
stands out and that we haven't gotten a good look at. Do you, Alexis?
No.
Okay. And yeah, can we just pan down this big colony, this big rock of funness over here?
I'm trying to click on these buttons so I log things, but there's so many things. Some plexaurid. These look like plexaurids to me. Almost all of them.
Yeah.
So shrimp in that hole.
It's a holy shrimp.
Right. I didn't see anything in there that needed anything additional, so let's continue on. That was such a nice flyby, I might try and mosaic it later. We take a zoom on that sponge on the left there. It can be pretty quick. That's good. That's all we needed. I feel like all I've said this dive is there's another plexaurid.
Yeah.
I'm really looking forward to seeing what that multicolored plexaurid looks like in the lab. And that
It is cool. They're cool. I'm more used to a purple and yellow versus blue and yellow, so I
Yeah.
want to see the color. It'd be fun to look at that at 2 in the morning.
Yeah. And here's another plexaurid with an astroschema star, brittle star. Crinoid on the wall. Still seeing these small encrusting sponges pretty much everywhere. Primnode coming down the right side of the screen.
Quivering plexaurid. You see how it's like they like
And shaking.
Updated waypoint bridge. Thanks, Tony.
Little plexaurid at the center of the screen, little anthomastus or mushroom coral over on the right.
looks like a marilla that's been overgrown by zoanthids.
Yeah.
We're above 1,300 meters. We're now at 1,286 meters, with our shallowest depth today being around 1,100. Yeah, 1170. But it's kind of at the top. Osa, corallivory to the
Oh,
right.
yep. Can we go ahead? We might sample this, so if you want to settle down in a.
Oh, that's Caligorgia. It's also a different coral.
One than the other.
Now it's fine.
All right. So it looks like we've got a example of corallivory here. So the sea star is working on munching on this primnoid. It looks like we got another Bursinga at the base. It's becoming a pattern.
You want manual?
Yep.
You got
This
manual.
is.
Thank you.
Can we look at the coral first? Just like come out a little bit so I can see the branching. Yeah, Caligorgia.
So it looks like we've got a couple brittle star associates, some hydroids there, growing on the dead coral, and then corallivorous goniaster sea star. So get the brittle star for Tim. Got another smaller one here. You can't see very well. It's wrapped around the other side.
Tim said maybe evoplastoma for the sea star.
For the sea star.
Yeah.
Lots of brittle stars. So look at how the tissue is dead here on this base, where it almost looks like the sea star ate its way up one side, but didn't actually eat all the way around the branch. You still got some live tissue on the left side of the branch. Yeah, this has still got some kind of something left on the branch as well. A little bit, and just a little bit of live tissue out here on the edge. All right, so
Will you?
what? All right, well then let me. I think we got good imagery on all that, right? All right, I think we're good here. let's keep on going.
Yeah. There's a big
It's
something.
a big, yeah, I'm not sure if it's a brittle star or a sea star yet. I'm guessing sea star.
But yeah, big sea star.
Yep. So the second one of those we've seen today, it looks like there's potentially, yep, there's a dandelion siphonophore just above it.
Oh yeah.
Keep motoring.
Yeah, keep going. We've gotten a good look at those a couple times in the last few days.
What's that red thing right there?
Which red thing? Where?
Is that a mushroom fall? That's sucked in?
On the left? Oh,
What?
that one.
Oh, I think.
Most likely, yes. It's
Yeah.
probably a sucked-in mushroom coral.
Keep going?
Yep, keep going.
Lots of flex art still.
Nice little bamboo stalk here with what? Hold
Anemone.
on. Oh, it's just an anemone, right?
It's just an anemone.
Yeah, it's true.
And a
Halosaur
crinoid.
just kind of photobombed us here.
No. Looks good. Shot pretty in.
Another one of these long-armed shrimp here. Two of them, actually.
White now.
We've been wanting to look at the sponges too, right?
Yep. Leaning towards dead.
What's that guy over there? That's a big
Yeah, looks
Yep.
like
Sponge
he's
is definitely dead.
just keep
having
moving
a
on.
hard
Yep, that's all I
time.
need.
What do you think of this coral over here?
It's purdy.
That looks cool.
Is
However,
that the one that we saw the fields of them?
No, it's not the same species, it's the same family of coral, but not the same.
So kind of.
So they're cousins.
Big relatives.
They're relatives.
what is
Yeah,
that?
what's that flappy thing?
What is the? Yeah.
What side do you want to be on?
On the
I
left.
think we want to be on the left.
Okay.
Oh, it might just be something that got caught.
I think it is, but it almost looks like
Look how it's growing. So the coral's growing straight.
it kind of looks like a sea cucumber that's been deflated and then got stuck.
Oh, you,
Right. Let's see what the
Looks like
unknown
California.
unknown floaty thing is here first. Too fast.
yeah, well, those sea
Yep.
cucumbers that we see
That
in
is
Florida.
This
That is a.
flying what is it?
It's not a plagiaria. It's
Oh.
got too much color, I think, to be a plagiaria. But yeah, it looks like a sea cucumber that is no longer with us. So we got a couple dead stalks here. Lots of brittle star associates. A couple crinoids. Looks like there's two different species of oafs.
Yeah.
The pale and the red.
Both Astroschema, so
Yeah.
they have the smooth arms.
And let's get you a close look at a couple of the polyps. Couple amphipods mixed in there as well. See the reflective
Yeah.
eyes looking back at us are amphipods. Need anything else here?
Nope, I don't think so. It's 2 colonies actually.
Right.
It has 2 bases.
Oh yeah.
All above
Right,
that,
let's go. You know, as many sharks as we've seen in like the shallow waters, we haven't seen any of the deep water
We
species
haven't,
yet.
yeah.
No 6 gills or anything like that.
It's all catch shark.
it's all cat shark, yeah.
That's right, you did. I missed that.
Big cat shark.
Jimbo shark.
Jimbo shark. It's a big cat.
Because he's a cat?
Yeah, big cat, big cat. Man. Feisty cat.
He is
Swords, and Enelopsamia. I still feel like I'm pronouncing wrong.
Say it again.
Enelopsamia?
Analopsamia.
Enelopsamia.
Who knows? Steve's probably like,
Yeah, he's probably
Enolopsa. Like, can't even like make it up.
These were everywhere.
Yeah,
Yeah, the
yesterday was a
Atlantic
really
gorges.
good dive for those. Even a little bit earlier today, we saw a good
Yeah.
little cluster of big ones. So this is a member of the family Chrysogorgidae. These are super pretty. I think of them as Dr. Seuss corals. Victigorgia, the purple coral over here is a Victigorgia. We saw a good number of those earlier, but then they've kind of been not around for a little while. It's the first one in a couple hundred meters we've seen.
Those are Gorgias as well?
They're their own family.
They're their own Gorgia.
Yeah, they're their own Gorgia.
There's a
Little
sponge.
Gorgia, Gorgia, Metalogorgia.
Acanthogorgia.
Remember that one.
Another nice little tube sponge here.
Are those euplectellins? No?
I don't know.
Oh, okay.
I have to sit there with
Sponge
it.
guy.
I have to sit there with a key.
Cucumber.
It's got some spots on him.
Yeah, it doesn't look so hot. These are the same general families we've been seeing all this dive for the most part. Lots and lots of plexords, the little yellow ones, and decent number of the primnoids as well. It's interesting I know the plexords tend to be smaller in the deep sea, but they're all about the same size class. We haven't seen
Yeah.
a
We
lot
saw,
of
like, the
variation in the sizes.
big one on the rock,
Right.
but
The big one we
we
sampled.
haven't seen like when we were in 2017, a couple times we came upon really large plexarids hanging off of ledges. Some primnoids.
Yep. Little anthomastus at its base. Another fish. Probably a snapper blanket, but didn't get a good enough look at it to be sure. I'm enjoying all the crinoids. I've always just thought they were super pretty organisms, and seeing them on so many of the corals is quite pretty.
Quite pretty.
Yes, when they're swimming is a lot of fun.
I don't think I've seen any swim on this cruise
I
yet.
don't think I
We
have.
had one small one, but it was only on camera filter.
Oh,
Okay.
another sponge.
Not a lot of sediment.
No.
Like, I mean, like big, like.
Yeah. Is that just another plex, or are we seeing? Can't tell. Yeah, I think that's just another plex orb.
There you go. Yeah, it
Yep.
just doesn't have very many branches.
Alright, I think that's all we needed for that one. Thanks.
It's another Caligorgia and Plexarid.
Anthomassus coming down the left side.
Big sea
Sponge
cube.
or sea cucumber.
Sea cucumber, definitely.
That one's healthy.
Well, we've come up quite a bit now. We're at 1,233 meters. Our shallowest depth we're expecting to reach is about 1170. So we only got about 50 meters to go until we get up to our highest point, and then we're going to go down into a little hollow area. After that,
It's a couple
Little-
little
yeah,
Anthomassus all together.
cluster. Another urchin here.
Lines in that one.
So,
Looks like somebody's trying to scratch it away. It's like, when you look at the ring, bottom of it.
the well, yeah. Yeah, the bottom of the well.
A couple different types of primnoids. Looks like we have Caligorgia and Norella.
Oh, shrimp. Still seeing these kind of ubiquitous small encrusting sponges everywhere. I think it'd be possible to sample that one, Jaron.
That flat spot?
That flat sponge, yep. We've been watching for like an hour now, trying to find a big one that would be easy enough to reach, and that's about the best candidate I've seen so far that isn't like small or under an overhang. Don't know how difficult this is going to be to peel off the rock.
Suction.
J-Rod said they.
Suction? You want suction or
Have you done one of these before?
one of these?
So I'm not entirely sure which which will work best.
Try suction.
I
Low.
kind of would lean towards suction, but if you tried to sample one of these before, I would bow to your
It'd be nice
expertise.
if we could find a rock that had it on there, huh?
Yeah.
We could sample.
Then bring out the suction with the new and improved handle.
See that.
I need to get you closer, huh? Looking like a knot line. Have we taken a suction sample yet?
Yeah.
We have not.
Jared was actually gonna come in at 8 to do that. What's that say?
Go juice on.
You ready?
Go juice on.
What's your spread? Zoom in on set. That was working. It's just I don't think you're gonna get the whole thing in that one go. I think everything. Is it okay getting like little pieces like that?
Yeah, we'll
I'm trying to second one this a little bit.
obviously bigger pieces are better, but we'll take what we can get.
I'll keep an eye out for a rock as well. That
Okay.
has one on it.
Yeah,
Yeah, it's soft. It's very soft.
yeah, that doesn't surprise me. Hopefully we'll be able to get some of the spicules out of it to take a look at that later.
I don't think you're gonna get lower than that.
There's
Okay.
another one that was like right to my right. Right there.
You want that one too?
Sure, if you think that's, you got a good angle at it.
What jar is that? 2?
What?
Jar 2?
I think it's jar 1, so that's 8 and that's 2 on the
Oh,
other
gotcha.
side.
We'll find some rock. Pour jug over it? Okay.
Alright, let's keep going.
Pour eyes,
Yeah, well. So when you're on watch, you're awake.
good.
Yeah, you should have.
Go ahead. I watched the guy. It's good.
Why don't you go to bed now? I'll stay on.
Oh, no. Yeah. Joking. Why would you choke that, man?
Keep your eye out for a rock. That's one of those little impressive sponges on.
Nah, I can't. Sponge watch.
Come on. They said they're gonna make me run my shit so far.
Does it make you a room?
Huh?
Does it make you a room?
Yeah.
So what else is going on?
So next sample number is 381. We have honestly comparatively very few samples. It's been lots of primnoids, lots of plexaurids, and that's about it, frankly. A few Ecological, 1 or 2 sponges, other than
What do you call
nothing primnoid.
What?
I don't know.
I don't know. That's a primnoid. Huh?
Yeah, that's fine.
Okay.
We did find one really interesting plexaurid that we sampled that was multicolored, the blue and yellow one. Okay, so then since then, basically nothing but small plexaurids and
I've been watching. I didn't know if there was. Do we need to? There's something we're looking to sample like a.
Be right back. I'll be right back.
Any priority samples?
I think so. Not that I'm aware of. I think we've got enough. The one thing
Randy wanted a Walteria.
which we got a small chunk gone that I'm not sure is enough for her.
I don't know.
But we've
Did you
only-
see that?
She kind of
It's like a
indicated.
piece of sponge like this big.
It's also early on. I mean, early on, so it's been a lot.
But she wants to use it for the respiratory.
Okay.
But that should be enough.
Oh yeah.
I mean, the
If
things
it's that big,
are
yeah.
tiny. Like,
Yeah.
Oh, yeah.
For the restaurant, right? Yeah.
yeah.
Anything else to be aware of? Can we use your computer?
I don't think so.
I got my computer.
Roger that.
I don't think so.
Yeah.
Right.
You want to know a sample number?
381 is next.
I haven't sampled a lot, so.
Okay, cool.
I feel like
Normally you don't sample as you sample less as you go, right? You've already sampled most of the stuff.
we have not sampled a whole
What's
lot.
that?
That is Narella.
That's the round.
I don't know. It's from nowhere.
Alright, you gonna see spider yet?
No.
We did
We
not.
haven't,
All
seen,
right.
we haven't seen
Okay.
the sea spider at all.
Let's go.
Well, they saw one or you missed
No,
it.
but
We're on.
sorry, since the
Oh, yeah.
like,
Since
Alright.
Attack.
Hey.
Are we, is there something you want to see here?
Nope. We're gonna just, I'm gonna get organized here and we're gonna just keep going. No, you know, I've been watching this old, so I know what we've seen.
Nice.
Right? So I'm super tired, but let's go. All right.
Only been
You
up
ever gotten
since-
the chair super tired before?
Super tired. One right there under the rock.
What?
It's like a superpower.
What?
Crab.
Oh, we can look at the crab.
Figured.
There we go. It's the one thing I will stop for.
Break for crabs.
We'll stop for crabs. A new bumper sticker. All right.
I'm gonna land out here. You're gonna see him from there.
That sounds good.
Get higher up.
That's right. Oh, he's come out for you. Look at that. Looks like a homalid crab with an anemone. Well, let's check it out. We had a lithoted crab earlier on a coral, which is really neat. That we,
What's that? Nice hat.
yeah.
Go for it.
Ready? Can we draw down the lights a little bit? Whoa, we got a runner.
Frame grab.
Yes, a homalid crab. Dude, he's gone.
He's like,
oh man, what's going on with that? God, he waited for me to zoom though, didn't he? He's like, nuh-uh. So they grab anemones or they grab coral or sponges and they put them on their modified back legs and hold it over their head as if to camouflage themselves. And maybe they do. Maybe it's a scent thing. Maybe they're disguising their scent with these animals on top of them. I don't know.
I think that's the fastest I've seen one run away
Yeah,
from
they-
us, though.
They don't usually take off like that, right?
No.
Never had to
No.
keep up to a crab with
He's coming for
Buddy,
you.
you're expending a lot of energy. Careful.
I just tilted down a little bit.
the.
Okay. I'll do a little bit of this. I'll do that.
It's crazy.
Let him run in. Nice job, Adam.
Oh, he's not gonna stop there. He could be on like the Great Crab Race and we just don't know it.
I went too far.
Gotta make it 3 miles today.
All right. I don't know why you try to hide with an anemone when you run like that. You're gonna run into something, right? It's laying over his head. That's a great shot. Little frame grab on that. Nice. It is fun looking. That's
yeah,
great.
look at that.
All right. Nothing but anemone here. Nothing to see here. All right, and we're off. Thank you, Adam. All right. Okay. I think it's clear that we're headed for 1171. Sort of our next high, a waypoint. It's only 30 degrees elevation, I think. Okay, 0701, we're on.
It's a big
Level
base
11.
on that. Hermioid. Excuse me.
All right. We're on Titov seamount.
fish.
And we're seeing some PRIMNM corals, Flexard corals in the bottom right there, the yellow with the atroschema. This is some sort of fish right here. What is this?
Mm-hmm.
Toadfish? You want to look at it, Adam?
Yeah.
I know you do. I didn't have to say anything. Yeah, absolutely. Yeah. Nice. You should do the camera. You're better at it than I am.
Oh no, go for it. It's all different than I just
Come on
this
now.
rock here.
Is that what it is? A toadfish?
Yeah. That's what I call them anyway. All right, we got a little bit of light. You good? What's that?
Is it injured?
Looks like it.
No?
A little scrape there.
Nice. Oh, definitely.
There we go.
Poor
Let's just
guy.
hold that for a minute. I can see the lights in your eyes. How'd that come up again?
He's in
Is
recovery.
that you?
Yeah, it's me. How that?
Look at all those sensory hairs along down the sides.
This is
It used to be
interesting
close to
for
the bottom.
wounding. Like, how does an animal like this successfully? I mean,
You want to sample that? Do some
no, gosh, no.
culture some bacteria out of that thing?
No.
I know a guy.
I know a guy.
If we weren't in the,
You've got the iris. We'll take it. Can you bring it down, Jared?
You know,
Yeah.
the iris is
It
just
seems like it's really hot, right?
here.
Or is it something else I'm doing?
No, it's. I'm just gonna try something quick.
Go.
White balance in auto and then come back to it.
Oh, cool. Let's see what
That
happens.
seems to change the ISO. Yes, okay. There we go. A little better?
Yeah. See, we got those hair-like projections on its little feet there. It's modified fins for sitting on the seafloor. Go back up again. Is it in some sort of auto?
It's the problem is you got control up here, and control here.
Yeah.
So sometimes when we switch between auto, it kind of
Just-
just loses its bearings and then it
Yeah.
just stops working. So you have to like.
sorry, I'm not getting a good picture of that.
I'm gonna call it a little bit. I see a little fishing lure just on its head coming off. See how it fishes. Just hold that for a minute. Are we good?
Yeah.
All right. Let's come out. Whew. Oh, whoops. Wow, that really stuck on me. Sorry. All right.
After having a big chunk taken out of it, it seems involved.
Something happened, right?
Did you think one of those
See that sea star 4 times
eels
now.
or something could have done that to me?
What's that?
Looked quite like a chunk, like a
I know,
What,
yeah,
out of that
what took
fish,
it
you mean?
out of
little
him.
round mouth.
Yeah.
A really round
Could
mouth.
have. You think it was in a war with another fish or something? All right.
There's a sea star right there, that big puffy one. Again.
That's a Hymenaster sea star. We saw that earlier.
On this dive
This
or
is
a
slime,
different one?
slime star. On this dive. The slime star there. We saw that earlier. See a lot of holdfasts on these rocks here where corals used to be. Or sponges. No way now. I'm okay with that.
Yeah, you gotta look for a smaller rock.
A lot of PRIMNM. They saw a lot of corallivory on the last watch. Sea stars feeding on PRIMNM corals.
Oh.
So we're diving on Titov seamount. It was discovered in 1961. It was named after a Russian cosmonaut the ship named Vityaz discovered it, and Titov, I think his name is GB Titov, was the second person to orbit the Earth. So they sort of awarded him with the name of the seamount. So seeing a lot of these,
Yeah.
I think we've seen these the whole time. These are the primnoids.
Definitely feel like we're in a bit of a forest, though.
Yeah,
For sure.
It's high density. It's high density. I wonder if we should take a Niskin. It's
Yeah.
not high diversity, but
I don't
it's
think
high
we've taken an
abundance.
Niskin yet, so.
Oh, I know.
Want to do Niskin?
Yeah, let's do a Niskin here, just because we got so much abundance here.
This is our first discussion.
First Niskin. We're at 1199 meters.
Alright, so I will press sample in C2.
What?
Number 381. Make sure it fires.
Roger that. So I see the yellow plexards are here, the primnoids are here.
Is that a confirmed fire?
Confirmed fire.
Yeah.
Thank you.
Okay, 381. You got the Niskin
Yeah.
number 1.
And we are at 1198 meters.
Yeah. What else is here?
Lock and dry.
Stalks are crinoids. Hey Cody, how are you?
Hello, sir.
How are you doing?
Good.
Good.
Keep this train moving.
Yeah.
Choo choo.
Yep. Hey James, can you put Fledermaus on the screen?
Yeah.
Showing Titov. Oh, there's Caligorges also here. We'll write that one down.
That's pretty.
We've seen that. I think they sampled it. They sampled Caligorgia somewhere. That may be Calyptophora.
Calyptophora, I
That
see.
may be Calyptophora. Looks more like Caligorgia to me. Oh.
Is that? I
Yeah.
feel silly
Let's take a
asking.
quick look at that one, Cody. Just to make sure.
Which one, bro?
The white one, sorry.
Yeah,
This.
that's fine.
I knew you were on it anyway.
There you go, sir.
All right, can I have the lights down a little bit, or whatever you want? Is it lights or is it ISO?
Alright, so this is a primnoid
Can't do it.
again still?
Yeah.
Okay.
Whether it's Paraclyptophora or Calyptophora, I'm not sure, but I think it's Paraclyptophora.
Working on
There's a nice mysid shrimp in there. A couple of them.
You may darken it a little bit, or you good?
Please, yeah, can you take it down just a little bit? Bunch of mysid shrimp in there. At 2 astroschema.
How's that?
I was like, that's great. Beautiful. All right. That'll be enough for ID. Base is covering a bunch of stuff.
It's
great.
It's cool.
Oh.
Ready for it?
Yep, ready. We're now at waypoint 4.
Yes, sir.
Joe?
There's another sea pen, I think, under that rock. Or it's a crack. Hard to say.
You see it?
No,
I
it
don't
went
see
away.
it there.
It's a crack.
Oh. Oh yeah. Can you zoom out to show the seamount? We're on. Oh, I see. And then turn it up. Yeah, right. And then can you zoom in a little bit? I'm just trying to. So we're seeing the same corals over and over again. We're going to pass by them, so we want to get to all of our waypoints today. But I don't know if you can see it on this seamount. There's several spines that come off, or large ridges, and we're running on top of a ridge right now. I think on the southeast side. But there's these 4 major ridges that often come off these seamounts, and those were formed when the volcano was eruptive and active, and these are actually dikes. These are actually dikes of lava that came down on different sides of the seamount, and it was much denser than the rest of the lava that was coming out. And so over time, millions of years, like 100 million years now, that those dikes remain in place, and the rest of the lava has worn down and worn away and eroded. So it leaves these spines, or these old dikes that formed the volcano or the seamount 100 million years ago. And so we're diving on a relict ridge caused by dikes of lava that formed when the seamount formed. So I always thought that was pretty cool. That's why you sort of see those ridges as James spins the seamount around for you. You see these raised ridges on the corners, basically. And so those today are what becomes habitat for corals and sponges like you're seeing on the seafloor right now. So this geological process that happened a long time ago.
No, that's fascinating.
And the nature of it, ultimately formed these habitats. So we know very little about the geology of these seamounts, though, and that's why we're collecting samples on these dives to give to our colleagues at USGS so they can date them and chemically analyze them. And the crust that's formed, this dark crust you see, maybe just a few centimeters thick, but it's a chemical alteration of iron manganese. It just coats these things over time. But inside each of these rocks you see is actually basalt from when it erupted on the seafloor. It's a little forest here. Primnoids. With some ophiuroids. I tell you, the primnoids are the ones that really like to host a lot of different species on them. The crinoids you see, that's the feather stars, the ophiuroids. You know what we haven't seen is an anemone on a coral yet on this dive.
Okay.
Just saying. As far as I can tell, we haven't seen
Just
one.
on the crab.
As soon as I say it,
So
we'll
the
see it.
hamad, yeah.
Where
there
is
you
it?
go.
You see it?
No.
Oh.
I said we just saw an enemy on the hamad. My bad.
Oh,
To
you're
get
right.
everyone
You're
excited.
Right. There's a nice big urchin. We've seen several of those too. Really, way more abundant though. I think it's the most abundant we've seen these primnoids here.
Yeah, I keep just typing like primnoid rich
Yeah,
area in here.
Just keep hitting the button, right? 1186 meters depth. Big stalk crinoid. There's a lot of animals that live in those, amongst the feathers of those stalk crinoids. You could see a bunch of them from here, worms and things. Hold on, what is?
Oh,
Is that a sea star? What is that?
Oh.
That's an anemone, I think. Let's go and take a
Oh,
look.
because you said it. That's hilarious.
I know. As soon as I say it, right? It happens every time.
You're good to go, sir.
Thank you, sir.
There's also a sea star. Nope, that's a crinoid.
So yeah, I had that's actually not a ring anemone. That's not a normal anemone we see on corals. This is, I wish I knew the name of it, but we see that out on the rocks a lot of times. So this was just found in an advantageous spot to get onto. Probably a dead branch of this coral, and just found it.
Definitely.
A lot of brittle stars here. A lot of skinny arm brittle stars here.
It's
Two
interesting
different
how
species.
it's like
What's
stalk
that?
almost looks. I mean, I know it's
Sorry.
not, but the stalk of the anemone looks like a glass sponge to me, just the way it's like porous.
Well, you see how this one gets on dead material or on rock, and I'm trying to pan down a little bit here.
There's a ton of little
There we
baby
go.
Astroschema.
So the ring anemone is the one that get on the corals that have a lot of polyps on them. They
bumps.
actually land and they absorb the polyps into the bottom of the anemone, basically. And so unlike this one, that's an OFA we haven't seen yet.
It's very pretty.
There's quite a bit
Good
of dead
color.
and live here, kind of funky growing. I think it's two different corals. I think a coral grew on a coral here. This is a super old coral here, right? It's been long dead, and now something has settled in here and grown
But you
out
can
of
see
it.
that base right there on that one.
Yeah, yeah, that's right. That's cool. Wow. Lots of activity for that little coral colony. All right, we're good.
Good?
We're good. There we go.
Keep looking for small rocks.
I just saw your picture flash up on the screen.
Oh.
All right, still seeing the yellow
Oh, there I am
plexaurids.
again.
718, 1182. Like when these manganese crusts form on these rocks, they get really smooth, become really weathered. It's really all, see where they're draping down. This is an old pillow lava, maybe some lobate lava.
Sea stars, and we only saw one of
See
them.
what? Yeah, can we take a look at that? Please. Thank you, J. Rod. I was looking at the rocks.
Were you looking at pictures of yourself?
I think she was. I don't want to.
Captain.
Hello, Captain. Oh, we saw this one. No, this is actually a new one, I think. We haven't seen this one yet.
I don't recognize it. I was like, maybe
You ready
it's.
to go?
Oh, you go.
Most of them we see are very
Go captain, my captain.
puffy. This doesn't.
I don't know. We've been making sure we document the sea stars because they are known to be predators on corals. But I don't think we've seen this one yet. So we do a good job of documenting these things best we can.
Thank you for that.
Sorry, there we go. Come on. Tilt down. Thank you.
I'll try to make it what we need.
Hold that for a minute. We'll take a few frame grabs of this. Make sure I have focus on it. And
Alright.
good, right? All right, great. Thank you. Come around front again.
I did the same thing. Same thing. No.
So by our earlier calculations, we were on like a 25-degree slope here to 30-degree slope coming up. Corals seem to like it.
Very
When
much.
we pick our dive plans, we often pick an area that has up to 40 degrees, 45 degrees slope, so it's pretty vertical. These corals love it because they're out into the current in a productive place. So yeah, this is really abundant. Lots of coral here. It's funny, a lot of these colonies are the same size. I feel like they're not these primnoids. I feel like they're just
Yeah.
it's like multiple recruitments, like small ones and large ones and different generations, if you will, but it's like all very similar size.
Yeah, it's interesting. I mean, not super high diversity in terms of corals, but the associates, I feel like, are pretty high diversity here.
Can we get a snap on this guy over here? Just got it. Can
There's
just
another one below it too. Maybe that same kind we just saw. See how we're going
That's
past
the same
it?
one we just saw. You're
I
right.
believe so.
Yeah, I think that's the same one down there. There's another one just below that rock.
Mhm.
I think it's that guy. I call these neomorphasters, but I think that's not the right name. So I'll just go ahead and make the mistake now.
I'm gonna call him skinny
That one-
legs.
Let's just go for that one. Yeah, I saw that one. I think we just imaged that one. That morph already.
Another one right there.
Yeah, right. So this is a different one. You're right. Good catch.
Yeah, he's spikier.
Yeah. And I just had a name on this guy yesterday because we did sample something similar to this, I think 2 days ago. They look a lot like Brisingid sea stars we've seen next to and on the corals, but they're not. This is outstanding. We have not seen this one before. I'll try to get in close on this.
And get a frame grab here.
Yep.
Got it.
Wouldn't want to step on that one.
No.
I'm gonna go
Do you
all the
think
way.
those spikes are poisonous?
I doubt it, but you
I
never
think it's
know.
just for physical defense.
Wow. Just gonna hold that for a second. Yeah. Amazing. There we go. All right. That's great. And then we had that one up here that we already imaged before. I'll go right in quickly. See if I can get past that. I think I am in manual. Yeah. Hard to see that one. And then the last one was this thing I call neomorphaster, but it's likely not.
Skinny legs.
That's an Atlantic species I think of. The stars are out tonight.
Stars at night.
Just singing it. Big and bright, deep in the heart of Texas.
Alright. I'm gonna go all the way in. Excellent. Holding. Alright, we'll come out now. That gives us several frame grabs.
Yes, I have a few frame grabs from that experience.
Yeah, so now we got it. Still the yellow
2H
plexards.
star.
So the primnoids. While we're here, I'm just going to sneak a peek into this plexard right here, because I do care about what lives with them. And let's just make sure we haven't really zoomed into one of these for several hundred meters. So let's just take a look at this ophiuroid. Trying to get some focus on this. There we go.
Nice. Alright, get a frame grab.
Went too far. All right.
It's hard because it's in a different.
All the way in. All right. Let's give a few seconds on that. It's really hard to identify these ophiuroids through video. Actually, you have to look at the bones of the disc or the ossicles and dissolve away the tissue, and then you get real diagnostic characters for these little stars. But we come to understand the morphs a bit better when we do the video. Thank you so much. That was great. Yep. Let's roll on.
doesn't end.
What's that? Oh, I know. On and on.
I don't want it to end either. It's nice to see.
Straight down.
What'd you see? I missed it.
There's
No,
a
it'd be nice
small
to
rock
see some.
I was
that
watching.
has that sponge that,
Oh, is that right?
yeah, we had a hard time sampling it off.
Yeah, I saw
think
that.
I can pick up that little rock that
All
has
right.
one on it.
All right.
Go for it. What was that? Fish?
Definitely. Good call.
Nope.
I'm sure that was a fish.
You don't think so?
I don't know.
Do you need to get
It
an image
must
of
have
it
been.
before I sample it?
Yes, please. If you don't mind. Just a quick. You got it?
Yeah. Oh, yeah. It's nice sign.
Which one you going for? This one right here?
That, no, the rock.
You want the-
Up and to the right?
Yep. Okay.
Okay.
On it. There's barnacles on it too. You're in chamber two? You're lined up?
I'm pick up the rock. We couldn't scrape it off.
Oh, you're gonna pick up that rock?
Yeah, we were gonna look for a rock that had it.
All
on
right.
it, 'cause the
All right.
Sample that we did.
All right. Yep. Green light.
There it's,
Camera's yours.
That is quite tiny rock. That's what to tell.
Oh, little squat lobster back there. Sorry. Where is this? Hey, Randy. Okay. Rock box. Can you hold it and let's image it before you put it in?
Yeah.
Please. Great. You got it. You got it. So what we want to do is look at, see if it has any kind of alteration on it before we put it in the rock box. Hang on a second. Is it?
There you go.
You got it, right? No. Hang on a second. I'm gonna go in. Is it otter now?
I'm gonna take it away real quick.
Yeah, take it.
See if,
So it was barnacle side down. J-Rod, can you just rotate it clockwise?
Price is right.
What's next to the sponge? That little like, is it like a limpet?
No, I thought they were barnacles.
Those are barnacles, but on the
Oh, I'm
left side.
sorry. Oh, there could be limpets on here. Little ones over here too. All right, it's got some of the. It's a little stain on the underside. Okay.
Would you call that
Great.
angular or not?
Yeah. Triangular.
Flat and triangular. I like that.
It is. Hi.
Hi.
Decided to sneak up right there.
Just
I
don't.
didn't know you were here.
Yes, please. Great. Thank you.
How are you guys doing?
Good. Someone I know has been up for a while, but I won't say who unless they want to reveal themselves.
Me? I know. I'm tired too. I only had How's that summer
Rick?
doing?
You got it already?
Seems excited now.
no.
Oh, wait.
Let's go.
Look at this. Have you guys seen any more black spots?
Give me that. Yeah. What's the number?
382.
382.
And we are at 1176 meters.
Look at all this. Oh, it looks so.
Just this is. We've been in this for a little. Yeah, I don't know.
The whole time.
For what?
We've been in this, half hour.
See any more black spots?
No,
Black spot.
no.
Nope.
Haven't seen any more half damaged corals or whatever. There's just one patch.
Yeah.
Geographically, there was one patch right like that.
I'm not really awake. I'll come hang with you guys for a bit.
Me either. I think Anna should sit here.
What?
I think Anna should sit here.
I think Anna should be there too.
You don't have the history you do.
You don't need
Stories.
history. Where? We got that one. We imaged that. We imaged like 4 sea stars.
Oh my goodness.
Different ones are there.
Sea Star City.
I
Oh
know.
no, I'm just gonna hang out with you guys for a little bit and then we'll see where
Three of
things
us
go.
will fit.
Is that an enemy?
Yeah, let's look at that. Like maybe Anthomastus. Yeah.
Oh.
Yeah, we need to go on that.
Jinx.
We're seeing the same stuff, but we're also 1175 meters.
Yeah.
So we're getting there, right?
I'm trying to figure out, maybe James can help us figure out when do we get to the top of the ridge? Where are we?
What's that?
Where are we in relation to the top of the ridge?
Oh, there's a squad.
Oh, there we go. Oh, we're rounding it now.
I know. We're there.
Yeah, we're there.
Oh.
All right. Well, so if you are listening in, this dive track is an interesting one because we're
I'm fighting somebody.
climbing up the outside of this
Fighting.
feature, getting
Right.
to a ridge,
Let's go.
which we're just about to kind of round the corner on, and then we're gonna kind of go over the ridge and into this feature that looks like it's been,
A depression, more or less.
yeah, like a.
I don't know if we were gonna sort of slide through it. And maybe it's starting now. Here comes the sediment.
Yeah, no, but it kind of like sinks down, and so we're not sure whether this will be like a big cutaway formation with fossil reefs, or whether or not it'll look like
Yeah.
exactly what it looks like now, or whether we'll see a change in species distribution and abundance because there'll be a current change.
I mean, so far we're seeing the same thing. It's like a $2 buffet, right? It's all the same thing.
Yeah.
What is that, a fish?
I don't know. I don't know. It looks like I can't.
It's a sea cucumber stuck in a
Oh,
coral.
You're right. Yeah, Ecological variant stuck on a coral.
Yeah. There are bad days and there are bad days.
That is a bad day to be
that's
a Ecological
That's
variant.
a bad day. And you're in the dark. You can't even see how to get out, right?
I know.
Mm.
Well, they don't have eyes, even if they could see how to get
Don't
down.
have hands full of flashlight either. So
That's
Urchin.
That's right. Not even a pen light. It's true. You can angle up a little bit of fence.
yeah.
That is helpful. Another sea star there. Do you want some coffee? Go back to bed, right?
I want to do one of the two. I'm trying to decide which.
Well, I don't know if you want to, how long that's going to be. If you want to, imagine you want to see the other side,
I want to see the other side.
right? And then you have to make a call, right? to-
Exactly.
So,
But then we're gonna be up in the lab. I've just put my,
well,
yeah.
we can.
Right. So
what does that mean?
If you don't want to, we don't have to.
I mean, we have Walteria. We probably have enough from
Oh,
knowing
you
for one more
wanted to
experiment.
leave.
Sponge while we have it.
Okay, cool.
Don't you?
Yeah. Yeah, if we're right back.
I think we should do
Leave.
I haven't looked at the experiment in a while, but I think we should do.
It's almost done. I mean, some of them are getting
First
down
of all,
into
it
the 60s, 50s.
All right, so
like an
I think
hour
we
or
should
two.
do another live dead, straight up, full plate sponge.
Sponge.
Yeah, so we
Okay.
might only have one shot with it. Don't you
Perfect.
think?
Yeah, totally. That sounds great.
Let's just get our first. Let's go for broke. See what we got.
So
Okay.
more Primnoid or more Walteria?
Walteria, if you've got it, but I think we have enough to do one experiment. Let's hope.
What's that? 4 M's? The white dots?
Yeah.
At this point, we're going to aggregate
Most likely 4
bacteria
M's or-
alone across multiple.
Yeah, I don't think they're
across
lipids.
many
You
plates.
can zoom in on it if you want
Yeah.
to be sure, but I think it's,
So I think the design that we
you
have
know,
didn't make
it'd be
sense.
nice if it were coral
10 guys
colonists,
ago,
right?
but
The
it makes
corals
sense
that were
now.
landing and growing, but
You think we should
Don't
have
think that's the
included
case.
bacteria?
We did.
Oh no, but do you think we should not have?
No, I'm glad we did.
Okay.
Well, we had one, but I don't think we need a gazillion of them because if it's way off from other plates, we'll know it. We have enough other samples.
Yeah.
But
we're still coming up, aren't we?
So I think we should. But there's a lot of Primnoid too. So if you think we don't have enough Walteria to run an experiment, you should grab some Primnoid.
71, 738.
They did take a little bit, but it's of that Paracalyptrophora putatively that we have to identify.
Yeah, so, I mean, let's look for the best Primnoid we can possibly find. And when I say best, I
What
don't
does that
mean
mean?
largest or smallest, just easiest to take a little.
We could put a standing
one
order in,
branch off.
like that one right there.
Any of these.
Yeah, kind of. I
I mean,
asked
you know,
for
I
it, it showed
mean-
up.
You want that one? Yeah, I
Yeah.
guess so. Yeah.
Sure.
All right, I'm gonna go back to bed, and you guys are gonna call me when the landscape changes over the top of the ridge, right?
Yeah.
Okay.
We won't make any final decisions without you.
I'm gonna be like
dead to the world.
WhatsApp me and like literally call the room. So I may be.
What's your number again?
456, I think. It's right on my phone.
Okay, great.
All right.
See you soon.
All right.
I'll keep it out in my room too.
Want to image it first?
Okay.
You ready?
Which one is it? Which does-
The Primnoid right here. We're going to take a bit of and make sure we get some
See that
ophiuroids
arm
with
on
it.
the side? See how that's?
What do you want?
Do you think
The clean.
just like right here?
Oh.
Do you think that's enough?
Yeah, but there's no oaths on it.
Oh.
Maybe that's a good. Maybe I mean, you know, you pick
Do you
the part
need
without
more
the oaths.
oaths?
Where? Tiny little things.
Or we can take,
Oh.
something over here.
It's like pointing at the triangle things right there. To your down and to your left.
Right.
He sees
Like
something
blue.
else.
They're right next to an ophiuroid.
What?
Right.
To your right.
To my right.
No, well,
Cody here. Oh, right
Yeah,
here.
you see something in here?
Yeah, here. What are those blue things he's saying?
What? Pteropods?
I don't know. Hang on a second. Let me.
That's what they look like.
Huh? Go in the manual.
Auto now.
Okay, that's the front.
What an
Just put me
eye,
in.
by the way.
Texas. All right. I'll see you guys in a bit.
Going in. So all the way in.
Hard to say.
Come on now.
you're in.
I am. Yeah, I am way behind it too. Here we go.
There you go. Good job.
I think. I don't know. They could be. They could be. I see tendrils coming off them like they're ctenophores or something. But you see them there at the bottom. You know, I see them leading out, but I don't know what those are.
Oh, they don't.
Would you like me to pause here?
No, you can keep moving forward to the left.
How does it jump so far like that? Okay. All right, it's your pick. It's your experiment. I don't have to have oaths. What do you want?
Well, for me, it doesn't matter. I just need pieces that I can cut into sections of 6 whorls.
How many branches do you need?
Let's do like this? Or do you guys prefer this side? I don't care.
Whatever.
Let's get some oafs for Tim.
Oh.
And I figure just take a little clipping on one side. Whatever's easiest.
How many branches do you want?
That's it.
2, 1, 3.
I was gonna say like 5, but
That's a funky situation there with that.
but it doesn't have to be the whole branch. It can be like the tops of 5 branches or something, halfway down.
It's a weird polyp situation going on there. Some overgrowth. Okay. Did you decide? You got it? He's already
Yeah,
in it.
I decided.
He's already in it. All right. This will be a quiver.
Assume so, but I don't know. This is gonna be sample 383.
Yeah. What's wrong with that?
Oh no, nothing. I don't know where it's going though.
Like that much of the branches or more?
Further down.
Yeah, a little bit more.
Just go all the way if you can. Look at the oak crawling away.
Where? Yeah.
He's like, I do not want to be part of
Definitely
this.
disturbed. These other guys are tightened down. Not going anywhere.
I didn't make this easy on him.
Hmm?
I said, I didn't make this easy on him. I have to be better with picking my samples.
It's all right.
Hmm? The escaping one? Oh.
What?
Oh, it says, Noonie asked, what are they saying when it sounds like they're saying oaf?
Oh, for D. I'll after this.
Will that answer that question in just a second?
Not that much?
Perfect. Great.
Snip.
Oops. Sample in C2. Quiver 12.
Okay. Did you get in situ sample?
Mm-hmm. When it was over the quiver.
Good. Like now.
Oh, okay. You want another
No,
closer to
that's
the quiver?
Okay.
You want another zoom?
No.
Nope, we're good. Thank you.
Sorry, we're just joking around. We're at 1178 meters. Oh, 71, excuse me. Okay, you can put this back over there.
Roger.
Thank you. Ophiocanthidae or
Ophiuroids. Ophiuroids. These brittle stars you see are in that classification, Ophiuroidea. So we all just call them Ophiuroids. They're 5-arm stars, if you will, the echinoderms. And so for short we call them Ophs instead of Ophiuroids.
Do you want those other ones right there as well? Next sharp one?
wait.
Sorry, not seeing them, but I
Oh,
was
here,
looking
Tim.
up.
Do you want that?
If I'm not sure. Yes.
If possible, if
Yeah,
not,
yeah.
it's okay
Yeah.
too.
I mean, it shouldn't contaminate another sample. That's the first thought.
Yeah. Leave no sample behind.
Look at that.
Very nice.
That's number. Awesome.
Great. Good sample.
Awesome.
We are still in a PRIMNM forest.
It's about 120 meters. And we are all PRIMNM forest.
With
Yellow
plexwords scattered
plexi.
about.
Yeah.
Astroschema for sure in them.
No, not many chrysogorges tonight, right? We're not seeing Metallogorgia. Okay, there's an old sponge. You don't want sponge, right?
There's a new one right there, hexanellin, and an anemone.
Did you want that or not?
Is that the wolfish they were talking about?
See the pink?
Yes. I think it is. I think it is.
So when she said we got Walteria sponge, is that like
It's
the
about-
conical one?
It's the one that has tons of stuff on it. It's a vase
Okay,
network, right, that has the.
so I think that's what we
Silica.
just saw. We'll go back in a second after we follow this guy. What a brilliant little color. He has a funny little face too, almost like frowning.
There you go.
Beautiful.
It's a bit tricky. They don't really stop swimming, do they? All right,
So
so
see
there's,
that one back
yeah,
by
I
the
see this
road?
one. That's not Walteria though. That's like
Okay.
you platelet glass sponge. There's an old one here, but that's also not Walteria.
Okay.
So
So yeah, no.
it's quite a bit of rubble there, isn't it?
Yeah. I will press the coral rebel button.
Can we look at that for a second, Cody? Let's just make sure that that's. I'm sure it's going to be a PRIMNM, but let's just double check and make sure. There's a big drop off right there. That's really probably the reason why we're seeing so many corals here is the current's going right up over that lip. Yep. All right. Going in.
With some sponge going on.
Wow, it is certainly accreted with all kinds of stuff on it. This is really old. You see, it's branching like a.
Yeah, it doesn't look super primnoid, does it?
Super what?
Like a primnoid branching? Well, it could be a different kind.
It doesn't. I don't. I can't recognize it. You know, it's so degraded. I don't know what it is. I mean, part of the says it's like a. I don't know.
Is it?
I thought
So
now
it does not
Samia
look
for a
like
second.
dead primnoid.
No, there's lots of stuff in here. There's a crinoid that have been here for a while. So this is just.
It's an Elosamia skeleton too.
That's what I thought. I said that, but part of me said it wasn't sure because of that over there.
Zoom back in and white balance while you're in there.
Sure. It's pretty neat though, right?
Yeah, it's super cool.
You want some of this? With white balance.
White balance?
There, the captain's got.
Special.
Captain's got the hair. Fantastic.
Oh, I thought it was at
No.
the store. It was Cody right there.
So do you want to?
Nothing fancy.
So do you want to sample any of this, Alexis, or not? What do you
Sorry?
think? Do you want to sample some of this?
I don't think so.
Now,
Unless you want
this
it.
is what sort of gave it away to me over here, right? This is.
Yeah, it's also you
See,
see the really
now it's
thick.
Samia, right?
Well, you see the thick ridges too down the main spine of it,
Yeah,
and that's
Yeah.
how.
right.
And then all the polyps are.
Oh, look, an ophiuroid.
It's like, what?
Oh look, Tim's zooming in.
I'm trying to get it now, Samia right there, but anyway. All right. I don't need that. I don't need
We're
to
gonna do
zoom
it
in.
now.
You got or you want manual?
Yes.
You're a manual now.
Thank you. All right. Well, anyway. So you don't. All right,
You're almost there.
I'm gonna give up. All right.
Auto right now. There you go. All right, now try your look. Okay.
I'll just stay there.
Got a crane grab.
Good. All right. Okay. So it's a pretty significant colony that had fallen over there. Did she leave?
Good to move up?
I'm good to move on if you are.
Yeah.
All right, Cody, let's go.
All right, so that one right there, click that one, and now you're gonna start floating it. And then, so, just a
That's cool.
inches away, and you stay straight, and then you're gonna be sure it's back, so you wanna start pushing forward, just barely give yourself a little bump spot.
Mm-hmm.
And then little lateral movements to keep it straight.
Al in the driver's seat.
You guys are always moving.
You want to start rotating to your port. You'll rotate very slowly. Rotating to your port, so you want to get back fixing.
Yeah, so some crinoids on
Is
some
there
of
a
these.
Victigorgia up there? No, that's just a shadow.
Yeah. Serious. It's a nice shot right over there. It looks good. Primnoids here, and lots of crinoids on these primnoids. Those are facultative colonists, associates on these. Not really with high fidelity on any particular coral. They will get up off the seafloor into the current to feed. But if we ever see us zoom in on those crinoids, you'll see that their cirri, the things that they use to grab onto the corals, actually abrade the coral. And we're noticing that a lot on this particular cruise. I haven't seen that so much as I have here. And that abrasion causes a loss of tissue, and then that gives space for barnacles and hydroids and other things to colonize the coral and make life difficult for the coral. So crinoids, even though they really have no particular function other than to get up to the water column, they really have an impact on the community. So still lots of sediment and sand here. Just making sure that's an astroschema there. Something different.
What is that? That's a different.
He's
associate.
just spread out.
Oh, okay.
I think he's just so. Lots of sand, lots of ripples here. I don't think the RV is feeling much current though. I think it's coming at us, if anything.
Yeah, it's coming out.
Yeah. And our heading is northeast.
That's why we do that.
Beautiful sand ripples here. Really fantastic.
Bone?
I left mine. Couldn't find.
It's like that panicked look.
How it is? I know. Anyway, Great rocks, same corals we've seen for the last, I don't know, hundreds of meters now. Not seeing a real change in community composition.
Do you want me to take a picture of you?
What the heck was that?
What do you picture?
Yeah, what?
How do you
Made
put your
it-
camera? Sorry,
I
you're
think it's like,
newer than me.
I think it's like that.
There you go.
Yeah. Right here on the edge of a steep slope and wall, which we thought would be here. We're 1,173 meters. Good. But you know what? Just gonna say it again. Not seeing the Chrysler gorges, the metallic gorges. We saw them early on, but haven't seen them much since. Haven't seen much. So we have cup corals. We see anemones here. See the same community composition with primnoas and these plexarids. Saw a few iridogorgia and trichrysigorgias early on, but not much since. Really interesting to see this slope here too. So, there's swimming holothurian.
Do the same thing
It's beautiful, right?
No, because then.
And now suddenly we're sort of out of that.
Yeah.
There's a little one down below, but you know, it just shows you how patchy this distribution can be of corals. There's another one there, but the abundance, the spatial patchiness. The issue is we don't have a good temporal sense of how things are changing. You know, the growth rates of these corals, and if we see corals of different sizes, does that mean different generations, different colonization events, recruitments? So, anyway, we're still continuing down the track here. We've got about 10, 20, 30, 40, 50, 60 meters to the next waypoint. So this is the area we call the depression. And we're just sort of coming around on this steep slope to our right and looking back at the slope. Seeing lots of sand interstitial between these highly modified basaltic rocks and manganese coating on them. Exceptional flying right now. I'm gonna pan the camera up a little bit so we can see a little bit more head. There we go. See individual rocks. Look at that spatial variability. Right? Some rocks are bare, some of them were heavily colonized by these primnoids. You can see some of the rubble on the floor, primnoids that have fallen off the edge. I think they preferentially, you know, they prefer these kinds of edges and lean out. That's nice. Thank you. Nope, still seeing the same things. Didn't expect that. The last thing I expected at Titov was sort of a string of the same corals over a great distance. I thought we'd see much more diversity throughout the dive. So who knows what we'll come up to next, but
Primnoids seem to be very adaptable. Based
Primnoids
on,
do?
yeah, I mean, just
Yeah.
based on the ranges that we see them all the way to very deep to 400 meters the other day, right before the missile quota.
Yeah.
And they're quite resilient in the lab. I mean, they last pretty long when you keep them cold compared to something like a plexaurid.
Right, so you've been putting a lot of these polyps into respirometers,
Yeah, and also keeping them for 24 to 48 hours and having them still be alive. I mean, after that, it gets dicey, but as long as you keep them cold, that really seems to be the main thing.
Right. So here's the top of a rock here, again with primnoids that large sea star long-armed sea star I've been seeing. Plexaurid, you see the yellow corals there that have the middle stars inside in the middle with their arms stretched out.
That's a cucumber.
That's a sea cucumber, yeah. Awesome. That's great. I mean, not quite a monoculture, but it gives you that feeling. But they're quite abundant here. This species of PRIMNM really likes this area. So right now we're headed mostly north and northwest. And that current is sort of hitting us head on. Not strong. Nothing to have us deviate from anything.
Like the people in the chat are like, it'd be a good place to have a picnic if you didn't die and breathe and stuff.
Yeah.
So imagining walking around down there.
I've always wanted to hop out of a submarine down here and walk around the rocks.
Oh,
It'd be
yeah.
fantastic. Just fantastic. What the landscape would look like, the vistas you'd have, right?
Oh.
What we see. It'd be amazing. So pretty heavy sediment drape here. And again, we're still climbing a little bit though. It's interesting. We're 1164.
And we got 50 meters to go.
Yeah. And then it should bounce back up again. So yeah.
Of course, once I type back to lower abundance, we enter another port.
I know.
I'm
As
just
soon as you
gonna
say
stop.
something,
Yeah.
But, well, we're also coming out over the edge, right? Where they
Right.
beautiful ledge here. It's so great to see these corals. Love these things. Not see much in the way of sponges here. You wonder why that is, but there's a little buttress coming out here, a little bit of a bench with some sediment on it. Maybe we'll see some different species when we get around the corner here. Is that Nalipsami or is that still? Maybe there'll be a hard coral here. No.
Base.
Nice. Great view here.
Yeah, I mean, imagine. This is so cool. To rock climb something like that.
Yeah.
Sure.
It's nice to go out over the wall like this and look back. There's a little crab down there. It looks like it might be a homalid or a brachyuran. No, that's like a chasion.
A jason?
Chasion crab, yeah, sort of a red crab or golden crab. Shrimp here a little bit. Is that rubble or is that pavement? Oh, that's pavement, so, okay.
You wanna have a look down there?
Is
No, I'm
that,
fine.
I think those are Anthomastus. That's fine.
Yeah, don't have to, but it's fun. It's all good along our track, so perfect.
Yeah, we have seen some Anthomastus like scattered around, maybe every half an hour. There's another one on the ledge.
Yeah, look at that. You can see the polyps are well extended. It's amazing how you see Anthomastus, that red coral you're seeing now. It's a mushroom coral, but they seem to colonize in little bunches. Like they're. So there's a nice sponge there. It's a demo sponge there. Looks like.
Oh, really?
Yeah.
I missed it.
Yeah.
Oh, there it is. Yeah.
Living on ledges like this, but those Anthomastus seem to gregariously settle. That's what we call it. And they come in bunches at the same time. And they tend to colonize around each other. Snaphid eel below the ledge there. I don't know if you saw that fish.
No.
Bottom left corner.
Oh, yeah.
The little guy might come out, but
Nice.
yeah, it's nice big demo sponges here.
Yeah, they're beautiful.
Yeah. Depth is 1169. 807 is the time.
That was a cool feature. Is a cool feature.
Still seeing plexards and primnoids up here on the edge. Really amazing to see the fields of these corals just continue on and on and on. Some with crinoids like this one here. Some without. Nice little fracture there. This rock looks so like globules. It's like globs of this manganese crust that gets on them. Smoothed over like an old rock wall, you know, in New England.
Yeah.
Great shot. Showing you the top of the cap here.
Does it remind you of home?
Oh yeah, it does. That's what I was thinking when I saw that.
We're both from New England, so Massachusetts specifically.
Yeah.
At the moment, anyways.
So nice to go over the ledge and look back.
Wow, look at that
Turn here.
panning.
Nice. Yeah. Thank you. There's that. Looks like a Synaphia blanket eel there. See it?
Mm-hmm.
He's vigorously do something. Not sure what. You see these edges where like a buttress comes and this fracture here, and the corals seem to love that. I wonder if they really like this manganese coating, and they're able to. It's so pitted that their larvae can probably get in there.
Is it that blue?
So that might be that blue
again?
thing again. We'll go back
With an
and look at that.
anthomassus.
Come back and look at which one?
Cut
Blue one?
the blue one, yeah. Maybe a snap. Just for a second though. It's really great flying. That's fabulous. What a frame grab that is.
Sorry.
Good.
Yes, sir. Had fun?
Thank you.
Didn't crush it
Wow.
too hard.
No. Fabulous.
You did great. Yeah, buddy. I'll see you around.
That was really nice. This will just take a second. This will just take a second. And I can get everything.
Yeah.
All right.
Yeah.
Not getting so much.
Yeah, you want to?
No.
Yeah, please come on. This is awesome.
Go ahead.
Thank you.
Currently not in focus. Let me know if you want to go down.
Okay.
No, that's perfect.
So this
You
is
get
just,
a nice beauty shot of that.
is that in focus?
What
That's
is
a blue-green plexard. That's a really nice shot of the blue polyps and the green skeleton.
These remind me of like blueberry bushes, especially when they're retracted. When we were trying to get like snip one the other time. Let's see if it captures on the iPhone. It does.
Really amazing. Astroschematid. That's a brittle star, folks. That's an ophiuroid or an oaf. Likely astroschema. I'm going to try to see if I can go in further on the. That's all I got with the. Nice to document the polyps that have 8 tentacles. You can see the 8 tentacles. Makes it an octocoral. You see the blue tentacles, and then on the tentacles there are pinnules, even smaller, like hair-like projections, so just to catch particles to feed.
Look how they're all retracted by where the astroschema's arms are
Yeah.
on the left and on the
Yeah.
right.
Oh, good. You're right. So that's not helping the coral feed, although right here in the center they're open,
They're
right?
fine, yeah. It's interesting.
So I don't know. And so I always wonder if the tips of these arms, you see the arms moving, if they're actually cleaning off the coral or going into the
Yeah.
polyps and taking food out, you know, scraping across those pinnules and tentacles. I love the holdfast, how it's buried in between those rocks, right? It comes out.
Mhm. It's big.
It's fabulous. Right.
It's nice to have the other one next to it as a contrast too. You can really bring it out the blue. Let me get one
Yeah.
more beauty shot. Aurelio. Okay.
Yeah. We're good.
Yep.
All right. Thank you very much.
Ready?
Yeah.
Ready, Fred?
Take them all the way out. All the way out. Hey, Albert. Some nice flying, Cody.
Got quite a few cucumbers as of late too.
That one's on the edge. I might have a roll off here in a minute. The animal's trying to eat or taking organics out of the sediment. Can you take down the light a little bit? A little bit, please. You go snapping the whole thing?
He's kind of moving on the edge there.
How's that right there?
That's good. Another vase sponge over there.
Some slippery
Good?
Okay.
Yeah, you good? I'll just do a quick.
Never noticed its back like holdfasts.
Yeah,
I don't know if that's
you
back or
rarely-
front, to
No,
be
you
quite
rarely,
honest.
you rarely see these on the rock like that. And I
Oh,
agree,
that's its mouth,
it's
I think,
unusual
in the
to see
front,
that.
right?
Sort of those suction cups or something like that
Yeah.
on there to hold on to the rocks. So maybe it's a specialist
Maybe
for the
like
rocks.
the Disney where they put plungers
Yeah.
on their feet and walk up the wall.
Yeah. So many of these have polychaetes that live with them, and they're on the underside. If it were to roll over, we might see a polychaete living there.
Is that a parasite or?
I don't think so. It's more like a remora, I think. I think it just hangs out there and is protected. Oh no, here we go. Let's get a better zoom on that.
Looks like there's a lot of things living on him. Or is it like all the fuzzies?
Yeah.
There's some cool feet he has.
Those are some cool feet.
Got some cool
I was
shoes.
hoping to see it walk or move, but it's
Seen
just
a lot
hanging.
of shoes.
He's a sneakerhead.
A lot of shoes. Some Air Jordans. All right.
You ready, Freddie?
I'm ready, Freddie.
Eunice
Let's go.
is so cute.
So I think is
I agree.
waypoint 8 the. Is that the other side? Thank you.
Oh, I thought you were talking to me, sorry.
I am.
Oh. Is that the other side?
Yeah. I think waypoint 8 is the call to make.
So there's a London around these cool.
There's lots of girls, and they're all the same. I think
All
They're
the same, too.
They are.
The way they're growing.
I mean, you know.
It had to be like a
you're
massive
right.
recruitment event.
Yeah. I think they like this edge, but they were inside too, right? So they're all over the place.
I think Alexa said that earlier too, a recruitment event.
Yeah.
Remind me like a job fair. This Ron Keller job fair.
I never thought of a recruitment event like that. You go to the high school gym to get.
Star right there?
Where?
Little pink thing?
Left side. I think that's similar to the one we
Yeah,
saw
we did.
earlier.
We did. We got that one. I'm confident we got that one.
Got all the military guys after
Okay.
school.
Right. Right. It's true. Alright. Man, this is just. Don't know what these streaks are on the rock. Don't know what those are just cracks that have been filled in. Those are cracks that form when the lava formed, or when you know what I'm talking about. You see those
Yeah.
stripes. And I think that
I've always wondered that, but I
they're just filled in
never
with
asked. There's another one of those guys.
Then you have to use mouse.
The frowners.
the whalefish.
Is that what it's called? The whalefish?
Yeah, I think so.
Or a common name anyway.
They're awfully cute.
They really are.
Cody's gonna dial it in here. mouth is open.
It's just like,
It's on the attack.
maybe he took a chunk out of that guy earlier. And a nice round
Yeah,
mouth
could've-
for it.
yeah.
Although probably too small. Ooh, what's that in there? Oh no, it's tricks in my eyes.
Hey, look, Pernoet.
Oh. You mean
now
these
now you
stuff?
see the stripes, right?
Yeah.
I think those are scrapes filled in with sediment. The striations, when the pillow lava often has striations. I don't know if that's the case or not here. If that's a remnant of the eruption, but things look so masked and covered by the manganese crust that you think that might not be the case, but I don't know. Trying to find some explanation for it. All right. About 200, 150 meters, 200 meters to the top of the other side.
What do we got?
Crinoid,
Oh yeah, thank you.
monster crinoid. Something else floating in the water.
It's a sea humor.
It's a holothurian.
Is that an anthomastus next to the crinoid?
Yeah. No. Yes. Yes. Wow, that took a minute to get. Look at this. Massive.
It's a big one, yeah.
Thank you. Massive crinoid. That's anthomastus.
There's a crab in that, or see the lobster?
Cody wants to check this out.
You
see
get a
little
beauty
animals.
shot. Hold on.
You can see animals living in here.
Beauty.
Sunlight on it. This is a stalked crinoid.
Do you call it a
I
sunflower?
think it's
sometimes?
Pro-isocrinus.
Some what?
Is that it's
What's that?
What, Jocelyn?
Nothing.
Nothing. I'm gonna zoom in when we're ready.
Go ahead and zoom in. Hold this right here.
As a group, these things are super old in the fossil record. 500 million years ago. Cambrian. You see these little white dots and things. These could be worms. There's certain parasites that they're in their own group and they live on these crinoids.
Are they mesotones? That's what somebody said on YouTube.
How'd you know that?
Somebody said on YouTube.
They did? What'd they say?
It looks like the crinoid has mesotomes.
Yeah, that's right. That's what they are. Kevin.
Thanks, Kevin. Appreciate that.
That's what they are.
But that photo bomber.
Yeah.
Cool concept.
It's cool. It's a cool thing. I can pull it back a little bit. Awesome. There you go. Plexard. PRIMNM. Plexard.
Stop.
PRIMNM. No. Well,
I mean, you're not wrong.
no. But you know.
It's funny
We're
how
here, we're
spoiled
on
we
it.
get. Like, we're like, oh, that's boring now, even though this would be super exciting on some of our other times where we like see nothing, right? So
But this is cool running the ridge, right? And this ridge is here because of the way the seamount was formed, right? And once upon a time, lava was flowing through a conduit right here, away from the seamount of the volcano. And these pits we're seeing are just places that have been weathered away over time, worn down. Really not seeing much else. No hard corals. No Crisic origins. Amazing. Maybe. Oh, 8, 22, 1165. You can see the patina on that manganese crust.
No, earlier in the cruise, people were very interested in what that could be, and we remember. I don't know if you hear, we like touched it, and it's just a patina, like you said. Another
That's a cute
anthomastus and an urchin.
That's a cute thing.
Some more
Driving down now along the wall to see if we see something different, but not same old.
Same old stuff, huh?
Same old. Stalks with
Supposed to come around and be
crinoids.
something huge.
No. Something sticking way out, you know,
Yeah.
big thing. Nope.
No.
Nothing. But no one. More demo sponges, that's for sure. That's nothing new there. Shrimp. Corals up there.
That one's already started.
Gonna jump back on top.
I guess so.
Look how that's
Yes.
under that rock. Do you see that in there?
Yeah.
That's kind of crazy. I wonder if
Oh,
it fell
right there.
recently.
It's PRIMNM though, right? Same coral?
Yeah. Yeah.
Tucked in.
See? Look at that. That's amazing.
Some champignons.
It's more of those sponges too, right? That's encrusting sponges on the rock there.
And you've got that big xenophyophore. Flying cucumber.
Yeah. Man. Alright, I'm ready. Ready you are.
What are you ready for?
Something different.
Something.
Whatever you got. You got
Alright, close your eyes
Got it?
real quick.
Okay.
We'll tell you when to open them.
I'm ready
Let's
though.
make some tracks.
We're doing alright, wait a minute.
That might be a blue boy. I can't tell.
What, is that something different in there? No. Same, no, it's the same.
I want it to be.
I was wishing for something. No.
Something different.
Hey, look. You want some of these?
If you want the orange and the yellow ones.
Oh my god, the color makes a difference.
It does
Oh
make a
my
difference.
You just put them all in your mouth at one time. They all taste the same.
They do not. They're all
You
flavored
couldn't tell the
slightly
difference if you
different.
closed your eyes.
I
Taste
could.
it.
Try me.
No way.
It's like,
Wow, well, so many corals. There's so much, right? So much life here. There's brittle stars and crinoids and all these things, but it's the same. Is that a purple thing over there?
oh, you give me squished ones, Cody?
In his back pocket.
Oh. Gross.
Melted too.
Alright, put one in my hand.
Oh my god, what happened to them?
Yeah, exactly.
Alright.
That's something different.
I'm looking at it. I see it too, but it's
Is
just,
it yellow?
It's just purple.
It's
You want to look at that?
purple?
Let's look at that. Let's look at that
Oh.
purple thing. I think it's a purple plexar. But does it? Yeah.
Oh, yours. Yeah.
All right. Could be a
Or is it a...
bit of bubble gum.
That's what I was gonna say. It looks more delicate than a
It's
corallium.
it's waving like a paragorgid.
Yeah, exactly.
Yeah. Well, that's different. Let's write that down. got it?
Yeah. I mean, beauty shot it. Hold on.
With the ophicanthid. That's pretty cool. I can go in further. Hold on a second.
And a crinoid. So crinoid and.
Going in.
You have manual focus.
I do. Excellent. Alright, that's the crinoid. There's the polyps. Alright, fine.
Is that a shell in there? Or is that the rock on the other side?
Rock on the other side.
Gotcha.
Or something, a white holdfast or something. Okay.
Yeah.
Super.
You asked for something different
I
and
did, and
something
he
different
delivered
popped
it.
up.
I delivered it.
Really weird how you're starting to do this.
It's weird. It's strange, isn't it?
Oh my
I wish I
God.
had $1 million.
There are limits.
There are limits, right? All right. Crinoid. Oh, something different here. Let's see. Just a crinoid, same primnoid, same sponges, same plexaurids.
Emerges most ones.
Wow.
RV Bridge, I see that new waypoint.
Roger that. Thank you, Bridget.
Nope. A little more sponges here. If that's
Yeah.
anything. A lot of holdfasts where corals used to be. That's clear. What color did you guess?
I guessed yellow.
It was orange.
Well, those are kind of citrus, to be honest with
They're
you.
the same.
But-
Those are the
okay.
same.
The red and purple is different. And so are the green.
Man.
But I see your point. Point taken.
Where are the squat lobsters?
Found that wrong.
Where's all the humanids?
There's a sea
Star.
star.
You've seen that sea star before.
Mhm.
Haven't we seen that star before?
Double
though?
stars.
Double stars. I know. No, I know. You just.
Tim is ready to get out of this area.
Oh, yeah, I just am. I mean, you know, I'm so thankful to be down here though and doing this. It's awesome.
Yeah.
You know.
You can get some targets
But
though
it's,
that
you
we're
know,
trying to hit.
It's
you
all
know?
information. I mean, it's all good information. I mean, now we now know what lives on the edge of this. The plexaur again.
Purple.
That's
Purple
it.
stuff.
Oh.
Georgia,
Purple
probably.
stuff on dead stuff.
Let's go take a look at that.
Do it.
Let's do it. I bet it's going to be an orange oaf too, and not the white one. I really would like the white one. Here's that same sort of analpsamia. Oh, it's growing on an old coral.
Is that the same thing?
It's really thick looking too, but it's probably analpsamia.
Yeah, it's a lot. Yeah.
Right.
I agree.
Slippery
It's
here.
very thick.
Oh, yeah. This one is super slippery.
Super slippery. All right. Well, I better get on it then.
Hustle.
Dun, 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 dun.
Tough angle.
See, it's even the orange morph. Coming in. Coming in. And I'm in manual? I'm in manual. Okay.
Good job. Nice beauty shot of that.
You know what? I'd like to collect this.
Oh. Would you like me to collect the dead thing?
Not in particular, but I really would like one of the oafs in a branch. So I don't know if that's here.
Okay, so not the
Well, it's the same. It'll end up being part of the oaf. Bless you.
Excuse me, I'm sorry.
Oh man. Oh God.
Well, we're all healthy, so.
My glasses are all messed. No. Let's see how big this is.
Do I have a hole in that shirt?
We can't take the whole thing. I want to be clear
You need
about that.
lasers?
Yes, please.
I bet you could get that left branch.
Lasers are all. There's one. There's two. Not very big. Oh. You
Always
never
such-
know what that door means.
I know.
Something good or something bad. All right. So I think this is all one brittle star, one oaf in here. I don't want to collect the whole thing because that's not a good thing to do. So I don't know if down here's better, that half, or like up here maybe. You know, and yeah, and get part of that. Might be the thing to do. It's A block at the base too. Did you see that? Apicopher. $11.53.
Is an apicopter a worm?
No, it's a mollusk.
Oh, then no, I didn't see it.
There you go. There you go.
Just the one branch, or?
Yeah, that's right where you are. Is nice, really nice. Yeah,
Watch out.
that'll be all right.
You able to get in there?
I'm gonna need that for
Right, and we're gonna press sample and see juice. Quite
yeah,
yet.
right when it's over the box, right? That,
Right back forward.
It's 11:15.
did you right?
It's
Sorry.
384.
Did you finish it? Before you close it, you're right. Can you get one more?
One more piece.
Just another piece.
The dope on it?
I don't want. No. I think that top,
What part do you want?
maybe where you were, but just one over,
Okay, so
might be the easiest to come in on.
you want the fork or just one more branch?
Either one. The fork's fine.
I mean, you want one, I can do one.
No, I think that would be brilliant.
The fork, okay.
The fork.
Like just above the oak blade?
Yep, exactly.
Would really like a high-powered microscope to know if that's fluorescent. I know we did those tests in 2017, but I just don't understand the reason for color down here where there's no light. Could be a defensive thing too, like a certain chemical that just happens to be purple. Beautiful.
Beautiful.
Alright.
Nice job. Thank you.
That's a good collection.
It's excellent.
It's busy. It's busy. I'm going to bed.
What? No one's allowed to sleep. What are you doing?
Good night.
Good night.
About to get really exciting.
Is
I
it?
know.
What do you
We're going up to the
What did you
top.
find, Cody?
Rocks.
Oh, goody.
All right, we're ready. Oh, is that me?
What are we thinking for off bottom, Tim?
I think we're thinking about. We got to get to waypoint 8.
What are we at
So
now?
we're 200,
Oh, we're
50
not very
meters
far
away.
from waypoint 8.
right? So I'm thinking
Sign
if we don't
Falkor.
see anything else, look at that. If we don't see anything else here, we're going to come up. So I'm thinking, you know,
Might
could
be
be
earlier
11.
than-
I don't know what,
Oh,
Yeah, I mean,
you know.
it's 1200. Well, we're gonna drop down off the edge there, not too far. But let's call that 15.
I mean, I'm thinking we'll be at. Yeah.
Yeah, it's gonna be like call it an hour from off the bottom
It's worth
to
a nap.
the first float.
Yes.
Because of the turnaround
Yeah.
after this.
So-
We have to wait.
It'll take us 15 minutes from first float to on deck, so you're talking an hour and 15 just
What's
from
off
just
bottom?
from
Yeah, okay.
Yeah.
Okay, good night.
Good night.
Good night.
I wish I could take some of my energy and my sleep that I took today and just give it to you like a
I
video
got it.
game
I
or
got
something.
it.
I know you're
all,
doing
all
it
those holdfasts.
really
on there?
great.
Holy monkey. That's a popular rock. Look at that.
Oh, we got another star.
Those are all holdfast.
Is
2,
that a
4,
ho-
6,
oh, that's
8,
another
10,
one.
12. I mean, come on. It's over 30 holdfasts there.
Say Holdfast City.
Down below, you think? Yeah. That's tremendous.
This rock was
That was a hop back in the day.
spot. That was the
That
spot.
was a spot. Look at that.
I wonder why they all didn't make it. It's so weird.
Yeah, right. It didn't sort of recolonize over time, but maybe it was a rough place to be. Cody wants to see what's down below. I'll help you out a little bit. Look at all of them still on the side. All right.
Plexorids
Onward.
and primnoids.
About to take a route here in a bit.
Yeah.
We'll see something interesting.
Gonna take this little right turn right here.
I hope so.
You ready? Almost there.
Hard ride. Hard ride.
You're after
About
the
10,
route.
10, 20 more meters.
Crap.
Things are about to change.
Oh, crap.
So what? You see a crab? What?
Oh.
Whoa. Something different. That's great. You ready? What'd you say? He just said suction. So we are inside the U.S. National Monument right now. And we don't collect anything unless we've seen at least 10 of them. Yeah. This is a nice example of a Eumenitor crab. A deep sea crab.
You said Eumidia?
Eumenitor.
Eumidia.
Yeah, they can have 3 to 5 ranges.
Oh, I see.
I'll just get a
Eumidia.
square up beauty shot here. You ready? You ready? Yeah, very cool. Oh, you would move.
Moving crabs has been
Okay,
also a theme.
I'm
Not
going
as
in.
fast as the last one, but
Ba-ba-ba-ba-ba-ba-ba. Damn it. Excuse me. Da. That's fantastic.
you just
See, the abdomen is
shut.
underneath.
There's one to the right as well. The little white guy to the right.
Let's see him. Go in there next. Da. See you later.
Now I can't unsee the squatting that you told me about.
Yeah, they have the abdomen tucked underneath. That's why they're called squat lobsters.
Have you heard about those parasites that make them think that they're
Yeah.
the male
Yeah,
ones are pregnant
you're right.
and they take care of them?
Absolutely.
That is so crazy.
Look at this one. This one has the rostrum is much longer.
Find that paper.
Arms are much thicker. Claws are much thicker. That's tiny. That guy is about the size of a quarter, I think.
Is that the same type of crab? Can't be.
Similar type, yeah.
Okay.
Similar group, yeah, but totally different species. Ingenious. All right. Good catch. Yeah, I'm ready.
Go, but take
Let's
the right.
see some
that
more
was
new stuff.
exciting.
Whew, I'm all like
Oh,
worn out here. That was awesome. Hands
Get
all-
some like clammy
hands all-
palms.
Hands all clamped up from being on the focus.
There's something new right there.
What is that?
No coral.
I don't know what that is. No, it's the same. Tinophore, that's Tinophore. Yeah, that's fine.
What is? Yeah, that's interesting.
Comb jelly.
Cool. It's always a
Talk
treat.
about bioluminescence, these guys light up. You know, see them.
You can see it,
yeah.
yeah.
Start throughout the water column. Cody. There you go. You know, I don't know why this was colored the way it is either, right? This has, you know, sometimes they're absolutely transparent. I don't normally see that
Sometimes
color.
they have
I don't
this
recognize that one.
other color.
So I'm not saying anything. We're going in on this one too. Could this be something new? No, it's a PRIMNM. It's the same.
Was there an associate in
It's
it?
the same. It's the same. You can tell it's a PRIMNM with the same branching.
From my right-hand turn.
Oh yeah,
All right.
it's the ashy schema.
And then you just take the off ramp.
Slide down the rocks.
Good. No, I think you're sliding.
And slide.
Slip, slide.
Everybody
it's got polyps.
tap your feet.
Oh man.
Or clap your hands, maybe that's what it was.
So
I
this
think
is,
I got that wrong.
It's only 9:44, guys.
I know. I was just thinking the same thing. It's not 3 a.m. or anything. But you can
Well, it
see
is
the
for
polyps
you.
come halfway down the
Oh,
axis. Right.
An astroschema or an anemone at the bottom
This
of
whole
the
thing.
base.
Oh, I was just gonna let me get this first while I got
Yeah,
some
yeah.
focus going. What? Hello?
Slide.
We're off.
Slide.
All right. It's still the same one. Yeah, you're still there.
It's still here.
All right. Oh, you're sliding.
Stay slide.
It's coming right through.
Stay.
Oh, here it comes.
Ooh.
Splash. We're in the splash zone.
Where'd it go?
I don't know.
Underneath. Look under it. Where's your?
Or on
You're
us.
good to go now.
It's gonna settle
that,
in the
thank you.
ROV.
You think you're stable?
Maybe. Let's rock those other planes.
I'll be quick about it. No, that's look the anthemastous polyps down below. Looks
Oh,
like.
word, okay.
Am I a
Getting better
manual?
at
Somebody
this.
You
come.
right, man. Yes.
But then again, there's only 3 like families so far.
There we go. Kind of pinkish polyps, though, but they're turned down, so they're primnoid. Right? You see that?
Yeah, quite a few polyps per whirl in this guy, looks like.
Let's get that schema in there. And then we'll drop down, take a quick look. Anthemastous colony. See that? Three polyps. Fabulous. Old stalk there. Old hold fast to me. Cool. So that's really pink in color though, isn't it? It's a different color.
Yeah.
Kind
I
of
agree.
morph, but
Ready?
ready.
Or we need a white balance, one of the two.
Okay. Right hand turn. We're on it. You're right. We're at waypoint 7-ish.
Jumping over.
11:54.
Look at that rock. I mean, I know it's just pronounced.
Oh my gosh. They like the edges, right? They like to be out in the current. Look at all the holdfasts too that are down and gone.
Lots of crinoids.
So we're at waypoint 7.
There's
Wow, nice rock.
The demo sponges again.
I don't see anything. There's a great pronoids, but I don't see anything else. Look at all the holdfast. There's another Humidor crab.
Oh yeah.
Thank you. Crinoids. Just hold that. Couple of plexaurids.
Yeah.
I don't see any predators. I don't think any. There's a basket star there.
Oh, is that what that is?
Yeah.
Cool. We saw one of those earlier.
Another
right?
crab up there. That's nice.
And the rubber really doesn't have to go that far, it's right below it.
What's right below you? Oh yeah.
All the rubber from
All the rubble. It's quite the rock. Reminds me of the wall we found.
a little miniature size.
Yeah. Demo sponges here. Yeah.
Another option. That's
Yep.
4. 5.
Yeah.
Is that a chrysogorgia?
There's a weird fish.
Yes, it is. Nah.
That's what I'm
That means that is too. Yeah, there's chrysos here.
Okay.
I saw. Yeah, let's can we look at that one, Cody?
Yep.
Thank you.
Oh yeah, look at it. That's huge.
Well, I see that red.
Gonna
face.
associate to the
There's
one on
up
the
there
right.
too. Yeah. Okay. Yeah, we can come down.
They everywhere.
Can we take that iris down a little bit?
There you go, sir.
Ready?
And you're in auto manual focus, sir. Manual.
I'm in auto manual.
Auto manual.
Sweet.
It's both.
Looks
Did
like
that
there's
do
a
anything?
little associate in there too.
In here? I see a
In the
red
top
thing.
one.
Yeah. So that's a shrimp up there. That's Bathypulmonella. It's got a giant elbow. There's another claw here. That's a Humidor, right? That's like the crab we saw just a minute ago. Yeah, you can just see that part of that big elbow, that
That's
big
what I
forearm.
saw.
It's got a massive forearm on it, right? It's like it's a Popeye thing.
Are those little white spots, are they what you thought they were?
These are barnacles here.
Yeah.
See, these are barnacles, but that's a holdfast.
So some of the little
That's a
white
whole- that's a
could be
whole
the
fest.
beginning of a whole fest?
No, we really see the coral with it.
I've
So
never
the-
seen these.
Yeah, that's the thing. We rarely see anything small like that. I thought there was a guy in here. We missing?
top.
Oh yeah,
Top
good
right.
call. Good catch. Whoa, Tim, what are we doing here? Let's try that again. Come on, let's go. Let's go. I'm all the way in. That is a red-faced crab, though. Okay. So there's chrysos here.
Another crab
Another
up
crab
there.
over there.
Crabby rock.
Yeah.
They're everywhere.
That's a chirostallid for sure. It's an associate for a coral, but it's hanging out there. All right. What else we got? Gonna do this. This is what you do
I
when
wonder how big they can get before they do fall. I mean, this one's huge, right? I mean, so it appears.
Yeah, oops, sorry.
No, you're fine. Look at him.
Yeah.
Is that
Yeah, I don't know
humidita?
I don't know who that is. No,
No?
that's a Kyrostalid, I think. That's a tough call right there. Tough call in the crab world.
He's like, bring it on.
You all see him? The way you really tell them apart is you need to have the crab
The
and
rostrum?
look. Well, no, that's just no. It's the underside plate. There's a notch in the center of the plate. There's other diagnostics too, but that's a big one for that group. So you really have to have it in hand. All right. So we've got some chrysos here. We've got primnoids here. Do we see Anthomastus here?
I have not
Should we
yet.
pop a Niskin here?
Yeah, let's do it.
You want to?
Good call.
You want me to come right above it?
Let's pop a Niskin.
Or you want to do it right here? Or you
I don't
gonna
think
put-
it matters.
It doesn't matter. It's
Let's
easier
do
to
it.
go
Let's
up.
do it. You want to go up?
No, whatever's easiest
Yeah,
for
that's
the
what it's-
ROV pilots.
I agree.
It's gonna be 85.
Oh, speaking of crab, there's number 10. Found 10 of them.
Oh God, freaking me out. Hold on, I didn't know it was there. God darn it.
Alright, Niskin number 2.
Is that your cup, Tim?
Mine?
Yeah.
No.
Number 2's fire.
Great.
Number 2?
That's at 1157 meters.
Yeah. What number did you say that was?
385.
Rajo. And Niskin number 2?
Mm-hmm.
Awesome. Okay. All right.
Move on.
Move on. What else we got here? Demo sponges. All right. We have to go out over the edge.
Don't do it. Don't do it.
I'm on the edge.
Man.
That was neat. I like that rock quite a bit.
So we're about 9 meters off. 8 meters off. Pretty cool. I haven't checked this. This is Randy's doing something.
I haven't gotten anything.
Nobody's doing anything.
Oh, it's a stock trianoid. Not,
Yeah.
Thought for a second.
I did too. Every time I see those.
So we're going to follow this down and around,
Okay.
right?
Yep.
Go to number 8.
That's fine. I think it is a little bit of a dip and then a slope up.
There's another eel or fish or something down the bottom right. You can see it kind of.
Wow, I didn't see it. Man. Primnoids. Crinoids all over them.
What kind of sponge is that?
Sponges. More demo sponges.
There she is.
I'm watching
No.
this thing.
Okay.
10.
It's getting pretty interesting, but it's also more of the same.
It is absolutely more of the same.
But it's beautiful. Hey, I watched you find a new crab.
Yeah?
Mm-hmm.
You had one eye open?
Yeah.
Whole time.
Supposed to have them both
Both open?
shut.
Both shut.
Another crab on the
I
primnoid
couldn't
over
help it.
there.
I thought for sure when you asked for $1 million, it was just going to show up on the seafloor.
You didn't hear that?
Just kidding.
Oh yeah. There's a cute
Yeah.
little crab.
Well, so you see where we are, right? We're at the waypoint. So just coming down and turn around. There's a lot of coral here.
It's It's interesting.
It's a
It's
lot
got
of
like this
biomass.
pitted landscape, right? And I just wonder if that makes it easier for these corals to
Yeah.
settle. We keep seeing that again and again and again. I think we might have really underestimated how important that little bit of texture. Maybe I underestimated it.
So important. Yes. And mostly in most basalt systems, that's really important, like hydrothermal vents and whatnot.
Thank you.
That's kind of gross.
Pronoid. Lots of holdfasts.
Noah Ridge.
I
It
think
is
so.
Primnoid Ridge. It's a good word
There's
for
some
it.
plexards there, but
Still
man,
have Team Texas in here, hey?
Team
Team Texas! Woo!
All really walk.
it's really hard to believe. Oh, these are the.
It just looks like there's so much more on the I mean, it looks like this has been a productive area for a really long time.
Very.
There's so many bases.
Yeah, it's ridiculous.
But then we have that one rock with like a million bases
saw
that
that. It was
nothing
like
was
the
growing
Podular
on
rock.
I
again.
mean, look at this. Goes all the way around. See over there? The whole
Yeah.
wall over there.
Yeah, whoa.
It's nuts.
What?
Is that sediment or is that
No,
sediment?
yeah, that's cinnamon on the far side, but look, you're gonna have
I mean,
another
it's-
Primnoid wall. These underhangs.
I know.
You know, I think we always think about everything sort of reaching out into the current to grab food, but the underhangs are productive too.
Yeah.
We've
Ryan
seen
and I were
some
talking
in
about
like
that
water.
under
the other
like
day. What?
under rocks, like almost in a cave.
Yeah.
You saw that one? That was
I don't know if
kinda-
it's because like there's a little sort of
Yeah, like it
microflow that,
accumulates
like a
there or
little,
something.
you know, a little boundary layer that helps everything move.
It's like, or that rock fell and that coral will eventually die. It's hard.
I don't know. But it doesn't feel like overhang is the worst place to be. We're moving down, right? Over the is that
Yeah.
what's happening? Okay.
We're moving down
Yeah.
to come over, I guess. No, there's the
Yeah,
bottom.
because we go to 117 to 1200 and then back up.
See, did you guys figure out these random piles of rubble?
Well, they're random piles of rubble.
Look,
And
there's
it's
another
pretty
one.
thick.
Yeah.
Yeah, I know, and
They're
there's another
totally,
one coming up.
they're
See?
totally encrusted.
Yeah.
The
They've
one
just
that
fallen
we saw.
down, right? They're
No,
fine.
I don't know. I wonder if it was something that used to live there and just died in place because you don't see anything above it, right?
Oh, there's that big overhang.
I don't know. I can see some of this stuff falling down and just becoming.
Maybe. It's another one.
Yeah,
It's not quite like the Rubble River from last
No.
time.
Did you not see the party above this?
Yeah, there's a big.
Did you see the party?
There's a party.
They had a good life up there.
Same
bro.
thing.
ROV
Oh,
bridge.
Dorian.
I see that new
Plexarid. All right, 9 o'clock. We're at 1,200 meters.
It's really a beautiful landscape. We're approaching waypoint 8.
Traverse.
I think we'll probably go to waypoint 8 and then pull in this plan. What do you think? Does that sound good to you, Tim?
Yeah.
All right, so that's the plan. Get to waypoint 8.
Sounds good.
Cool enough that I feel like we should.
Just
So, you know,
finish it,
when you
yeah.
look at the map, this clearly looks like it's like a cutout, right? And then it just sort of fell. And you see all these rocks here. But then again, you see, I don't know, it's just really hard to tell about timing. Just trying to think about.
I don't think we've seen any more Walteria though.
Yeah, you're right. More dead rubble.
That's huge.
More Primnoid.
Well, I don't know. There's a
what are you
but there's a
thinking?
bunch of sponges. Like there's another something.
Yeah,
down,
I
you're
know, I
headed
know.
west, buddy. What do
I'm
you
wondering
get?
if we should just like collect a different kind.
You're headed west,
No.
right? So
Yeah.
you can
We
just
got
turn.
plenty to do work
back
with.
it
Then
down.
we're
Yeah.
back in the water
Back
like
in
4
the
hours.
Back
after we
in
get
the-
out. So.
Call on that.
Yeah, who planned that?
Soon
I'm just
below me.
kidding. I'm teasing.
You were all on board with the
fun
plan.
driving back?
I do not want anything but that.
I'm kidding. It's like that was mean. I'm sorry.
It's okay. What are we here to do but dive, dive, dive?
Plexar,
We're going to spend the next 4 years
Primnoid.
analyzing data.
I know, Bert said to
Primnoid,
me, haven't you
Plexar.
guys been so excited for this trip? It's like, yeah, we've been excited for a very long time to be here.
It's an amazing, amazing gift to be here, which is my cue while we are on air to thank all of the incredible people who make this possible. All 39 people on ship. Without whom this literally would be impossible. We actually are so lucky to have our captain in the room with us.
So honored to be in his presence.
So really, thank you, Al and Captain Al, and all of our ROV team, our MTs, everybody on the ship makes it possible. In addition to a huge shore team, we are in the Pacific Remote Islands Marine National Monument, Howland and Baker Unit. We are on Titov seamount, and we're very grateful to the U.S. Fish and Wildlife Service for giving us a permit to work here. This is a really special part of U.S. territorial waters, and we're really glad to be in the monument. We are also here thanks to the help of NOAA's Deep Sea Coral Research and Technology Program, Conservation International, of course the Schmidt Ocean Institute, Boston University, Woods Hole, and Harvard. I mean, just a lot of people all over the place to make this happen. And this is a 3 or 4 year process in the making in order to get, be able to get this expedition here, which is really the middle of nowhere, but also this is where it's at. If you want to look at some really interesting equatorial seamounts, which we do. So we crossed the equator yesterday on solstice, which was so exciting. Halfway through the cruise, halfway through the year, the exact half point. It was fun.
It's gonna be a good omen.
It is a good omen, and that is another sponge. And we are here exploring equatorial corals in the Phoenix Islands Archipelago, basically mirroring what we did in 2017 below the equator in the Phoenix Islands protected area in the Republic of Kiribati, which we've been working with for 15 years. And now working north of the equator in the U.S. EEZ, the U.S. part of the Phoenix Islands, and in areas beyond national jurisdiction, international waters. And it's such a treat to be working in collaboration with Kiribati for so many years, but also now to be working in my own country, our own country, and kind of trying to put the whole archipelago together. It's a 10-island archipelago, and it's really such a treat to be able to explore it all. As we go down PRIMNM Ridge, as Cody likes to call it. So we are headed towards waypoint 8, and for those of you who are trying to, who are wondering what our plan is to get to waypoint 8, which I know doesn't mean anything to you, but it's our next and last waypoint. It'll be coming up probably in the next half an hour to an hour. Then we will leave the seafloor, do a little bit more mapping on Titov seamount, and then go right back in the water 4 or 5-ish hours from now. I don't know how long that'll quite long it'll take by the time we come up, and then we map and then go back down. So
Just an hour and 15 to an hour and a half.
yeah.
To have it on deck.
Ooh, hello.
Aurelia?
Plus then,
3,
Oh, wow. The name
6,
Aurelia
about
comes
6.
to mind for this.
6
Is
or
that a
7
type of
hours.
jelly? Or is it something totally different?
I think it's a tumbling sea cucumber, no? No, what is that?
It's a
jelly.
medusa. It's
Yeah,
a
you're
jelly,
right.
but it's a
Huh.
We're super grateful
So-
to our marine technician in the room, Marine Tech James, who is doing an awesome job of managing this live stream and of helping to capture all of this data so we can make it publicly available, which we're doing right now on the YouTube channel, but also archiving all kinds of associated metadata with various public resources as part of the Schmidt Ocean Institute's mission to make data, high quality data, publicly accessible to help democratize the oceans. They phrase it differently, but that's my paraphrase.
I think that was pretty good.
So we're coming down deeper. We've come down 50 meters.
And then we started making our way.
Oh, look at that ski slope. Let's go skiing.
We'll figure it out.
Yeah.
3 hours and that.
We're only about 4 or 5 miles away from the actual mapping. I mean, I don't know. It's going to take a lot of time to do
You came down about 100 meters.
Still coming down.
You're turning to the north though, right? You're going run along the side of it.
Seeing how far this goes, because at first it said 30 meters. It's a little bowler.
Right.
But
It's just gonna keep going.
it was.
And your altitude is 3, but
I'll fly.
it's gonna be slow.
Do is fly right from here. You can't see it.
No. It's supposed to come back up though. You can see that we're in that little. If you see the dark patch down in the center of the map.
I agree. Typically, sea spiders are all eggs.
Are you answering your social media?
I just saw she said, or they said, sea spiders make me go wild. I don't think we haven't seen any of this.
What's that?
Sea spiders.
On this dive?
No, the last time
no, we
was
did.
great.
No,
You
we
did?
saw one.
Okay.
It was on the rock. We tried to collect it. The leg and it. Current was so strong it took it away. I think that was this dive.
Yeah, the little pink fish, are like commonly called whale fish.
Okay, I thought we had the dice here.
They look like they're frowning.
Somewhere specific then, just in case of running across it.
Floating cucumber. Snailfish. Interesting.
Not much down here. Oh,
Yeah. There's
not a
some
lot down here except
Primnoas.
this big Primnoan rock. And that's what it is when they come with sponges. Still, after what we saw at top, this is not all that impressive.
You're right.
Nice glass sponge.
Pretty far. A little bit on.
1274. Got another 20 meters.
Another Plexaurid.
20 meters in 2 minutes. 3 minutes.
It looks pretty cool. The pattern of the legs, just that angle.
Looks like plastic
I know.
every time.
Luckily, it's not.
I'm not sure what the expected waypoint 8. It's just a pile of rocks.
It's nice looking. Is that a Venus flytrap?
It is a Venus flytrap. Acting a Skyfa.
Oh, you got an Iridogorgia.
A little one. That's what that is. I'm not sure that's what that is. The axis isn't really turning. But yeah, it actually, I think it might be.
Take a look at
It's
it.
good. No, it's okay. That's okay. It's just a little one.
So
1287. Man.
Is that enough time for you guys to get samples off?
well, it kind of goes back down. samples do we have from this?
So we
Anna,
should
can I see
be
the
coming up
basket sheet?
slope shore,
We just
right?
ended on
Or what?
385 for that last Niskin, you know. It's
What?
not super
It's a pretty light
populated.
basket, we'll be fine. Yeah, it's good. It's really good. Okay, I'm good with this plan.
Great.
So this will make us
Another option.
It is wild. I know we're in completely different currents and stuff now, but that you can have so much abundance next to these sandy slopes with a coral here or there maybe.
We're actually proceeding down slope to come up to another high, I think, at some level.
Okay.
This high at waypoint 8 is supposed to be,
1300?
thank you, 1186.
And then we go back down to 1300?
I don't think that's a suggested
Okay.
one. If it's looking good, we need another thing. But
We can do that if you want.
right,
Okay.
we can do that. So we got to come up 100 meters
But we can.
in the next 100 meters.
Yeah, they've got my sonar closed. It's slowly coming up.
Yep.
Does that work for that timing for the edge point?
Well,
Or
yeah.
box time?
Well, everybody between 12:30 anywhere in that middle.
It's all just slope draped with sediment. Well, the rocks are pretty clean, so
Yeah, one PRIMNM. Yellow sponge.
That's good. What about on deck?
squid, plexar. Another plexar, a bit of a whip. Might have hit stickopathy. I didn't see that one.
Yeah. Swords.
We thought this would be a desert.
It's a desert.
We thought this would be like a desert, yeah.
You gonna name that desert?
You could name it. Call it whatever you want. Welcome to the desert. 1288 still.
Bikini Bottom.
It's a slope though.
But it is, sir.
That's a slope. You don't drop anything. It's just some rocks down there. It's just some rocks. No sea pins.
Yeah, interesting. No
No
peduncles.
real tracks or trails. Oftentimes we'll see gastropods, snail trails, or oh, that's all right. I mean, there's something coming up. No. No sea cucumber trails.
Take a look at this little fish. Little ship moves.
Yeah, it's fun. Another one up there too.
Yeah.
Got like a tripod fish without the tripod.
Tripod desert.
Just sitting on fins. Alright, you got it.
It's got quite the pair of antennae.
Just like a tripod fish without the long rays, extended ray fins.
Hey, I see that our line ends. Are we going to continue at the line?
Well, we'll finish up the line, so.
What is
yeah,
that called
just
again,
probably
Tim?
to the
Where?
waypoint
The
I
thing
gave
in the
you,
water?
probably your last one.
Siphonophore? I don't know if that's what that is.
Or is that just not? I
Well,
No
can't tell.
worries.
well,
Take
but-
all the time you need. I was just wondering.
Look like
how
a
far-
larvacean
how far
house.
back-
That's a pretty.
Yeah, I think
Nice
it's
slope there.
It's a very decent slope.
Are you ready?
Yeah,
Can
we
you
got
do
100
it?
more meters to go, so.
That sounds good. Thanks, J-Road.
What is her watch? When is Bell's launch?
Not too sure.
Can't tell. Is it 4 hours long?
They do 8. Yeah,
8 on?
They work 8.
Okay.
Standby.
Data seapan down there,
That's
essentially?
good.
Nope.
You can just snap on the C pin right here, Cody, or you moving?
I will go set up right in the light for it.
Yeah.
Yep. That's what that is.
Alright.
Yeah, we've seen this guy already. We played with this earlier today.
Okay.
What are we doing? We leaning?
Do a little frame
Alright.
grab and move on.
Yeah. I'll
Okay.
zoom in a little bit. And getting on and getting on. Boom.
Nice.
All right, you got it.
Mm-hmm.
Okay. Up and out. Up and out. Maybe a Plesionica shrimp there. A little bit of outcropping rock I see coming up. Rock. It's a lot. Rock. Rocks on a steep slope. Maybe too much sediment for coral. I mean, for corals. They could set up on these rocks and have the nice upwelling current come up. Let me see what samples we got here going on.
Yeah.
Yeah, no, so they get the votes. So, yeah,
What's in there?
That's where the sponge is.
Yeah. Roger. Okay. We've been out of the corals for a while. Besides that pen.
Alright, so I don't want to update this because it's definitely some format.
50 meters away. Mm-hmm.
You can change the time for when the event's actually happening.
Crinoid, primnoid,
That's the same.
astroschema.
It's a primnoid with a big crinoid, so it's that's fine. I mean, this is it. This is what we've seen all day with this crinoid and ophiuroid.
That's it.
This primnoid coral. Enhance.
Okay, so, alright, so I'll
Coming in. There's your crinoid.
Look at that mouth.
There's
That's
your
cool.
protostar back there. It's going to move its
Oh,
arms,
we
we
got
can see.
a beauty shot of that. So
Right there, yeah. Something nipped that little star. It's regenerating its two of its arms. See that white stuff? That's just a skinny little arm that's coming back. All right.
Oh, that's interesting. Look at their hold fast. That's cool too. Like on the dead branches.
The
behind.
Siri, yeah, right. Cool.
Is that what they're called? Siri?
Siri.
I don't know. I mean,
I got nothing.
whenever you're ready to go.
All right.
We give you a call and
Okay.
Yeah, I feel like the astroschema is just like brushing things. And I don't know if it's because
Yeah.
we're here or if that's what it does all the time.
That's what it does all the time. Yeah. We're out. Look at that. Lexard with it too.
Mm-hmm.
Iridogorgia. There's a fish. Urchin.
Ooh.
Really small.
What kind of fish is
That's a
that?
big one.
Is that the big-eyed one? No. It's different.
I don't know what that is, I can't see.
There's Iridogorgia. There's some corals down here.
Mm-hmm.
Alright.
All right. Yep.
Another bookstore.
Yep.
Essentially, it's just a trash.
Oh, well, we're not coming up more. Just saying.
Yeah, that's right. Yeah, that's 4 miles. That's 8 miles. And then 4 miles.
Seapen? Yeah. Umbella?
Yep.
Umbellula?
Wait, is it? No. Oh, yes, it is. Sorry. Excuse me. Balula. Look at you.
I know.
Deep sea biologist.
Repetition helps.
Yeah, it does.
Be a repetition
does.
person.
Do it every day.
Not managing.
Steve is also really good at kind of explaining things off the scene if you're lucky enough to like get in a room with him in the library and stuff.
Hello.
Hey, you wanna
Hello.
come up and join us?
I
Hang
would
out?
love to come up and join you. I have a plan. So we will. Ooh. No, it's good. Look at that base. Sorry. You see
That's
that?
a massive holdfast
We
there
can-
for what fell there. It's a
I mean, you
bamboo.
can see what fell. Yeah, that's wild.
Do you want to look at it? I normally would look at it, but I don't need to. Same.
Okay.
It's pretty massive.
There's an Arena Gorge in there too.
Lasers on?
Sorry, I'll tell you my plan after we look at this holdfast. This is crazy. Look at that base.
Lasers are on. There they are.
It's all
It's hard to
about
see them.
that snow travel.
There we go. Monster. Yeah.
Oh my
Coming in.
goodness. You know, I think the rubble we were seeing was this, right? All over the seafloor? They were old bamboos, no?
One was an Alexandria.
You sure?
ISO.
Alexis thought so.
No.
But not all, not all.
I just some of
Yeah,
that looked
first
really
one
big
was
and thick. Look at that little baby Regiborgia.
Yeah.
Mhm.
There, Cap. Good flying today, sir?
It's a monster. 10, 20, 30 centimeters. 25 centimeters across. That's big.
It's just huge.
That's large. You can see there's
So
a
what
dead
we're looking-
bamboo laying next to this.
Timmy's got it.
Partially buried.
So that's 4 miles up to them.
What's your plan, Randy?
8 miles
Well, pending your
miles from there.
approval, the plan is to get to waypoint 8 and come on up. And then we have a pretty light basket, so I think processing should be pretty straightforward. Hoping fast because we have about
Ready.
an hour and a half to 2 hours of mapping and then we're back in the water. So,
2 hours?
yeah, so by the time we come up, it'll be an hour. And then, right, and then 2 hours of mapping and then an hour deployment. So 4 hours from whenever we leave, we'll be back on the bottom-ish,
Yeah.
between 4 and 5 hours. And we'll do another 12-hour dive. So you can look at which one. Want to click that off?
Mm-hmm.
Want me to get you anything from the mess?
Nope,
Drinks?
nope. I mean, we're almost there. I mean, we're
Yeah, yeah.
20 meters away, so.
I'll take a latte.
Okay, I'll cut out with
Do you want me to go
Carl's.
do it? One of you?
No,
Yes,
I'll go
please.
down
Let's
because
do this waypoint.
but I need to wait until, or if you want to break, go ahead. I need to wait till
Yeah.
we're too close to the end.
I don't know.
Yeah, beautiful sea pen with a little shrimp.
Do you think
Okay.
we'll be working at 1 o'clock?
It's not nighttime.
I think we'll be just about, you and I will be just about done.
John wanted to talk at 8 a.m. his time. I thought we would be.
Dude,
Vegan?
we're supposed to be
Yeah,
like
I
11.
thought we would be.
Yeah, that's
The basket would
I
be
don't think
coming
it's-
up.
that's-
But then I didn't know.
That clock?
This we planned that like a couple of days ago. Like,
Is that in there?
be like, we're gonna talk, we're gonna talk, we're gonna talk, and then we never do.
I mean, it's been a busy,
there? Are we there? I think
we
we're there.
kept a really busy ship.
No, and I'm happy.
I think
Like,
we're-
What is that?
it's really nice.
What?
Waypoint 8 is finished.
And I guarded my sleep today.
We
like
found
you said,
waypoint 8.
so-
I didn't, so I'm not, but that's okay. You and I, what we'll do is, this is coming up, we're on the experiment and get it all ready, so that all we have to do is
Yeah, we'll get everything filled.
Snout nose. Look at that
put it in
pointed
and then I
nose.
will go to bed.
Yeah.
Five versus five.
And then we'll let Steve and Deb stand
Five
away and I
versus
will go
five.
get some rest and I'll be okay.
Yeah, we're at waypoint 8.
We're at
I
we're
mean-
waypoint 8. Oh, and this is just sand. All right. Well then in that
Well, it's
case,
a very nice fish, little fish at waypoint 8.
let me,
Look at those eyes. A little frame.
Stay right now.
okay, well, for those of you who have joined us for this fantastic dive, it has been indeed fantastic. And just as we hit our last waypoint, it seems like we've left the rocks and we are here on sediment, looking at a very cool fish.
Beauty shop.
It's kind of like a chimera, but it's not really. What is this, Tim?
It's like it's not a chimera, but it certainly is like it, isn't it?
It sure is. I don't
Does
know
anyone
what this
on
fish
YouTube
is.
No, I
know
don't
what this
recognize it.
is?
I really don't. It's crazy.
We have all of our fish people.
Like a
Type of
snub
rat tail,
nose.
someone said.
It's not a. It's not
We
a
are
core
clearly not fish people. So, oh,
Oh,
here we go.
Bacilla
Corinus. Corinus.
Coelurynchus.
All right. Well,
Thank
There
you.
we go.
Unique.
Thank you very much.
Take your word for it.
We are very grateful for our friends on YouTube who are very into fishes. We love them too, but we don't know as much about them because we're here looking at corals and sponges.
In 2017, we had Luke,
ROV
who was really into fish, and that was fun.
Yeah.
Okay, so I can see as right before we leave, we'll introduce ourselves one last time and then tell you what our plan is. So I'm Randy Rogen. I'm the chief scientist of this expedition. I'm from Boston University. We also have
Tim Shank, deep sea biologist from the Woods Hole Oceanographic Institution.
and my favorite person to be on the air with, in addition to Anna Gauthier.
Da da da, Anna Gauthier.
Who is a finishing PhD student enrolled at Harvard University, who's co-mentored at Boston University. We have Team Texas on the ROVs. We have J-Rod and Cody. We have James on the Marine Tech. And we are so jazzed to have been on the seafloor with you. We have an aggressive turnaround time. We're on Titov seamount in the Pacific Remote Islands Marine National Monument, the US EEZ Howland and Baker unit. That is a mouthful to say every time, but so good. We are going to be back on the seafloor in about 4 hours, maybe 5. And so we're going to come up now and come back down on a different side of this very same seamount. So we'll see you very shortly back on the seafloor. Thank you for a great dive, and thanks for all of the fishy